Good afternoon. My name's Roger Cohn. I'm the director of the Life Sciences Institute. 50 years ago, on April 22nd, 1970, the first large environmental teach-in was held here at the University of Michigan, and that led to Earth Day. So this year we're celebrating the 50th anniversary of Earth Day, and in recognition of this, the LSI faculty chose the topic of the biological diversity for this year's symposium. Our co-directors, Cheng-Yu Lee, Bing Yi, and Ken Cadigan have put together a very interesting schedule of talks for the next 2 days, and I hope you all will enjoy these. To launch this year's symposium, I'd like to invite Dr. Marshall Runge, Dean of the University of Michigan Medical School and Executive Vice President for Medical Affairs, to make some welcoming remarks. Marshall?
Thank you very much for that kind introduction. And it's a pleasure to welcome you all to what is really one of the university's most important events, the Saltiel Life Sciences Symposium. Let me first extend my deep gratitude to all of the faculty members, scientists, trainees, and staff members who are advancing societal knowledge and health through discovery. We're living through a period of great problems that demand great attention of our best minds, and the need for good science has never been more apparent, and there's no better place than the LSI. I'd also like to thank the LSI's passionate supporters whose philanthropy and commitment have allowed the symposium to bring such remarkable scientific leaders to campus each year. The multiple challenges we face as a society shine a bright light on the importance of this year's theme, Broadening the Biosciences, Exploring Diverse Approaches to Biologic and Biomedical Research. In today's world, this includes all manners of diversity. You may have seen the article published in yesterday's New York Times on the paucity of Black microbiologists. And all of us are facing the challenge of bringing increased ethnic and gender diversity into the science of tomorrow. From a biologic standpoint, there are great advances that can be harnessed, breakthroughs in everything from subcellular imaging and the use of data science and advanced bioinformatics for analysis, and everything in between. This year's symposium features research grounded in novel perspectives and approaches that defy convention from a wide range of disciplines. How will these interact with each other, and how will that bring unimaginable creativity in scientific endeavors? I can't wait to see how, but I know that your ingenuity as researchers is precisely what we need to successfully emerge from this difficult moment in our history and these challenging times which science can bring solutions for. In addition to increasing the diversity of our scientists, we must also consider the diversity of our population who may benefit from the biological and biomedical discoveries you pursue. And I was pleased to see that as part of the agenda. And as has been said by a number of dignitaries, including most recently by Charles Drew, and who's the Dean of the Morehouse School of Medicine, we find, and this is a quote, we find ourselves at another inflection point where the status quo cannot be continued, and true change will require that government and industry make every effort to achieve the kind of diversity in our clinical trials that will allow us to understand variations in biology and how we can best target all of the approaches that you're thinking about for the improvement of health. So thank you again for directing your talents and creativity at these vexing problems and challenging problems. And it's now my great honor to introduce the namesake of our annual symposium. Professor Alan Saltiel is a renowned diabetes researcher and director of the Institute for Diabetes and Metabolic Health at the University of California, San Diego School of Medicine. Before joining the UCSD faculty, he served as the director of the University of Michigan Life Sciences Institute from 2002 until 2015. And the name of today's event speaks to the impact of his leadership and scientific contributions. It was in 2016 that this annual event was named the Saltiel Life Sciences Symposium, thanks to an endowment made possible by generous support from LSI board members, friends, and faculty. Dr. Saltiel will introduce this year's keynote speakers, and please join me in virtually welcoming back Dr. Alan Saltiel.
Greetings from San Diego. I'm very sorry that we were not able to convene today in person. I certainly treasure my trips to Ann Arbor for the symposium, and I regret not being able to be there to connect with all of you in person. So hopefully maybe next year we'll be able to do this. I think we all know this has been a hard year filled with many challenges for all of us. And I want to, before I start, want to take a minute to acknowledge the loss of someone who was a real champion of diversity, of someone very dear to me and a friend of the LSI, especially in our early years. And this is Professor James Jackson, who we sadly lost a few weeks ago to pancreatic cancer. James was an outstanding scholar, a leading social scientist who had a huge impact across many fields, including sociology, psychology, political science, and public health. He was also a terrific academic leader as director of the Institute for Social Research for many years. He was very helpful to me, advising me in the early years, especially of the LSI, on academic and scientific matters. He was a collaborator. And he was a friend, and he had an infectious optimism, and he kept many people on track over the years, including me. He will be sorely missed and really was a great loss. As you know, the keynote speaker of this symposium is named for Mary Sue and Ken Coleman, due to the generous gift that they made to the Institute several years ago to endow this lecture. Mary Sue was a legendary leader as president of the university. She was singularly responsible for allowing the LSI to emerge as the scientific force that it is today. She was, and she still is, a valuable mentor to me and a great friend. As you know, Mary Sue just stepped down as the president of the American Association of Universities after a successful, highly successful 5-year term. We owe her a great deal of gratitude, and it is really an honor to have the lecture named for her and her husband, Ken. Well, as you know, for the past few decades, biological and biomedical research has largely focused on approaches that prioritize a handful of model organisms or that center on specific subpopulations among patients when drawing on connections between human or mouse genetics and disease. And many of us have used these approaches as the foundation for our own research programs. And we know that these have led to significant discoveries about human health and disease. But with all that we've learned, what might we be missing? Over the next 2 days, we will explore what can be learned through new technological advances that broaden our scientific lens. All of our speakers today and tomorrow are developing innovative approaches to address major scientific challenges across the biosciences. And it is my honor today to introduce our keynote speaker, who more than anyone has worked to include diversity in studies of human genetics and health. Dr. Sarah Tishkoff is the David and Lynn Silfen University Professor in Genetics and Biology at the University of Pennsylvania, holding appointments in the School of Medicine and the School of Arts and Sciences. She is also director of the Penn Center for Global Genomics and Health Equity. She's a world-renowned expert in human evolutionary genetics, and her lab studies genomic and phenotypic variation in ethnically diverse Africans. Her research combines fieldwork, laboratory research, and computational methods to examine African population history and how genetic variation can affect a wide range of traits, why humans have different susceptibility to disease, how they metabolize drugs differently, how they adapt through evolution. This work has shed great amount of light on the need for greater diversity and equity in human genomic studies to ensure that biomedical research is optimized for diverse populations. Dr. Tishkoff is an elected member of the National Academy of Sciences, the recipient of many different awards, including the Pioneer Award from the NIH and the Burroughs Wellcome Fund Career Award. So please join me in welcoming our 2020 Mary Sue and Kenneth Coleman lecturer, Dr. Sarah Tishkoff. Sarah?
So, thank you for that incredibly kind introduction. It's a huge honor to be here today, and particularly to be introduced by you. I'm so excited. I'm just sorry that I can't be there in person. but I am really looking forward to having a discussion and answering questions after the presentation. At the Life Sciences Symposium. I just wish I could be there in person with all of you, but I'm looking forward to meeting you virtually and being able to answer some questions after my presentation. So I want to start by talking about some of the key challenges in human genomics research. The first is that we need to do a better job at characterizing genomic and phenotypic variation across ethnically diverse populations. We need to understand the evolutionary processes that generate and maintain that variation, and to better understand how gene-gene, gene-protein, and gene-environment interactions contribute to both normal variation as well as disease risk. The focus of my research is on Africa, and there's a number of reasons for that, but one of which is the fact that all modern humans originated in Africa. These red dots represent the locations of fossils of anatomically modern humans, the oldest of which is dated to around 300,000 years ago. After this origin in Africa, somewhere around 50 to 80,000 years ago, relatively small numbers of people, could have been in the hundreds, migrated out of Africa, giving rise to populations across the rest of the globe. So you can imagine that this demographic history has shaped the pattern of genetic diversity that we see in global populations, so that we see more diversity in Africa and less diversity outside of Africa. Now, after modern humans left Africa, they ran into archaic populations such as Neanderthals and Denisovans, who had been out of Africa for hundreds of thousands of years. And we now know that they intermixed, and we see evidence of integration of those archaic genomes into modern genomes. We think that somewhere between 2 to 6% of non-African genomes contain this archaic DNA. So part of what we're interested in is reconstructing human evolutionary history. But the other thing that motivates our research is to better understand the causes of health disparities. So for example, hypertension is more common in people of African ancestry relative to other ethnic groups in the US. Type 2 diabetes is more common in people of Native American ancestry relative to other populations in the US. And undoubtedly, there are one of the main risk factors for health disparities is likely to be socioeconomic factors, as well as structural racism in this country. But that's not to say that there aren't also some genetic risk factors as well. And one of the challenges is trying to differentiate the genetic from environmental effects. So this is from a perspective we published last year. We looked at all of the genome-wide association studies that were published as of January of 2019. And we looked at the composition of these studies in terms of ethnicity and found that about 80% of the individuals included in those studies were of European ancestry. Around 10% were of East Asian ancestry, around 2% African ancestry, around 1% Hispanic ancestry, and less than 1% everyone else. At other common databases like Nomad or GTEx, again, we see the strong European bias. And that's really problematic for a number of reasons. First of all, it impedes our ability to fully understand the genetic architecture that is genetic risk factors for human disease, and it could exacerbate health inequalities. Our ability to translate genetic research into clinical practice or public health policy could be incomplete, or even worse, it could be mistaken. It could result in inaccurate assessment of pathogenic variants in clinical genomic studies. So for example, one of the ways that people classify a variant of unknown significance as being pathogenic is by looking to see how common it is. So if it's very common, then it's less likely to be pathogenic. But most of the databases are from Europeans. So if we look at other ethnic groups, we may see that that variant actually is common. And that would change our assessment of whether or not it's pathogenic. Lastly, attempts to use estimates of genetic risk, for example, polygenic risk scores from European-based studies in non-Europeans, may result in inaccurate assessment of risk and lack of appropriate interventions. So for those of you not familiar with polygenic risk scores, these are calculated from genome-wide association studies where you look for an association between genetic variants and a disease. And if you see a strong association, then it is assumed that they are physically linked together in the chromosome. So you simply are going to sum up the effect sizes of all the genetic variants included in that genome-wide association. As an example, if someone did a GWAS for heart disease and calculated a polygenic risk score, and then you look in an individual who wasn't included in that study, and if you see that that individual has a lot of the genetic variants that are associated with high risk for heart disease, then they would have a high polygenic risk score and perhaps would get some sort of intervention or screening. So there's some debate about how useful these are, but the reality is they're being applied right now in the clinic. Now, this is actually a very nice study done by Alicia Martin and colleagues. And this was based on simulations, but there's empirical data to back this up as well, that when genome-wide association studies are done in Europeans and polygenic risk scores are calculated in those populations and then applied to other ethnic groups, they don't perform very well. In fact, they perform worse in the African descent populations. Now, why is that? One of the reasons could be that the genetic variants that are tagging or associated with the causative variant for a disease are not the same in different populations. So there may be a different tag SNP, single nucleotide polymorphism, in Africans or Asians or other populations, but they may have the same exact causal variants. But a second possibility is that there's a different genetic architecture. There could be different genes or there could be different genetic variants due to differences in their demographic histories and adaptation to different environments. And if that's the case, the only way we're going to know that is by doing GWAS in more diverse populations. And then lastly, there could be these different effect sizes could be due to differences in allele frequency, gene by gene, or gene by environmental effects, among other factors. So I want to tell you a bit more about Africa, which, as I told you, is the focus of our research. So there are over 2,000 ethnic groups in Africa who speak languages that have been classified into 4 major language families. In blue, we could see populations that speak Afro-Asiatic languages. In red are populations that speak Nilo-Saharan languages. These are typically populations that practice pastoralist lifestyle. The most wide-ranging language family is Niger Kordofanian, also known as Niger Congo. It originated in West Africa, and the biggest subfamily are the Bantu languages. Somewhere around 4,000 years ago, after the development of iron tools and slash and burn agriculture, which enabled them to cut down the forests and to produce crops and to sustain large populations, they migrated across Africa into the east and west and into the into the east and south and to the west and south, and they really altered the genetic landscape in Africa. And then in green, we see populations that speak with a click, and they've been classified as Khoisan, including the San hunter-gatherers from Southern Africa, and 2 groups called the Hadza and the Sandawe from Tanzania. So as I said, there's been a major bias towards non-African populations in the field of human genetics. So to help alleviate that bias, my research group and our African collaborators shown here have been doing fieldwork in Africa for the past 20 years or so. I thought I'd show you a little bit of what that's like. This is from Ethiopia. We're mainly studying minority populations that practice traditional indigenous lifestyles. They live in very rural areas, very remote, difficult to get to. So we have to bring a 4-wheel drive vehicle. We have to bring all of our supplies with us. This shows you the outside perspective, and this shows you the inside perspective, which is really crowded. This is from Tanzania. These 2 individuals in the upper right and lower left corners are members of the Hadza hunter-gatherer population that I mentioned. The woman on the upper left is a Nilotic speaking pastoralist. And here we can see the setup that we use for doing anthropometric measurements. And then in Cameroon, these are some of my colleagues and collaborators from University of Yaoundé, and other members of my lab who participated in fieldwork. Now, we're particularly careful to do this research in an ethical manner, and that means not just getting IRB consent from our universities, but going through a very detailed ethical review in each country, and ultimately getting research permits. But before we even get to that point, and that typically I would say takes about 5 years on average, to get to the point that we're actually doing field work, we spend a lot of time going to the communities, talking to them in terms that they can understand about what the goals are of the project and the risks, if any, and the benefits, if any, and we translate into the local languages. And then ultimately, after getting community consent, we get individual consent. We also think it's really important to return results to participants. So again, we put this into layman's terms, we translate into the local language, and give presentations and address any questions that people may have. And I can tell you they really appreciate this. And training and capacity building in Africa is really important. I feel honored that I've had an opportunity to train a number of postdocs and grad students, and ultimately the goal is that much of this research should be done in Africa. So in these regions, we collected blood, and from the blood we can obtain DNA and RNA and frozen plasma. We also get very detailed ethnographic information about diet and medical history if they have it. And then we had the challenge of processing samples in regions with no electricity. So in the past, we had to hook up the centrifuge to the car battery, but now we have a generator and we can actually set up the lab in the bush. And then we measure phenotypic diversity using non-invasive methods. So we get very detailed anthropometric measurements like height and weight and limb length and so on. We look at a number of cardiovascular, lung, and blood phenotypes. We look at metabolic function, lactose tolerance, glucose tolerance, for example, and infectious disease status when we can. And then we use an integrative omics approach. So we are generating whole genome sequence data at this point. We're getting information about blood proteins and biomarkers for cardiovascular metabolic traits. We're looking at epigenetic variation. We're looking at gene expression from whole blood. We're also doing single cell sequencing for immune cells. We're looking at the gut microbiome. And we're looking at using, together with Chuck Brandt, looking at using mass spectrometry to characterize the metabolome of the blood from diverse populations. And then we integrate this data with information about environmental factors and how they together are influencing complex traits and disease risk. Now, one of the reasons that these African populations are particularly informative is that while their environments are very different from each other, within any particular village, the environment is very homogeneous or relatively homogeneous, making it possible to try to disentangle genetic from environmental factors influencing traits. So, as an example, we could look at people who have the same genetic ancestry, but they're living in a rural or an urban environment and compare these traits of interest, or we could do the opposite. We can look at groups that live in the same environment, but they have different ancestries as indicated by the different colors here. As an example, the Fulani population in Cameroon have relative resistance to malaria for reasons that we don't yet understand. They don't have sickle cell mutation or the other common mutations that are known. And the San in Southern Africa have a higher risk for TB relative to neighboring populations. And again, we don't know the reasons for that. So this is from a study we published a number of years ago, but it remains one of the largest studies of genomic diversity in ethnically diverse Africans. We genotyped more than 2,500 Africans from 121 ethnic groups shown here. And while that is a lot, you could see that there's a lot of gaps there. So there's a lot more that needs to be characterized in terms of diversity, and there are many groups who are doing this. So the H3 Africa initiative, for example, is doing a lot of genomic analysis in Africa. So from this data, we can probabilistically infer ancestry. So these different colors represent genetic ancestry. And if we look at the non-African populations, each of these is made up of lines. Each line represents an individual. We can see that the people shown in blue are those who self-identify as European or Middle Eastern. Here we have people who self-identify or who are from India. Here we have people from Pakistan, from East Asia, shown in pink here, from Oceania in green, and from the Americas, shown in purple. But we can see from all the colors in Africa that there is a lot of genetic diversity within and between populations. So in fact, when we repeated this analysis just in Africa, and then we pooled individuals based on geography or language and show the proportion of these different ancestries, you could see how much variation there is in Africa, and that populations in Eastern Africa and Western, Southern, Central, Northern are genetically divergent from each other. And you could also see just from the faces here that there's a lot of phenotypic variation as well. And this is a result of the demographic history of Africans and adaptation to very diverse environments. So more recently, a former postdoc in my lab, Shawa Fan, did a study where, in collaboration with David Reich's lab, we did high coverage whole genome sequencing of 94 individuals from 44 African populations, speaking all of the major language families. And from that data, we can construct a phylogenetic tree. And so here we have the lineages that diverge most early in the tree are the San populations. Those are the ones that speak with clicks in Southern Africa. And then we have the so-called pygmy populations from Central Africa. And then the other populations cluster mainly based on geography or language. If we were to look at non-Africans, they would be up here in the tree. And we can use coalescent modeling to make inferences about changes in the effect of population size in the past. So if we look at this starting from the present, we're going backwards in time, we can see that the populations start to differentiate at around 250,000 years ago, which corresponds very well with the fossil data for the origins of modern humans. But we can also see that in the past, say 50,000 to more than 100,000 years ago, the two hunter-gatherer populations, the San and the Pygmies, had the largest effect of population size. And it was only more recently that they had a dramatic decrease in their size and other populations increased in size. So you should ignore these marks on the left because this method doesn't do well for estimating very recent effective population sizes. We can also infer the time of population divergence. So the San diverged earliest from other populations over 100,000 years ago. We could see divergence between the San and the hunter-gatherer populations around 70,000 to 80,000 years ago. And we see differentiation between the populations speaking other major languages somewhere between around 20,000 to 40,000 years ago. But even between groups like the Hadza and the Sandawe that speak with clicks and they live in Tanzania, they diverged around 20,000 years ago. And two San populations that live in Southern Africa that speak slightly divergent languages, they differentiated around 30,000 years ago. So there's been a history of a lot of substructure in Africa. More recently, we did whole genome sequencing of 180 diverse individuals. And I just want to point out that we were able to find so much novel variation. So we found 33 million single nucleotide polymorphisms, around 17% are novel, and many are predicted to be functional. And of those predicted to be functional, many are in regulatory regions. So now I want to tell you about phenotypic variation in Africa. So we looked at a number of anthropometric markers and cardiovascular metabolic blood markers. This is work that was done by Matt Hanston in my lab. And this is showing where these populations originate from. And then we integrated them with some genotype data in around 2,000 individuals. And these populations have very different diets. So the hunter-gatherers have a diet that's very heavy in vegetables and fruits and nuts and tubers. The pastoralists have diets that are heavy in milk and blood, to a lesser extent meat. And the agriculturalist groups have a diet that's very heavy in grain and maize and vegetables. So what we've done, I'm actually showing you here a number of anthropometric traits, and we're actually just looking at the females in the population. And we've color-coded the populations based on their subsistence pattern. So what we can see is that the shortest populations are the pygmy populations at one extreme, and then at the other extreme are the pastoralist and the agriculturalist populations. For BMI, we see that, again, the hunter-gatherer populations tend to have low BMI, whereas the agriculturalist populations have higher BMI, as do the Herero, who speak a Bantu language similar to the agriculturalists, but they adopted pastoralism. And it's actually, as part of their culture, it is considered desirable to be very heavy, particularly the women, and we definitely see that. If we look at waist circumference, again, lowest in the hunter-gatherers, highest in the agriculturalists and the Herero. Percent body fat, the same thing, lowest in the hunter-gatherers and highest in the Herero. C-peptide, we see variation, low in the hunter-gatherers, higher in the agriculturalist populations. For glucose, we don't see as much variation, but we do see that it's somewhat higher in the agriculturalist populations. So together with Chuck Barant at the University of Michigan, we did this project where Chuck did a targeted metabolomics analysis looking at approximately 700 lipids in 439 ethnically diverse Africans. And again, we then did a principal components analysis of that data, and we've color-coded according to the subsistence. And what you could see is that we can differentiate the agriculturalists here, and we can differentiate many of the pastoralists. There's more overlap between the hunter-gatherers and the agro-pastoralists. If we look at, for example, just the sum of different types of lipids, like triglycerides, for example, we see that hunter-gatherers consistently have very low levels, and the pastoralist populations have relatively high levels. But if we just look at 3 pastoralist populations that have different ancestries and they live in different geographic regions, we can differentiate them based on their lipid expression using principal component analysis. So that suggests that there may be genetic factors that are playing a role in lipid levels. So together with Hongzhi Li, we integrated this data with SNP genotype data and did a genome-wide association study and identified 314 lipid-associated genetic variants. Now, when we look at the expression levels of lipids, we could see that they tend to be very correlated. Not a surprise, those that are in the same metabolic pathway tend to be correlated. And then not a surprise that many of the genetic variants that we identify influence the levels of multiple lipids that are along the same metabolic pathway. But what we wanted to know is how much of the variance in lipid levels is due to diet and how much is due to genetics. So we partitioned that and looked at the amount of the variance due to diet, and that's shown here. And we can compare that to the variance explained by genotype, shown here. But notice that very different scales. This is going up to 0.2, this is going up to 1. We can more directly compare in this figure, and we can see that genotype explains around 56% of the variance, and diet explains only about 7% of the variance. I should say this is a work in progress, and we still have some additional analyses to do, but it's a very intriguing result. So lastly, I want to tell you about some of our studies of the genetic basis of adaptation. And one of the adaptive traits we're interested in is skin color. So in blue, I'm showing you melanin levels from populations around the world, and we can see that they're correlated with the UV levels. And it's thought that when modern humans left Africa and went to higher latitudes, that there would have been selection for lighter skin in order to be able to better synthesize vitamin D. This is synthesized in the skin in response to UV light. And at lower latitudes along the equator, there'd be selection for darker skin to prevent skin cancer and folate degradation. Now, almost all studies of skin color, genetics of skin color, were done in European populations, and we wanted to look at Africans. So the way we did that is we used a spectrophotometer that shines light. We look at under the arm to look at an area that's not exposed to sunlight. We can also look at the wrist if we want to look at tanning response. And based on the wavelength of the reflection, we can infer the melanin levels. And what we see is a lot of variation in Africa. So the San hunter-gatherers, who I told you have the oldest genetic lineages in the world, have the lightest skin pigmentation in Africa. And we see a lot of variation, with the most darkly pigmented people being the Nilo-Saharan-speaking pastoralists from South Sudan. Nick Crawford and Matt Hansen in my lab then did a genome-wide association study of a relatively small number of individuals, only around 1,500, and that is very small when you compare to what people are doing in European populations, looking at tens of thousands or even hundreds of thousands of people. But because of the large amount of phenotypic and genetic variation in Africa, they're particularly informative. So we were able to identify 8 independent loci at genome-wide levels of significance in 4 regions of the genome, and I'm just going to step through those quickly. So the strongest association was at a gene called SLC24A5. This was a non-synonymous variant that the allele associated with light skin color is at almost 100% frequency in Europeans. It's common in India and Pakistan, and it's also common in East Africa. But we wanted to know, did this variant originate independently in East Africa, or was it introduced by migration from out of Africa? So to address that question, we constructed a haplotype network. So haplotypes are how genetic variants are arranged along some short region of a chromosome. In this case, I believe we were looking at around 70,000 base pairs. Each circle represents a haplotype. The size of the circle indicates the number of people who have that haplotype. And this is like a phylogeny, because the circles that are close to each other are very similar to each other, and those that are more distant are very divergent from each other. And then the different colors represents the proportion in different ethnic groups. So what we see is that Europeans have this non-synonymous variant on one very long haplotype that is very common in the population. They seem to have a signature of recent selection for this variant. And if we look at the East African populations, that variant is on the same haplotype background. So what that means is that we think it was introduced back into Africa due to more recent migration from out of Africa. The next strongest association was at a gene called MFSD12. At the time we started this study, it didn't even have a name. It was just open reading frame, whatever. But it was given a name later based on homology to a group of transmembrane transporters that facilitate transport of ions, amino acids, peptides, among other substrates. Now, the only clue that we had at the time that this may play a role in skin pigmentation was a study of vitiligo, in which they showed differences in RNA levels in the depigmented and darkly pigmented skin in people with this condition for this gene. And then at the same time, my collaborator, Elena Oancia, I found that there were very high levels of RNA for MFSD12 in melanocytes relative to other skin cell types. So we found 2 independent associations at this gene, one at around 8,000 base pairs upstream, which is at a MIT-F transcription factor binding site. MIT-F plays a role in melanocyte development. And this enhancer region interacts with the promoter of MFSD12. The other variants are within the MFSD12 gene. There is one non-synonymous variant, but we actually think these are more likely to be influencing gene regulation. So for the variants within MFSD12, we can see that the variants associated with dark skin, shown in purple here, are only present in African populations. These are African Americans and people of African ancestry in the Caribbean. When we look at the variant upstream of MFSD12, we can see that the variant associated with light skin is common in Europeans, in East Asians, in India, in East Africans, and very common in the San. The variant associated with dark skin color is common in Africa, South Asia, and Australo-Melanesia. In collaboration with Kevin Brown and Stacy Loftus at NIH, Kevin Brown had generated transcriptomes from 106 melanocytes with mixed ancestry. 15 had high African ancestry. And then a graduate student in my lab, Derek Kelly, looked at the correlations between gene expression and ancestry. And he found that people with high African ancestry had very low levels of MFSD12 gene expression. And if we look at the correlation with variation at that upstream enhancer, the variant associated with dark skin, again, is correlated with lower levels of gene expression. Then another former postdoc in my lab, Marcia Beltram, used luciferase expression assays to test whether this region was acting as an enhancer, and she showed that it does indeed have very strong enhancer activity. And then in collaboration with Mickey Marks and Shanna Bowman, Mickey knocked out this protein, MFSD12, in the melanosomes obtained from mouse. And he showed that when he knocked it out, that he saw an increase in melanin levels, the dark pigment. So that's very consistent with what we were seeing, that people with darkly pigmented skin have lower levels of this protein. But what was a bit more mysterious was the fact that the protein does not co-localize with other proteins that are located within the melanosome. Rather, it co-localizes with proteins that are in the lysosome. And that was unexpected. It means that this protein is somehow indirectly impacting melanin levels in the melanosome through a mechanism that we still don't know, and we're currently working on to try to better understand. And then in collaboration with Bill Pavin at NIH, he knocked out this gene using CRISPR-Cas9 in the agouti mouse, and it had a really dramatic impact on the pigmentation of this mouse. So the mouse loses the pheomelanin, that is the yellow-red pigment, and then you see this underlying gray color. Now, in the lab at NIH, they noticed that these that the CRISPR knockout mice had a phenotype that was really similar to a mouse that was in the Jackson labs called the grizzled mouse. And it was first identified in 1948 and characterized in the 1960s. And then the gene was mapped in the 1990s to a region that is a 2-centimorgan region that's synthetic to MFSD12. So they sequenced the genomes of these mice, and they found that the only difference they had was a 9 base pair in frame deletion in exon 2 of MFSD12. So I like to say that this is one of the rare examples of using a human GWAS to map a mouse mutant. So I was very proud of that. So in collaboration with Yun Song and Ethan Jewett, we looked at the gene genealogy of these regions. And so the way to look at this is the lineages that don't have this open circle are the lineages that are associated with dark pigmentation. They have a variant associated with dark pigmentation. And the lineages with the open circle have the variant associated with light pigmentation. And in this case, the ancestral allele is the one associated with light skin, and it's very old. We see that both of these are very old, on the order of 1 million years. The third region was at DDB1. DDB1 plays a very important role in DNA repair in response to damage caused by UV. And in people who have mutations in this gene or in the genes coding for proteins that interact with DDB1, they have a disease called xeroderma pigmentosum. They can't be exposed at all to sunlight because they're not able to repair the DNA, and they then get skin cancer. But what does this have to do with pigmentation in humans? We don't know, but what we do know is that this gene is the gene that causes the color in tomatoes. So it is influencing pigmentation, we just have to still figure out how. So again, we found two independent associations, one about 40,000 base pairs upstream of DDB1 that is interacting with the DDB1 promoter. And then we found another association that was really a number of variants and very strong LD over 100,000 or so base pairs. So we can't say for sure what the functional variant is, but this is one of the candidates that's within transcription factor MIT-F binding site, or I'm sorry, a transcription factor binding site for MIT-F, and is known to interact with the promoter of DDB1. Again, we look for correlations with ancestry, and in this case, high African ancestry is correlated with higher levels of DDB1, and the variant associated with dark pigmentation, again, is associated with higher levels of DDB1. And again, we show that this, the variant in the region about 40 kb upstream is acting as a very strong enhancer element. We then use a statistical test of neutrality called Tejima's D statistic, and it's basically based on, it looks at the allele frequency distributions. So for example, it's looking at how many singletons there are in the population, how many doubletons, and so on. And this blue line is showing you what you would expect under a neutral model. So when there's been recent positive selection, you expect to see an excess of rare variants. And if there's been balancing selection, if diversity is being maintained, you're going to see an excess of common variation. And what we see is a very strong signature of recent positive selection. So we see very negative values. These are from the some of the 1,000 genomes populations. And this is in the Chinese, this is in Europeans, and it's over more than 500,000 base pairs. I think it's even more striking when we look at the heterozygosity level in this region, because the variation is basically wiped out in Asia for over 500,000 base pairs when we compare this to Africans, for example. And when we construct the haplotype network, this is over about 200,000 base pairs, we see that in non-Africans, there's this very long common haplotype, and that's a signature that you expect with recent positive selection. And when you look at the gene genealogy, it's really striking. You see this rapid coalescence in the non-African lineages that have the light allele with coalescence around 60,000 years ago, corresponding with the time of migration out of Africa. So it looks like when this variant was introduced outside of Africa, the light allele, it swept to almost 100% fixation. The last region where we saw an association was at OCA2 and HERT2. These were already known to be associated with skin color. But we found variants that were independent from those that had been previously found in HERT2. So in Europeans, variants in this region are associated with skin color and blue eye color. But as I said, these arose independently, and then we found a non-synonymous variant in exon 10 of OCA2. So these two variants in HERT2 were shown to be correlated with gene expression of OCA2. And the OCA2 variant, we could see that the variant associated with light pigmentation is common in Europe, in the San, and in some Southern Asian populations, and in East Asia. But it wasn't correlated with gene expression, rather it was correlated with alternative splicing. So the variant associated with lighter skin pigmentation was associated with a shorter transcript that was missing exon 10 that codes for this third transmembrane region of the protein. Now, it's still in frame, so a protein is being made, but it's missing that transmembrane region, and that's going to have a major impact on the protein. Now here we see a signature of positive selection over HERC2, but in OCA2 we see a signature of balancing selection in some of the African, in the San population in particular. And if we look at the gene genealogy, again, we see a signature that could be consistent with balancing selection, so maintaining both alleles. We see a very old time to most recent common ancestry, more than 2 million years old, and that's unusual for the human genome. So lastly, in conclusion, we found that in half the cases, the ancestral allele was the allele associated with a light skin color, and that the age of the derived mutations was typically older than the origin of modern humans. So what does that tell us about the evolution of human skin color? Well, this poor ape lost its body here because it had a autoimmune disorder, but we could see that the skin color is relatively light. Anthropologists have speculated that this may have been the ancestral state in humans, and that when our ancestors moved out of the forest and into the savanna, there would have been selection for more darkly pigmented skin. But I wonder, given that we see both light and dark alleles segregating in Africa for a very long period of time, there could have been variation in Africa, and there could have been variation in different regions of Africa. And the other thing that was really interesting is that the only place where we saw the variants associated with dark pigmentation outside of Africa was in South Asia and Australo-Melanesia, 2 regions of the world where we see very darkly pigmented people. And what this tells us is that these variants were brought out of Africa during the out of Africa migration, and then were maintained in those populations due to natural selection. So in conclusion, there's an urgent need to characterize more diverse populations. We need to better understand how and why the genetic architecture of complex traits differs among ethnic groups, and how well polygenic risk scores translate across ethnic groups. That's going to require integration of diverse types of data, and we need to understand gene by gene and gene by environment impact on complex traits and on polygenic risk scores. So with that, I will end and just thank the many people who contributed to the study and the funders, and I am happy to answer any questions that you may have.
Okay, Sarah, thank you very much. I'm giving you a big hand on behalf of everybody. I think it's time, we have a little bit of time for question and answer, and I want to open it up. I'm going to ask attendants to use the Q&A feature on Zoom, which is on the bottom of the screen, to submit questions.
And while they're doing that, can I just mention that I noted a mistake when in my presentation, I misspoke that the variant at exon 10 in the OCA2 gene is not a non-synonymous variant. It's actually a synonymous variant, but it does impact alternative splicing. So just caught that when I re-listened to this.
I guess, I don't know, can you see the questions, or should I read it?
Yeah, actually I can see it. And the question is, what % of the heritability for skin color is explained by the GWAS hits that we found? So now I'm trying to remember, but if I recall, I think it was on the order of, I'm thinking around 40%. I'm honestly forgetting. It certainly doesn't explain all of the variation. I'm 100% certain. So this is a complex trait. It's not perhaps as complex as a trait like height, which could be influenced by thousands of genes, perhaps hundreds or maybe thousands. There are more genes of stronger effect for skin color, but there are many genes. So we're still in the process of identifying them. And one thing I could tell you that if we ignore the genome-wide significance level and we just ask the question, let's just look at the top, I don't know, 1,000 hits, right, in the GWAS, and say, what are they doing? What genes are nearby, and what's the function of those genes? There is a strong enrichment for pigmentation. So we're finding a lot of interesting candidates that just don't reach that genome-wide level of significance, but there's a lot more there. So one says, It's known that vitiligo is associated with protection from melanoma. Do your genes act in melanoma? The only thing we don't know, I did see one study. So after we published this, some other studies came out and they found that MFSC12 is associated with skin color in South Americans and animals. I think it was in dogs and horses or something like that. And then there was a study that suggested that it's playing a role in risk for melanoma. So I know that at least one study has come out. Okay, so now there's another question here that says, do we have any evidence that Neanderthal or Denisovan genes contribute to any distinct phenotypes or diseases in the human population? So yes, there is some evidence. Now keep in mind that Neanderthals and Denisovan integration is mainly outside of Africa. We do see some Neanderthal ancestry actually in East Africans. That's part of another study we're working on right now. But outside of Africa, people have shown that the regions that have Neanderthal and to some extent Denisovan ancestry may play a role in certain phenotypes. One of the classic examples was adaptation to high altitude in Tibet, for example. So there was a variant introduced from Denisovans that was present in Tibetans at high altitude. Some have suggested it could play a role in risk for type 2 diabetes and other immune-related traits. Now, in Africa, that's almost certainly happening as well, that there probably was integration with an archaic population, but we just don't, it's harder to find because we don't have any reference genomes yet from archaic populations in Africa because the DNA just doesn't hold up very well. It says, do you have enough sequence data to give some insight for current day African American individuals to find more information about their ancestors from Africa? Are the sequences available for this purpose? So I would say not yet. I can see right now people have tried to determine ancestry of, say, African Americans from Africa with limited success, I would say. Now, part of the reason is that they have relied on SNP arrays that are biased. They were typically variants found in non-Africans, and they tend to be common variants. Now, also because the West Africa, the source of the African slave trade was predominantly along the western coast of Africa, the largest proportion was actually from Angola. And we have almost no information from Angola. So part of it is that we're just missing information. As we start doing more sequencing, I do think as you start to find more variation, we're gonna find some that are specific to certain ethnic groups in Africa. So it might be more possible, but we also know our prediction is that given the history of the slave trade and the fact that we don't have assortative mating post-slave trade in the US, that you're going to see as you slide along the chromosomes, you're going to see ancestry from different parts of Africa. It's not going to point to just one region in Africa. It's probably going to be from different regions in Africa, but that will be interesting to look at in the future. So let's see. Do you find any evidence of differential rates of splicing, especially affecting function and phenotypes across the major population groups? Very good question. So we don't know. We have that example from that variant that we found at OCA2 that we think is impacting phenotype. And it certainly shows very different frequency in different global populations. I can say that one of the studies we're doing now is we are looking at gene expression from blood. And from that, we will be able to look at splicing patterns, and then we'll be able to integrate that with genetic variation that we find. So it's an outstanding question that we're looking at. We want to know differences in gene expression within and between populations and how they're impacted not just by genetics, but also by the environments in which they live. So I hope to have that answer soon. Have you looked at differences in repetitive elements within your whole genome sequence sets for changes, given that these may show more rapid variation in changes than steps? Not yet. That's a great idea. There are some, as people who look at whole genome sequence data know, the sort of the low-hanging fruit is to look at the single nucleotide variants and small indels. It's harder to identify structural variants, and it's challenging also to identify these repeats, but there are some algorithms for doing that. So that's certainly something we'd be interested for looking at in the future. And then there's something about any speculation as to the explanation for the missing heritability here. So again, going back then, I think referring probably to the GWAS for skin color, I mean, I think one of the obvious reasons for the missing heritability is that there are other genes out there. So there are a lot more variants that are playing a role. There could be possibly some gene by environment interaction that's going on here as well, or some epistasis, but I do think there are just other variants that we're missing. And, you know, I want to point out that we wouldn't have been able to easily find the variants that we did if we were looking at Europeans, because there's just no variation. There's been strong selection for light skin color, and you just don't see the variation that you see in Africa. So you have to have that underlying variation. Now, at the same time, there are going to be variants in Africa that are fixed. They're at like 100% frequency associated with dark skin color. So those are more challenging to find with the GWAS, but one of the ways we're looking for those is we look at regions of the genome that are very different frequency, or have population-specific variants that differ in lightly pigmented and darkly pigmented populations. And that has also turned out some really interesting candidates by looking at that as well. And then there's one that says, do you think the diversity of pigmentation genes might reflect changes in isolation over time? Possibly. I think, you know, one of the outstanding questions for me is what is causing the variation in Africa for skin color? Because there's a lot, and it's not due to recent admixture with non-Africans. This is naturally occurring variation. We see some evidence that lighter skin color has evolved in the San in Southern Africa. It's possible that darker skin color has evolved, for example, in the Nilotic populations, maybe because of pleiotropic effects of these genes. Like, they have many important effects. And so that could play a role, but there's also been so much migration and you know, genetic drift. One of the difficult things is differentiating the different evolutionary forces. So again, that's something we can look at in the future. Uh-oh, I think I answered all the questions.
Well, I just have one more question to throw in there, which I won't type because I'm just listening.
Okay.
In your metabolic and diabetes studies in Africans, have you confirmed or reproduced some of the GWAS hits that have been found in Caucasian populations, especially in the ones like in the Finnish populations or the Sardinian populations, et cetera?
So what we're doing, we have not, our study, the groups that we study are not groups with disease such as diabetes, though we do see diabetes in some of the individuals in some populations. It's actually something that we're trying to figure out right now, because when we see diabetes in some of these groups, they're not associated, they're adult onset, but they're not associated with high BMI. So their BMI is like, you know, 19, 20 on average, but they have adult onset diabetes. So we're trying to figure out what's causing that. And we do, however, have information about glucose, standing glucose, metabolic pathways, and absolutely the goal is going to be, because we're generating a very large data set right now, both for the metabolic data with Czech-Barrant, but we're also getting much more genome sequence data. So when we do that, we can do association studies. And one of the questions we want to address is, do we see replication of hits that we see outside of Africa, in non-Africans in Africa? We don't want to simply ask if the variant that shows an association outside of Africa shows one in Africa, because if that variant's just not tagging, we just need to look in the region to see if anything else in that region associates. So that is something that we've started to look into a little bit for some lipid traits, and we will do more of, but I think it's a really interesting, outstanding question.
One more question.
Okay, it says, oh, you're hitting me with the tough questions. Is there any way to look for epistasis among your current GWAS hits? And if so, is there any? Oh, that's really hard. It's always hard to look at epistasis because we have so little power for that. So the truth is we haven't looked yet. And I think it's one of the things that when we're embarking on a study that's going to be a much larger GWAS. And when we do that, that is one of the things that we want to look at. But as I'm sure the person asking this knows, it can be extremely challenging to distinguish epistasis, but it's something we'd be interested in looking at.
Okay, well, thank you very much. I want to thank you again for just a terrific and very exciting talk. And thanks a lot for coming to give this talk and to
Oh, it's my
participate in the
pleasure. Just wish we could be
symposium.
there together, but
Yeah.
maybe another time we'll have an opportunity. It's been my pleasure. Thank you for inviting me.
Okay, well, I want to turn over the symposium now to Bing Ye, who I think is going to introduce our next speaker.
Thank you, Alan, and thank you, Sarah, for a very nice talk. I'm a faculty member in the Life Sciences Institute and a co-organizer of this symposium. Now, it's my great pleasure to introduce Dr. Barry Trimmer, our next speaker. Dr. Trimmer is the Henry Bromfield Pearson Professor of Natural Sciences at Tufts University. He received his BA in zoology from University of Cambridge in England, and his PhD in neurobiology from University of Cambridge. He then came to the US for his postdoctoral training, first with Ed Kravitz at Harvard, and then with Janice Weeks at UC Berkeley. As a neurobiologist by trade, Dr. Trimmer studies the sensory processing and locomotion in the caterpillar of Manduca sexta, also known as a tobacco hornworm. What makes Dr. Trimmer's work extremely unique is that he uses the insights learned from the locomotion of the soft-bodied caterpillar to design softer robots. Dr. Trimmer is the founder and director of the Tufts Biomimetics Device Laboratory. And he studied at the journal Soft Robotics and has been the editor-in-chief since 2012. After Dr. Trimmer's pre-recorded presentation, he will join us in the Q&A session. And for those just joining us, please use the Q&A feature to add questions during the talk for Dr. Trimmer, and to use the thumbs up icon to upvote questions that other people ask to prioritize these questions. Now we'll turn it over to Dr. Trimmer's pre-recorded presentation.
Okay, I would like to first thank the University of Michigan and the Life Science Institute and the symposium organizers for putting together such a wonderful set of talks. And today I want to cover a broad subject. I want to introduce the idea that we can start to use biology to make better machines. And it's not an obvious way of thinking about the world, but there have been huge advances in molecular biology and biology in general, huge advances in technology, computer architecture, and the speed of computers, and massive advances in material science. And these together, I'm going to try and convince you, are going to allow us to make completely new technologies. And one of those new technologies is living robots. And what I want to do is describe some of the work from my lab which illustrates these different points. My lab is only contributing a very small amount to this. There are labs all over the world performing this sort of research. But I hope I can show you examples of what I mean and the important aspects of how you build much, much better robots that are going to be able to do things current robots are incapable of. So I am a neurobiologist by training, and my laboratory is working primarily on this animal here, which is Manduca sexta, the tobacco hornworm. And I'm going to spend quite a bit of time talking about how soft animals control movements. We also are doing quite a lot of work on what caterpillars sense, how they feel the world. The work that we've been doing on the caterpillars has led to this research program of trying to develop soft robots, robots that are completely conformable. My lab actually works on these non-pneumatic crawlers. They don't use any pneumatics or fluid flow. They don't blow up. Don't require compressors. So they could potentially be completely autonomic machines. And we are looking at the way that the robots might be controlled, this so-called high degree of freedom movement. And the last topic that's of research in my lab is to sort of skip over the idea of having to create new materials to build soft robots and actually grow robots out of living tissues. And that's based on insect cells, and we also are looking at the fuel for robots. So I'm primarily going to be concentrating on the top topics there, the ones that you see, a little bit on controlling high degree of freedom movements. But the take-home message here is that biological robots will have to use soft materials. And if they're using soft materials, we're going to have to develop novel control systems to make those robots work the way we want them to. And so those are the two topics that really come together. And the talk is organized around this general trajectory. We're going to start talking about biomechanics and how materials are important. Then we'll talk about neuromechanics, the neural control of machines, of animals. And then we're going to do a little bit about how you might design soft robots based on these principles. And finally, we'll talk about how you might see a path towards living robots. So the first part is to think about materials. And we don't think about materials enough when we're talking about machines. And that's because most machines are, of course, in this region of materials. And what we're seeing here is a plot of Young's modulus, which is a measure of the stiffness of a material, against its density, both of them on a log scale. And our machines are all pretty much on the top side of this slide. They're up on that top right. We build them out of extremely durable, extremely tough and stiff materials. Animals, of course, are towards the lower left side of this plot. Animals have to grow, and they are made of predominantly soft materials. And although things like the whale and humans and cheetahs do have hard skeletons, it's a relatively small proportion of the whole animal. For a human, it's 15% of your body weight. The rest is squishy and soft and liquid sort of materials. Now, why on earth do we build machines only out of hard materials? Part of the reason is that those machines can do things we cannot do. And so we're able to exploit these amazing properties of materials that are not present in animals. But the other side is a little bit of a bias. We tend to think of soft materials as somehow deficient. They're not quite as good as hard materials. And the first thing I want to do is convince you that that's not true. And I'm going to turn to my favorite sport, which is MotoGP motorcycle racing, as an example. And I'm going to show you a short clip of Marc Marquez, the world champion MotoGP rider, in 2016 at the Japanese circuit Motegi, double ring circuit. And he's approaching turn 11, which is a right-hand 90-degree turn at 300 kilometers per hour, slowing down to 75 kilometers per hour. You might have noticed he was weaving a little bit. And if you look close up at what was happening, you can see that the rear tire is not actually touching the ground. So while he's braking, it takes about 2 seconds for this to happen, he's entirely on the front wheel on a contact area of approximately 5 square centimeters, so probably about the size of a credit card. There's about 2,500 newtons of force on that area, which converts to approximately 700 pounds per square inch of force acting on that tire. And that's immense force, and yet we accept that a soft material is the way to do it. And in fact, Marc Marquez, even talented as he is, could not actually do that, make that performance and get around that corner without the compliance of soft materials. So in engineering, soft materials are amazing things. And in biology, we already know that they're amazing things. So if we take a look at a large octopus moving through a transparent tube, I think you can appreciate the amazing ability of these materials to deform. The octopus can completely squeeze itself down. So soft materials are pretty impressive things. And one of the reasons we don't use them extensively in moving machines is because soft materials are complex and thought to be difficult to control. So I like to think of materials in a slightly different way from the traditional way that an engineer might. In a stiff, so-called articulated system, something like a limbed robot with stiff parts and joints, or an animal that has a very stiff skeleton, we can imagine that all of the different things that contribute to the control of its body can be overlapping and interacting components. So within the environment, we have these structures, we have commands, which would be the nervous system, or in a robot, it would be the microcontrollers. We have some sort of peripheral nervous system, which would be the senses feeding back into that control. And we'd have material components, a relatively small part. We don't build the materials to be part of the controls, usually. In a soft system, such as an octopus, I'd like to argue that the proportions change dramatically. It's an embedded system in which the materials actually form a large part of that control. And perhaps, although structures are also still important, you can imagine that the commands, the brain, is actually able to control these movements with fewer commands. This is counterintuitive. Any engineer thinking about this is likely to say the more complex the materials, the more squishy something is, the more control commands that you need in order to make it work. And I'd like to put forward an idea that that may not be true, that may not be the case. I actually think that materials can be thought of as mechanical calculators. And that because animals have always had these materials, the nervous system exploits the properties of the materials in order to control the body. So let's look at an example. Here is a linear elastic material, a simple rubber band, if you imagine. This is actually the material we build our robots out of. And it's quite straightforward. The stress, which is equivalent of the force over an area, and the amount of stretch, so the strain of this material, is completely linear, pretty much, with a little bit of wiggle. So for every force that you apply, or every stretch that you apply, there is the corresponding value. Now, that's not much of a calculator, okay? It's a simple linear relationship. But most materials, most elastomers, and certainly most tissues, are much more complex. So if you do the same sort of stretching, so here is something being stretched by a force, and then shortening again, you can see that they don't follow the same path. It depends which way you're pulling the material or releasing it as to what forces you see. And this is referred to as a hysteretic work loop. And so what's happening here is that there is energy being dissipated. It's actually not returning as much energy as you put into it in this particular case. And this is a form of linear viscoelasticity, pseudoelasticity as we call it. And there's another property as well, which is much of these materials change the relationships between those factors as a function of time or the rate at which they're being moved. And I think you can see right away that even these equations are much more complex and in fact are a form of computation. So as long as the control system knows, and I'm saying that in sort of inverted commas, that there are these relationships in the materials, they can be exploited to make the commands much more straightforward and easier. It moves all of the controls to a higher level of hierarchy. So how does this relate to tissues? We can look at muscles and see how muscles perform under these conditions. This is a muscle from the animal I work on, the caterpillar. And if you stretch it out, it follows one particular curve. And as you release it and shorten it, it comes back along a different one, this typical hysteresis loop. And if you now stimulate the muscle, so you're stimulating it continuously, making it really, really actively trying to shorten, it becomes a much, much stiffer material. And you can see that stimulated muscle now has this much steeper relationship and a much bigger difference between the two ways of developing force. So muscle's stiffer when it's stretching, it's softer when it's shortening, and it's essentially changing from one material to another. Under these particular conditions, this is acting as a so-called viscoelastic damper. It is a clockwise work loop. It is losing energy, it's dissipating energy. And in an animal, under these conditions, it would be acting to absorb shocks or to absorb energy in some way. So, if this is true, how on earth can an animal use this? What might it be doing to use these sorts of properties? And that's where we can start to look at the neuromechanics of Manduka. So this animal is not entirely unique, but is one of the few animals in which we can make a direct relationship between the movements and the neural commands that are controlling those movements. So we can actually measure what is going on in this creature. If we look at a cross-section through a single body segment shown here, we can see that there's a lot of complexity to it, and the muscles have been reconstructed in this micro-CT scan. And you can see that they attach to the body wall, and here's a particular muscle we call PPRM. It actually controls this leg, the proleg, and causes it to retract when that muscle contracts. And you can see that because we know where the muscles are attached, we can actually mark the so-called origin and insertion of that muscle, and we know where they are, and we can monitor from outside the shortening of that muscle. So when we take a look at a caterpillar performing just normal behavior, freely moving around, we can track the position of that muscle, the length of the muscle, continuously as the animal moves around. So now we actually have a measure of the strain cycling of that muscle. The other thing that we can do with Manduka is to record the neural activity controlling the movements of the muscle or the stiffness of the muscle. So in this video, you're going to see that I've got a recording electrode stuffed right into the top of the muscle here. And this is recording the electrical activity that the muscle shows when it's activated by a neuron. And below, you can see the spikes, which is the electrical activity of the muscle in response to the neuron controlling it. This gray line in the middle represents the video frame that you see in the movie above. So when the movie starts to move, you'll be able to see that as a body segment moves, there is firing of that muscle. So I'm just scrolling through. And here we can see the electrode. The muscle is in that segment. It's controlling the movements of this leg. And if I advance forwards, you'll be able to see that before the leg actually moves, there's a whole lot of activity happening. It's not yet moved. But that muscle is now being activated, developing force, attempting to shorten, but certainly becoming stiffer. So now we can actually monitor electrical activity and the length of a muscle while the animal's moving around. And it turns out that in Manduka, with very few exceptions, each muscle is controlled by a single neuron. So in theory, we can record all of the neural commands going out to the different muscles in the body. So we can now take that muscle and look to see what sort of forces develop and how it performs under natural conditions. So we take it out of the animal, and we know the movements, we know the EMGs, the electrical activity, and we can simplify it a little bit by, instead of giving this complex normal shortening, just simplify it to a series of ramps. So we can shorten the muscle, measure the forces while it's shortening. And we can stimulate it in exactly the correct timing for normal crawling in the animal. And you can see here is a muscle, this is a muscle in a dish in which we're stimulating it and making it shorten. So now we can ask the question, what happens? What does this work loop look like in an animal that's actually moving around and behaving? If you remember, I showed you work loops of passive muscle when it's not activated and active muscle that's being stimulated. But here, of course, it's being stimulated for a brief period during a cycle. And this is the resultant work loop. And I think this is absolutely fascinating because it shows you that during a single cycle of movements, the muscle is acting as a damper. It's a clockwise direction work loop. And then it's being stimulated and acting like a motor. It's developing extra force and putting energy out into the world. Here it's an actuator. So this is absolutely wonderful because it actually is something that can be exploited. And not only can you get a work loop of this particular form, but the animal can change the timing and the duration of the stimulus applied to the muscle. So let's see what happens then. Here we see a variety of conditions. On the left is showing the effects of varying the stimulus timing. As you advance the timing from its normal position that would be happening in the animal, you can see how you change this work loop dramatically. It becomes a much more dissipative damper. Equally, if you keep the timing the same but change the duration of that stimulus, you have these complex figure-of-8 work loops that change dramatically depending on the duration of the stimulus. This, I would argue, is a computer. This is something that is a property of the mechanical system that the nervous system has evolved with and must always be using this as one of its ways of controlling the body. So, do we know how it actually works for a moving animal? And the answer is no, not yet, not in detail, but we do have some clues. And I can show you what happens when the animal's doing two different things and how it changes the activity of this muscle. So we have these flexible arrays of electrodes that we can implant into the body wall and record from muscles. And we have multiple channels, so multiple electrodes, and we can pick out of these traces the activity of individual muscles as the animal's moving around. And here, what I'm showing you is the activity of a particular pair of neurons during 3 different steps. So this is step 1, 2, and 3. And the step is for that particular body segment, and it starts with the pink line. That's when the leg is starting to lift up, to the next pink line, which is when it comes down. So this is one cycle of a step in one body segment. And this is a muscle in the back, in the body wall of the animal, which is helping to shorten body segments as the animal crawls. And what you can see is when it's crawling, by and large, the activity is before and during and somewhat after the initiation of the swing phase of the leg movement. And we'd expect that the work loop would be something like this. Now, if we have the animal climbing vertically, so the same animal, we've just flipped its substrate around so it now has to climb, it actually climbs with more or less the same gait. It doesn't change its gait, which is very different from what we would do. But you can see there is a change. There's a change in the timing of that activation of that muscle to the end of the swing phase, so as the leg is being put back down again. And here we'd predict to have a very, very different work loop. And our hypothesis is that this is the way in which the animal is able to compensate for the increased loading during climbing, and that it is exploiting this work loop property of the muscles. Okay, so we've learned something from the neuromechanics. Can we apply any of this to building better robots? And there's many, many aspects of what I've been talking about, the materials, the neural commands. I want to focus on just one part, which is how we actually design these robots and how we can build a control system based on what we've learned. And before we do that, we've got to think about why would we want to build soft robots? It doesn't seem obvious that soft robots are going to be a huge boon to mankind, but I would like to argue that they are, and these are the main points. First, they're going to be robust to large and sudden forces, so very, very robust. You could drop them out of an aircraft and they would just bounce and not break. And I don't recommend doing that to the $3 million fancy robots that are made by Honda or other companies. They will break. They can change their shape and their size, so they can access what we call denied spaces. This might be people who are trapped. It could be internal channels in some sort of machine. It could be a wiring system in a major mainframe computer. So this is something that would be very, very attractive to have. Very important aspect is they can be made safe for use in human and natural environments. All my life, people have been promising me a robot. It'll help me do things in the house, and I don't have one. I have my vacuum cleaner, and that's about it. Part of the reason is that those very stiff materials and controlling the system makes it very, very dangerous, because if there's no control, that robot could actually hurt you. Imagine if you make them out of soft materials. You could have these robots moving around your home. They could move through the forest. They don't damage things. They can interact with the world without actually breaking things. They have better potential for interfacing with tissues. That's quite obvious. If you try to get a steel bar incorporated into tissues, it just won't happen. The tissues do not like extraordinarily stiff materials. Tissues like to be interacting with compliant materials. So if you want to make a prosthetic, it would be good to use soft materials. That's also true for surgical robots and endoscopy. You'd want to have soft, pliable things. An aspect that I think is going to make a big difference, because economics can drive a lot of these changes in technology, is that these could be really, really cheap to produce, because you don't need huge foundries to make titanium and steel or ceramics. These soft materials can be made in very, very energy efficient, low temperature ways, and they could be extremely easy to make. They're also going to be light, very low density, so that could be very, very important for all sorts of roles. And if you now have soft materials, you know how to control them, you have this huge array of materials that engineers can select from for different purposes, including biological materials. So now you can build robots out of biopolymers, make them biodegradable, sustainable, and they can use biofuels, which are extremely safe. And we'll come to that towards the end of the talk. So I think these combined factors make it highly likely that we're going to be developing a whole new generation of machines to fulfill these roles. So in my lab, we've made a small start on this, and we're doing it primarily to try and understand a little bit more about the way the animal moves. But in doing so, we've learned that we could probably make useful machines. And the approach that we've been taking is to either print the robots directly, so we can use soft materials in a multi-material printer, and make all sorts of different shaped robots, different numbers of limbs. We can also use our 3D printers to make to make molds that we can cast materials in, so that we can now use a whole range of materials depending on what we want from the robot. And then we assemble the electronics and make our robots in this way. The big problem for most people in soft robotics is trying to find a decent actuator, the equivalent to a muscle. Many people use pneumatics, which means you have to have a big compressor somewhere and blow air or maybe fluids, hydraulics, into the system. We actually use what we think of as a muscle mimic. And this is a shape memory alloy coil. And although it's not a perfect mimic, I just want to show you that it has some properties that make it resemble muscle. So you've seen this work loop of the muscle before over here, this figure-of-8 work loop of a Manduka muscle. You can actually control this so-called nitinol coil, bimetal helix, and make it generate similar, a little bit sharp and not quite as well-defined, but you can make it develop these complex work loops by stimulating with sort of sub-threshold stimuli and controlling just what the temperature of this shape memory alloy is. So they're similar in some ways to muscles. And here is a little video, a little short description of some of our robots. And I think what you can see that with these shape memory alloys, these robots actually are very, very lifelike. This video was actually developed for a robot that we designed to roll so it could move really, really quickly and get around some of the limitations of slow actuators. So once you build the robot, you've now got to figure out how to control it. And you can take what we've learned about muscles in animals, and you can also come at it from a more engineering perspective and say, what do we need to do to control it? And two of my colleagues who worked as postdocs in my lab have started to develop two very, very different approaches to controlling these robots. So first, Takuya Umedechi is designing control systems based around central pattern generators. And CPGs are known to exist in nervous systems and control rhythmic motions. And the way this works is that there are a series of segmental oscillators that are coupled together and influencing one another's activity to generate motion. And so it requires sensory feedback to adjust the phase of these oscillators. And it really is affected by the interactions with the environment, local sensing. And in fact, a lot of the interaction between the oscillators can be generated purely mechanically. You don't actually need to have the oscillators directly influencing each other through their wires. You can do it mechanically, and he's been able to show that you can generate crawling and inching movements with these robots with this sort of control system. The other approach is from Vishesh Vikas, who's at the University of Alabama now, and he has taken the view that we're never going to be able to completely describe the physics of a complex soft material device. So we shouldn't be trying to model it in order to control it. We can model it in order to design, but not control. So the approach is what we call model-free control. And it's based on the idea that we have control primitives. We discretize the states that the robot can be in, and then learn how to transition between states and what those transitions are going to entail. And the different transitions give you different sorts of behaviors. And you can optimize for those behaviors. So in this particular case, these robots have two states that each end can be in. They can either be sticky or they can be slippy. And so you can get these different movements by switching between sticky and being slippy. And those are defined as these nodes. And when you do the transitions between these nodes, you find that you can get crawling by these three sorts of transitions described here in this array. Or you can get inching with a different set of transitions in this array. So this is quite a powerful technique. It's also very robust. So if you get damage in a system, it can quickly figure out new transitions to be able to move around in the world. And this just shows you a little bit of some of the gates that these types of controllers can give you. Again, you can see here that the transition from sticking to the ground or slipping is achieved entirely through the material properties that vary around the circumference of the foot of this robot. So although we're using shape memory alloys, that's not a very well practical sort of thing to do because they're rather slow and unpredictable. And if we want to make a practical robot, we might want to use a more conventional motor. So we've been developing these motor tendon platforms. This one here is using two motors, one at each end, pulling on a tendon. So again, they're working like a muscle. They can only generate force in tension. They cannot actually push on anything. Again, this robot uses the transition of sticky to slippy feet, and is actually the fastest soft robot currently being made. It's the fastest around. The other thing we can do, and I'll show you those other robots, but just to introduce this other idea, softness is not an easy thing to define sometimes. A big bowl of cookie dough is very soft, but it's not very elastic. When you push into it, it stays deformed. It doesn't respond back very much. Also, softness is a bulk property to do with the relative size of the forces compared to the stiffness of the material. And what we find is that you can actually change the stiffness of the material by forming it into a foam. And what we see here is that these elastomers or polymers can be made even more foam-like and therefore much, much softer than they would be otherwise. And so that's really a very, very powerful thing. And in fact, we can use very, very stiff, hard materials and create structures that make them perform like soft things. So you're aware of steel wool or cleaning coils like this. You can also make metal foam. And of course, what those are doing is moving the overall bulk metal properties down into this soft range while maintaining some of the properties of abrasion resistance, toughness, that type of thing. So we've built foam robots. And this first one, Slabo here, built by Nico, is designed to be complicated because we wanted to learn control systems from a complex movement. And it's simply a big block of foam that's actuated by motor tendons. And you can see the complexity of its movements. We've also built a robot that we call Monolith, which is a reticulated foam. It's got 3 motors inside. This is completely untethered. It doesn't require any wires or compressed air hoses or anything like that. It's got its own battery on board, its own control system, and its own motors. And we think that the Monolith is approximately 0.5 meter in size. It's quite big. You can make it large because it's incredibly light. It's just a reticulated open-cell foam. And our plan is that something like this might be really, really helpful in surveying very, very difficult terrain, perhaps even for extraterrestrial use, perhaps on Mars. So finally, how do we transition to living robots? And this is something that's still, very, very in its early stages. But a lot of people are thinking in this way, thinking about how we use tissues and cells to make a machine. And our approach is to use insect tissues. Most tissue engineering is being done for biomedical purposes, so it's being done on vertebrate cells. It could be done on human cells. It's usually done on mammalian cells. The problem with mammalian cells is that they require very, very strict environmental conditions of oxygen and carbon dioxide and temperature and pH. And insects have an open blood system, no vasculature, and they're extraordinarily tolerant of environmental conditions and temperature. So we would argue that if you want to make a robot that can move around in a natural environment, you might want to use an insect cell. So the way we're doing this and approaching it is to take eggs or developing adult tissue during metamorphosis, and you isolate myocytes. These are cells that will become muscles, and you culture them. You create a bioincubator in which you put scaffolds that the cells will recognize. They will join those scaffolds, proliferate, differentiate, turn into different tissues, and you can imagine that this would be a module for one of these robots. Now, I'm not advocating that we're going to make new species. That's not the idea here at all. It's very, very important to me that we don't grow brains, and we don't need to. Our microcontrollers are perfectly adequate to control our machines. So I would imagine that we would simply clip in a microcontroller. It would be powered by a very, very low power battery, just enough to give control commands. All of the power for the muscles is generated by them from their own natural fuels. You also probably don't want to have a reproductive system in these living robots. And you can imagine making these biocomponent robots. So what sort of thing have we done? Well, you need to be able to integrate all this. You can't just make an actuator by itself. Living systems are very, very integrated. So all of the materials they need and the fuel they need and the chemistry that they need is present. So we need to be able to integrate. And you can imagine having some sort of fuel source, the muscles attached inside this machine, and there's chemical transfer from the fuel to the muscle. It's important to recognize that lipids, fats, are as energy dense as kerosene and gasoline. They are extraordinarily high energy hydrocarbons, but they can be turned into mechanical energy at low temperature, our body temperature. And so they're not being burnt in the same way that we do in a car engine. So they're relatively efficient, very safe. They're producing low levels of CO2. They're perfectly safe. If we were in a packed auditorium here, everybody would feel safe and comfortable with the levels of CO2, moisture, and everything else. If we replaced everybody with an internal combustion engine, it would not be a pleasant place to be. We wouldn't want to be here. So I'm pointing out here that the biofuel is an extremely effective way of making a machine last a long time in a safe manner. So when we culture these insect cells, we can measure all sorts of aspects of their metabolism. We find that here over 34 days of culturing, they actually aren't generating huge amounts of lactate. They do not seem to use all of the glucose. And I have to point out that these cells are not being refed. Unlike vertebrate cells, we just put them in culture and leave them, and they grow perfectly fine. And you can see that they have triglycerides, which they use up and internalize. They've got plenty of fuel in these cells. So, what do they look like? We can produce the cells, just raise them at room temperature, and for months they grow normally, and you end up with these contracting networks of muscle cells. Extremely robust. You could not do this with vertebrate cells. Vertebrate cells would have been dead within hours or at least a day of the conditions that these cells survive for months and months. The challenge now is converting these contracting networks of cells into actuators, and there's lots of ways of doing this. We would like them to do it themselves. We'd like to know enough about the way they differentiate so that they will produce a fully formed muscle. But in the meantime, before we can do that, we're sort of forcing them into making constructs. So we constrain them with the growing chambers that we put them in. They can be in these troughs or in loops, and make as many cells as possible into one location and kind of hope that they work together. It's not ideal, but it does create muscle-like tissue that can contract together. So the cells are contracted together. It's nowhere near as good as muscle. It's not as well coordinated, but that's where we're now working. Okay, a lot of challenges there. So to finish, let's just think a little bit about where this goes and what the advantages are going to be. I've touched on a whole list of advantages, but one of the things that strikes me is that so far our robots have failed in most circumstances that we would like them to be able to help. And in a disaster situation such as this earthquakes, where people are being buried, no one has yet been able to get a robot that could go in there, find a person, and save their life. It's not yet been possible. The robots get stuck. They do not go through rubble very well. They're also very expensive, and they're easily lost. I would like to imagine a whole army of very, very soft, incredibly cheap, less than $100 for each robot. You could have 10,000 of these in an emergency trailer, and you drop them all over the emergency sites. They burrow down, and when they detect the presence of life, they emit a beacon that you see on your cell phone. You now know where that person is buried. That's the sort of thing that I imagine the robots could do. So we call that search and rescue, exploration. You could use it in surveillance, reconnaissance. You could also use it for mapping minefields, and because you don't care about the robots and getting them back, you can just leave them there out in the minefield. Now you know where the mines are. So that's one area that I think is important. Another is we've already mentioned, diagnosis and surgery, prosthetics, biocompatible drug delivery. You can just imagine all sorts of things that robots could do inside the body to be beneficial. And something that isn't obvious, but we think might be very useful, is this idea of having a tree-climbing silent robot. And you could use it for biodiversity surveys. You could use it, deploy these all over the forest, and you don't really disturb the natural environment or the animals and different species in the canopy. Right now, the way we do it is in very, very difficult conditions, very haphazard climbing or erecting cranes in the canopy. Robots could do this very, very easily. And I think one of the most immediate uses of soft robots is going to be in space vehicle and extraterrestrial applications, as shown here. It's very difficult to repair a large antenna or solar array on a space vehicle because they are so delicate. They're like the thickness of a drinking straw and thin as paper. Very, very easily damaged by anybody going out there trying to repair them. We can well imagine that soft robots could be packed, very low density, extremely light, and then they would blow up again as they get up into space, and they can crawl around on these structures and help to diagnose problems and repair them. Okay, just some ideas. I'm sure that you have more of your own. So finally, I want to thank all the members of the lab. I'm showing here most of the current members and people whose work has contributed to this talk. They're all very, very talented people with different backgrounds in biology, engineering. And I'd also like to thank our funding agencies. Most of this work has been funded by the National Science Foundation, with some contributions from the Keck Foundation and DARPA. And with that, I'll take some questions. Thank you.
Well, thank you, Barry, for an inspiring talk. Now we can answer some questions. Attendees, please use the Q&A feature at the bottom of your screen to submit questions. And also, if someone already submitted a question, you can upvote it by use that thumbs up icon. We'll answer questions with the most votes first. So this question is from Hua Yang Cha. He says, he or she says, amazing. Is there any specific reason to choose an insect as a model? And what's the reason for that specific species, meaning Manduca?
So it's a very good question. So in terms of the neuromechanics, it's a really fortuitous finding actually that the caterpillar only has single neurons innervating each muscle. So it allows us to translate spike patterns of neurons generated by the nervous system into specific movements that the muscles generate. So it's a lot simpler to sort of understand the relationship between those things. So that's in terms of the neuromechanics. I think that I would love to be able to work on the octopus. I think the octopus is probably the most amazing control system for soft movement in an animal. But there you've got approximately 50 million neurons in every single arm, just in the periphery, not even in the central nervous system. So it's really, really difficult in an animal like the octopus or other mollusks to be able to relate the central neural control to the actual movements. And Manduca, the caterpillar, allows us to do that. In extending a little bit to why use insects in the living robots, the tissues, because they don't have a vasculature, because they don't control their body temperature, well, they do behaviorally, but they don't actually have to control their body temperature to survive very much. They can exist at a huge range of temperatures. The cells are semi-autonomous. They basically survive very, very well without lots and lots of support systems. So they're also very tolerant of the chemical conditions. They can use alternative fuel sources. Their insects are all extremophiles. They live in a very, very difficult environments, very selective environments with rich nutrients in one part, but with absolute deficiencies in others. So their tissues are very, very good at responding to this and metabolism. So I think that's one reason they make excellent cell sources for living robots.
Right, excellent. We have another question from Nicholas Teets. He asked, just prior to pupation, caterpillars go through a wandering phase where they stop eating and actively search for a place for pupation. Is there any evidence that the neural control of locomotion is plastic and changes throughout the life cycle?
Yes, there is. So we tend to think of the crawling locomotion as being very stereotypical, and we imagine almost a hardwired system, but I think that's false. I don't think that's the way their nervous systems set up and function. Although they look as if they have the same gait as they're moving around, they actually have very, very small but probably important variations in their stepping patterns that happen continuously while they're moving around on different substrates. And as you point out, they do change during metamorphosis where they go through this wandering. It's actually an interesting field of study because when they wander, they walk and walk and walk and walk. And if you take a caterpillar that's not wandering and you remove part of the brain called the central complex, it will start to walk and walk and walk and just keep doing it. So that part of the brain must control the suppression of walking. When the animal wanders, if you remove that part of the brain, you get the opposite effect. So there's a whole lot of questions that we could try and ask and answer in looking at the ways in which the animal can adapt and change its nervous system, both developmentally and behaviorally, depending on the context.
It's very interesting. Now, the next question is from our LSI Director Roger Cohn. He asks, what is the state of materials research to make a synthetic material with the properties of muscle?
Thank you. That's a very, very good question, and something that is important for the field. There are numerous materials that have muscle-like properties. The ones that have been developed, I think people are very excited by, are electroactive polymers. These are materials that when you deliver electricity to them, they change shape or deform or change their size. Usually it's a size change. It's been difficult to integrate these into robots because they have a lot of drawbacks that become very evident. So some of the materials require enormous voltages in order to actuate, and that can be a problem for all sorts of applications. Other materials rely upon the movement of ions in an electric field, and so those are all wet. And because they're wet, it's hard to integrate them with modern day electronic systems. But there's progress being made all the time in this area, and we're waiting for the perfect material to be developed that we can just plug straight into our soft robots. Right now, I think people are using a variety of compromised materials.
Right. So the next question again is from Huayang Cha. To transfer electrical signal to software robots, you will need some wires, which is made of metals, like copper, right? So do you have any idea how to make the wires much lighter?
Yes, so it's actually fairly straightforward to make fairly soft or compliant conducting materials. There's a huge number of people working on this. I think some people might be aware of John Rogers' work in this area of trying to make stretchable electronics. So that can be done. Some of the approaches to make serpentine systems that can be stretched so they don't actually break the wires or deform them. Others actually have materials that can stretch and conduct electricity. My vision for this is that the internal wiring of the soft robot is going to be built into the scaffold that you use to seed the growth of all the cells. So the cells will grow upon a biopolymer with an already arranged set of wires, which are the equivalent of the neurons in a living animal. So they would just have to carry very, very small current. They're not actually powering the muscles, of course. They're only powering the signal to tell the muscle to do something. So I think it's completely doable. Yeah.
All right. If no more questions, now thank you, Barry, for a very exciting talk. And
you're very welcome. I'm enjoying the symposium. Thank you.
Thanks. Yeah, we'll move on to the next speaker. All right. Now, it's my great pleasure to introduce our next speaker, Dr. Shai Revzen, who is here at the University of Michigan. Dr. Revzen is an associate professor in the Department of Electrical Engineering and Computer Science, and also the Department of Ecology and Evolutionary Biology. He holds a PhD in biomechanics from UC Berkeley and a master's degree in computer science from the Hebrew University in Jerusalem. And he did his postdoctoral work in robotics in the GRASP lab of the University of Pennsylvania. At Michigan, he runs the biologically inspired robotics in the dynamical systems lab, which aims to extract the mathematical principles of biological motion control and applies these principles to robotics. Dr. Revzen is also a co-founder of a company developing novel electrocardiac diagnostics. Dr. Revzen is giving a live presentation followed by a Q&A session. And so just a reminder for the audience, please use the Q&A feature to add questions for Dr. Revzen during his talk, and to use the thumbs up icon to upvote questions to prioritize them. Now, I'm handing it over to Dr. Revzen for his talk.
Thank you so much. So this is a great pleasure to speak to you all, especially after my dear colleague Barry, whose work I've followed for many years. It's very exciting. So today I'm going to talk a little bit about how the physics of slithering can be used to teach multi-legged robots to walk, and maybe talk a little bit more broadly about those physics. My lab, the birds lab, which has nothing to do with birds because I work on legged locomotion, studies a whole bunch of different animals. And as our host mentioned, we do a lot of mathematical modeling and then carry these ideas forward into a variety of robots. I won't be talking about all of these today. I'll be talking about a very restricted subset. So I'll be talking some about nematode locomotion and cockroach locomotion, about one specific modeling and optimization procedure, and 3 of our hexapedal robots. And to summarize my talk in one slide so that you can snooze for the rest of it, this talk is about the following: that in the sense of being governed by a connection, which is a kind of mathematical object that I will shortly describe to you, multi-legged animals and robots are like slithering snakes and swimming nematodes. That is, they follow the same kind of physics. They are very, very similar. We aren't. This is very non-intuitive for humans. It takes actually at least 3 legs on the ground to cause this change in the underlying physics to appear. And so it's pretty counterintuitive for us. So what is this connection? Well, the idea of a connection comes from geometric mechanics. This is a whole body of theory that was developed in the 90s, in part by University of Michigan's own Tony Block and many other luminaries like Gerald Marsden. And it teaches us something interesting about friction-dominated motion and about other things, but specifically for friction-dominated motion like slithering or swimming in low Reynolds number. It teaches us that there is a connection between the body velocity, which in matrix form would be for a matrix G, G inverse G dot, and the body shape and body shape velocity. So the connection object is what's denoted by AFR here in this equation, and R being the shape of the body, and R dot being the rate of shape change. So the important thing to note here is that for every specific shape of the body, there's a linear relationship between the body velocity and the rate of change of shape. This is very, very different from what we have in general Newtonian mechanics, where the body's momentum plays a role. And so it primarily governs situations where friction and viscosity completely remove momentum. Our goal is to try and build data-driven models of this connection, but to try it out, we did the following toy problem. So consider the nematode Caenorhabdus elegans. What is it optimizing when it's swimming? Is it trying to swim as fast as possible or as efficient as possible? Well, we could get at this problem, which is a notoriously hard problem for biologists because we can't generally make the animals move differently from what they do. By first using some classical physics model for the swimming, in this case, the Purcell swimming model, then we can calibrate using some animal data, which we got from some colleagues in the University of Pennsylvania complex fluid labs, to make sure that our model is a good predictor of how the animals actually move. And you see the graphs here have pretty good correspondence, rotations not so much, but definitely translation pretty good. Then with this, we can build a data-driven connection model and use this model to test the consequences of swimming gait changes. Now you wonder, well, why not use this top model, the Purcell model? Well, because that's not our goal. Our goal is to use a data-driven connection where we don't have a physics model. We only have reasons to expect that the physics will be governed by a connection. So how do we do that? Well, we partition the cycles of motion into bins, and for each bin, we reconstruct this connection equation using 2 parameters, a parameter that goes along the cycle theta and an offset from the cycle delta, build an expansion of that, which is just Taylor's expansion, and we end up with a multilinear regression, the sort of which most people in the life sciences are quite familiar with. So this is just a generalized linear model for each point along the cycle. And once we built this model, we can go ahead and have our data-driven model of the physics. And then we can use it to figure out what the optimal motion is by first using regression to get the data-driven connection model, and then use that model to improve the motion. So we now use the model that we derived to make a small incremental change in how we move. Now we have a new motion. We identify the model around that motion, rinse and repeat. And if you wonder how well that works, well, it works really, really well for Purcell swimmers. In the paper that's marked here below, we put in 9-segment swimmers, initialized them to a whole bunch of different initial conditions, also studied them at various levels of noise. And what we find is that the results of the optimization are robust to initial conditions and generate the same cost of transport. So basically the same efficiency of motion as you would sort of expect. The interesting observation though, is this is a very high dimensional optimization. This is an optimization that is occurring on a 264 dimensional space of possible motions. And usually these kinds of optimizations are very, very hard to do in practice. So what did we get for nematodes? Well, those of you who love nematodes might recognize that right here in the middle is what a real nematode does. If the nematode was trying to move as fast as possible, it would do the motion up top. And if it was trying to be as efficient as possible, it would do the motion down below. So what nematodes actually do, a lot more similar to energy efficiency, minimal cost of transport, but not quite. But the interesting question is, can we use this to optimize robot motions? And the answer is yes. So here you see the same protocol that we applied to the simulation, now applied to a physical robot where we don't have a model of the physics. This 5-segment robot starts out with a completely ineffectual motion, and within 10 cycles of optimization, you are literally seeing all the data that was required for the optimization. The robot reaches a pretty convincing serpentine gait. So this is very, very fast learning in terms of learning how to move, much faster than any other methods we've encountered before. So 9 is pretty convincing, but still going sideways a lot. And iteration 10, we're doing a pretty convincing serpentine motion. So what about legged locomotion? This is where I started from. Well, we could try and build a connection for the legged locomotion model. And if we tried to build a connection for legs and assume Coulomb friction, so we have legs here, they contact the ground, they interact with the ground through friction, a bunch of things break down. We basically don't get the right form for the equations. But if we do some unspeakable violence to Newtonian mechanics and Coulomb friction and replace the physics with an ansatz that has a mixture between viscous and Coulomb friction in the friction coefficient, we get an equation with the right form. And when we tried this on a robot data, we got this. Whoops, my apologies. We got this. So a very good correspondence between the robot data in blue and the model data in green. And we see the same thing for our second hexapedal robot. And this was too good to be true. This is way better than we had expected. So we asked our colleagues to give us data for a 4-legged robot, the Hammer robot from Harvard. And this shouldn't even work for a 4-legged robot because it doesn't have 3 legs on the ground at the same time. And yet it still works pretty well. It still works pretty well. So at that point, we had to check this on animal data. And what you see here in red are the raw model predictions. in green are model predictions that are filtered to have about the same spectrum as actual animal motion, and in blue, the true animal motion. And here the R squared is not that good. It's only about 0.7. It's still way better than most other cockroach models I've worked on, and I spent most of my career looking at cockroach locomotion. So at this point, we have this puzzling situation because we have a result that's too good. That's too good to be true. The physics are not right, but the predictions are really good. And so we went to the Army Research Office and got some money to build a new robot. And this new robot has force torque transducers on every single leg, and we will be able to figure out why it is that by using the wrong physics, we got the right answer for these hexapedal robot. But there's an important point here that, that one of my students really decided to drive home. And that was that we are doing our optimization without actually having a model of the physics of the robot. And so my student went ahead and collected some sticks, and he took these sticks and just chopped them off and attached them to our motors, and then made a robot. And this robot can learn how to move with our method in about 20 minutes. Why is this working? Because this robot too is friction dominated. It doesn't build up momentum. It turns out that any system that does not build up momentum can learn how to move fairly quickly. And when we can learn how to move fairly quickly, we can also start looking at a more interesting problem, which is how to learn a library of maneuvers that enable us to navigate anywhere we want to on the ground. So let's say we wanted to move and rotate. That's a three-dimensional space of rigid body motions in the plane. Let's try and construct that out of elementary motions that are different. So we could do, say, motion A and motion B, and then motion A and motion A one after the other, or then maybe A and B, or B and A, or B and B, and so on and so on. Look at various horizons of this construction and try and pick a collection of motions that together give us a good ability to navigate in the plane. And if we do that for our swimming, we start out learning some maneuvers. So you see what happens first cycle, second cycle, third cycle in terms of colors. In a few iterations, this algorithm learns how to move, and then we disable the robot. We generate an injury by locking the joint that's marked with a black dot. And with a few additional cycles of learning, we recover the ability to move around. So this is pretty spectacular. Usually we think of in robotics that when we add degrees of freedom to a robot, controlling it becomes harder. But what we see here is when learning is very quick, it goes the other way around. We have the more complicated robots, and I'll show this in the next figure. We have the more complicated robots that have a larger number of joints, and they achieve a better solution faster than robots with fewer number of joints. And the interesting thing is then when we injure the robots here, the new learning process is faster than the original learning process. So those robots that have enough degrees of freedom, and in this they're very much like animals. Animals have many more degrees of freedom that they can actuate than they need. They can recover from injury much better. And so this gives us some interesting insight how using these simplified physics for high friction locomotion that came to us from slithering and low Reynolds number swimming, we're now able to build legged systems that not only learn how to maneuver very rapidly, but they also recover from injury very rapidly. And with that, I'd like to acknowledge my students and colleagues and funding sources who have participated in this, point out the tragic end of our stick robot that it met right before COVID shut down our experiments, and point out that what we've learned is that the Stokesian or geometric mechanics govern multi-legged systems and not just slithering ones, and that we can use this to rapidly learn how to move with both legged and slithering robot bodies. And with a wink to Barry's talk before me, we now also have methods that will work for soft robots as well. Thank you.
Thank you, Shai, for a very interesting talk. Now, we'll answer some questions, and attendees, again, please use the Q&A feature at the bottom of your screen to ask questions. Well, our first question is from Barry Trimmer. So, very exciting. Could this be adapted to quasi-static caterpillars?
So there's an interesting question: what happens with animals that develop a little bit of momentum and then lose it? I think in the case of quasi-static caterpillars, this actually directly applies. The only thing that requires the additional paper we're working on right now is that for soft systems, not all the degrees of freedom are actuated. You can sort of think of them as having a whole network of degrees of freedom, only a few of which are actuated. And in setting up our learning algorithm to work with that took a bit more work, but we've basically resolved it. We just haven't finished the publication.
He has a follow-up question. So not a friction dominated, not really a slithering.
Yeah, so quasi-static and attaching to the substrate works fine as well. It's not, so systems that attach to the ground and then a part of the body moves and attaches and then another part of the body moves are also Stokesian. These are piecewise holonomic systems, as they're sometimes called, and they are a subset of the systems described here.
Thank you, Shai. I don't see other questions now. So thanks for participating in the symposium.
My pleasure, thank you.
Oh, just 1 second. There's one more question that just came in. Now, have you dealt with obstacles?
Have we dealt with obstacles? So if we apply the idea of using maneuvers like I have shown here, or maybe this is a better example. So imagine you took this worm. It has 3 different maneuvers that it can use to basically rotate and translate. So now by composing those together, you can navigate around obstacles. Basically, we're putting together an alphabet with which you can navigate through space in an arbitrary way. So in that sense, we already have one kind of solution to obstacles. And the other kind of problem you get with obstacles, if you have some sort of persistent interaction with them, as long as the dynamics are still friction dominated, we could probably learn a solution to.
All right. Thank you, Shai. Thanks again. And we'll move on to the next speaker now. All right. So our next speaker, it's my great pleasure to introduce our next speaker, Dr. Elizabeth Tibbetts. Dr. Tibbetts is a professor in the Department of Ecology and Evolutionary Biology here at the University of Michigan. She received her Bachelor of Science from University of Michigan and her PhD in Neurobiology and Behavior from Cornell University. She did her postdoctoral research in the Arizona Research Labs at the University of Arizona. Dr. Tibbetts studies behavioral evolution, focusing on how communication systems and social behavior co-evolve to shape the way animals look, think, and act. She uses social insects, primarily paper wasps, in her studies. Dr. Tibbetts is giving a pre-recorded presentation followed by a live Q&A session. So again, the audience can enter the questions in the Q&A feature at the bottom of your screen, and use the thumbs up icon to upvote the questions to prioritize them. Now we'll turn it over for Dr. Tibbetts' pre-recorded presentation.
Thanks so much for inviting me to be a part of this symposium. I'm going to talk a little bit about my research on paper wasps and what they can teach us about the evolution of animal minds. So humans have incredibly variable facial features. You hang out in a crowd, which many of you probably have not done recently, and you'll notice that everyone's face looks different. Now, the fact that we have so much variation in our facial features is essential for our social communication system to function. Just imagine what life would be like if everyone had the exact same face. It would be a crisis, right? How would you differentiate between your friends, your students, your family? If everyone looked the same, we would not be capable of our sophisticated recognition behavior that humans have. So variation is essential for communication. With that in mind, I spent a lot of time studying paper wasps that have variable facial patterns. And today I'm going to talk about what they use these facial patterns for and how you can use this variation to study the evolution of communication and cognition. Now, first, I'm going to take you a long, long time ago, back to when I first started my graduate degree. So when I first started graduate school, I was interested in the evolution of cooperation. Now, one of the cool things about paper wasps is that they have really interesting social behavior. So nests can either be started by a single queen who nests alone, or they can be started by a group of cooperating queens. And when many queens cooperate, the queens form a linear dominance hierarchy. The dominant wasp gets all the reproduction and doesn't have to work, and the subordinate wasps do all the work and don't get to reproduce. So from an evolutionary perspective, it's kind of a mystery why those subordinates choose to cooperate. So I was interested in studying the evolution of this cooperative behavior. And to do that experiment, what I did is I went out and I marked all of the wasps on a nest with little colored paint dots on their back. So for example, you can see there's the blue wasp here and the yellow wasp over there. And then I would videotape the colony's interactions. Now, one day, unfortunately, I ended up with a video with 2 unmarked wasps. Now, as a new graduate student, I was like, why not just watch the video anyway, see if there's anything interesting going on? And as I watched the video, I realized that I could tell the wasps apart. Now, this is even though they didn't have colored dots on their back. So this is not the video, it's just an example video. You can see 2 unmarked Polistes fuscatus paper wasps. And if you look carefully, they actually look really different. Maybe you don't see the difference. I've been told by some people that I am crazy for thinking they look different. But you'll notice that there is variation in how many yellow abdominal stripes they have. This one has a bunch of yellow abdominal stripes. This one just has 1. So long ago, after watching that video with 2 unmarked wasps, I realized their color patterns actually look different. So the next day, I went out and I started looking much more carefully at the wasps' color patterns. And what I discovered is that they have incredibly variable facial patterns. So here are portraits of 6 Polistes fuscatus wasps. You'll notice that there's a huge amount of variation in this central part of their face. There's also variation in their eyebrows, in their outer eye, and in their abdomen. So not too many people get this close up and personal with paper wasp faces. So it's not something that people had noticed before. But, I was shocked by how much variation there was. And then I set out to do a series of experiments to try to understand how they use that variation. And what the experiments taught me was that Polistes fuscatus wasps learn each other's facial markings and use the markings for individual recognition. What that means is when 2 wasps interact, they memorize what the other individual looks like. And then next time they meet, they don't need to fight again. They just say, oh yeah, you're Susie. I met you last week. You beat me up, so I'm going to give up immediately. Now, individual recognition is thought to be a relatively complicated form of communication. And that's because it requires a lot of flexible learning and memory. So I was pretty excited that we'd found individual recognition in paper wasps because it had not previously been found in insects. But of course, I was interested in learning more about the sophistication of their recognition abilities. So it would be individual recognition if a wasp could, recognize 2 other individuals and remember those individuals for a few hours. That would be individual recognition, but it would be, I don't know, kind of boring individual recognition, not super sophisticated. And you compare that with something like what humans do, where we remember thousands of individuals, we maintain those memories for decades. Now that is sophisticated individual recognition. Obviously, wasps aren't capable of that, but I wanted to know more about what they're actually capable of. So the first thing I found is that wasps use individual recognition to identify all of their nest mates. So paper wasp colonies, as you can see, aren't actually that big. So there might be maybe 30 wasps on a nest. Wasps learn all of those individuals and remember how they fit into the colony hierarchy and their roles within the colony. Wasps also use individual recognition during interactions off nests. So in the early spring, right after wasps come out of hibernation, they spend a few weeks flying around and they fight with lots of different individuals during that time. And they're kind of figuring out who they want to cooperate with. So they have lots of battles, they huddle with lots of individuals. It's a very exciting and complicated time in a paper wasp's life. And what we found is that wasps, after they fight, they remember who they fight with. Even if they only interact for a short time, if they are reintroduced to that same individual a week later, they will remember her again. So they have pretty decent memories for individuals that they interact with. So one thing I was really intrigued by was the idea that maybe wasps don't just learn by interacting with other individuals directly. Maybe they can also learn about individuals just by observation. So, and this idea kind of made sense to me because if you watch lots of wasps interact in the early spring, what you'll see is they'll hang out like on the side of a house or someone's deck or something like that. And there'll be some wasps fighting. And then standing in a ring around the fighters, you'll see other wasps who look like they're observing the fight. So I was interested in the idea that these observers were learning a lot about the fighters just by observation alone. So learning by observing contests alone is something that is common in vertebrates, especially in primates. It even does, though, occur in fish as well. And the basic scenario is some individuals watch like Jody and Susie fight. They see Jody beat up on Susie. And then even if they've never interacted with Jody or Susie before, they can say, wow, I watched Jody be super tough. So if I ever encounter her, I'm just going to give up immediately. I'm not even going to bother fighting. Now, learning about individuals via observation alone is also kind of sophisticated because it dramatically increases your social network. Instead of just paying attention to those few individuals you interact with directly, suddenly you're paying attention to this wide social network and paying attention to interactions among others, which is relatively more sophisticated. So we wanted to test whether wasps could learn by observing contests alone. And this is a project that was done by 2 undergrads, Ellery Wong and Sarah Bonello. And I had these fantastic undergrads do the project because I didn't think it was going to work. I actually thought there was no way that wasps would be capable of this. And, you don't want your graduate students to waste time on a project that's never going to work. So I decided it was an okay project for undergrads. So the way the experiment was set up is we had a couple wasps fight, and then we had a couple wasps observing the fight. The observers were separated by plastic so they could see, but they couldn't interact or fight directly. And this is a video of what these experiments look like. You can see here's one observer who's watching these 2 fighters interact. And then there's another observer over here who seems to be ignoring the fight. So after these initial observer trials, we ran a second set of trials with the same individuals. So during the first set of trials, we had observers and fighters. And then in the second set of trials, we matched up observers and fighters in another set of fights. So basically what we did is sometimes we took an observer who had watched a particular fighter before, and we had them interact. In these trials, we called them the experimental or social eavesdropping trials, because in these trials, the observer had watched that particular fighter before. So it's possible they learned about that individual fighter by observation. And then we also had control trials where we took an observer and paired her with a different fighter. So these control trials were similar in that in both cases, wasps, they were observer wasps and fighter wasps, but the key difference is in the control trials, the observer had seen a different individual fight. So social eavesdropping was not possible. And then we compared the behavior in those experimental and control trials. So what we were trying to do is differentiate between 3 alternative hypotheses for the factors that could influence behavior in that second set of trials. So our first hypothesis, the one, of course, which we were most excited about, is the social eavesdropping hypothesis. The deal with social eavesdropping, again, is that wasps are observing and remembering specific individuals. They're remembering that Jody is a badass, so when they encounter Jody in the future, they're like, whoa, I'm not even going to bother fighting you. But of course, there are more boring alternative hypotheses. So one of those alternatives is something called priming. So with priming, just seeing aggression changes your future behavior. The idea here would be an observer sees a lot of fighting, so it makes her really aggressive in the future. It doesn't matter if in the future she's interacting with the same individuals or different individuals, just seeing aggression changes her behavior. And then another kind of more boring alternative is that rival fighting experience influences behavior. This is also known as winner-loser effects. So the idea with winner-loser effects is if fighter 4 wins a fight, then in her future interactions, fighter 4 is going to continue to be more likely to win. And again, that would have nothing to do with eavesdropping. It would just be that prior experience matters. Okay, so we tested these alternatives. We could see evidence of priming and rival fighting experience in the control trials. We could only see evidence of social eavesdropping in the experimental trials. So we set the wasps up in these second fights, and first of all, we found no evidence of priming. So observing aggression of a different rival had no influence on subsequent behavior. You can see there's no correlation between the aggression that a fighter initiated that an observer saw in trial 1 and the bystander aggression in trial 2. So just seeing aggression of a different individual doesn't matter. So we can also reject the winner-loser effects or prior fighting experience hypothesis. This graph shows you the aggression initiated by a fighter in trial 1, correlated with the aggression the fighter initiated in trial 2. And what you can see is there's no significant relationship. So we can reject our alternative winner-loser effects hypothesis. So now we're down to our last hypothesis, the exciting social eavesdropping hypothesis. And what we found was pretty strong evidence for social eavesdropping. So bystanders were less aggressive towards fighters that they saw initiate more aggression. This shows you aggression initiated by a fighter in trial 1, and then the bystander aggression towards that same fighter in trial 2. You can see when a fighter was super aggressive, bystanders basically instantly gave up. They were like, wow, I saw that you're tough, I'm not going to mess with you. So it's really good evidence for social eavesdropping. Also, in support of social eavesdropping, aggression in that second trial was influenced by how much time the observer actually spent watching the rival. So if you remember from that video, the wasps often watched each other, but there were some individuals that just never watched. And we're interested in learning more about those never watchers, but we couldn't find anything associated with it so far. What we found is bystanders who spent more time watching were also less aggressive in future fights, suggesting that the more information they get, the less aggression they need to figure out their dominance in their future interactions. So together, all of the interactions in those trial 1s had a huge effect on behavior in trial 2. So statistically, the R squared was like 80%. So 80% of the variation in aggression that we saw in the second trial was accounted for by what that observer saw in trial 1, which is possibly the highest R-squared of any of my behavioral trials. Behavior is so messy. I was shocked to find such a big effect on behavior. So we can conclude that wasps do learn about rivals via observation alone. And, I've been studying wasps for a ridiculously long time, and this experiment surprised me more than any other experiment. I actually didn't think that they would be capable of learning by observation alone. So apparently I'm still underestimating wasps. And this result is cool because it suggests that wasps do actually have a large and complicated social network that they keep track of. So it's not just who they fight with directly, they're living in this complex social environment where they watch others interact and integrate it into their knowledge of their larger social environment. Again, which is remarkably sophisticated. So the fact that wasps were capable of this social eavesdropping made me wonder whether they would also be capable of something known as transitive inference. So transitive inference is using what you know to make inferences about what you don't know. So for example, if I were to tell you that John is stronger than Sally and Sally is stronger than Pete, that's what you know. And then it can ask you to make an inference. Okay, based on that, what I told you previously, do you think John is stronger or weaker than Pete? What do you think? Chances are you guess that John would be stronger than Pete. And if so, yay, congratulations, you are capable of transitive inference. So for a long time, transitive inference was thought to be based on logical deduction and thought to be a hallmark of human cognition. People thought that only humans were capable of transitive inference. In fact, the Greeks used it as an indicator of, which creatures were actually logical. And they said that humans are the only logical ones because we're the only ones capable of transitive inference. Now, of course, most things that humans think we're special about, we are not actually special about. So subsequently, there have been many studies showing that animals are capable of transitive inference. So primates often use transitive inference. It's also been found in some species of birds and even in fish. So far, there's only been one test of transitive inference in a non-vertebrate. And that study was done in honeybees. And what they found was that honeybees failed the transitive inference test. They just couldn't do it. Now, of course, I think that wasps are awesome, much better and smarter than bees. So I decided to test whether wasps might be capable of transitive inference. And the way we did the experiment is through something called a 5-element training procedure. So what you do is you train individual wasps on a bunch of premise pairs, and then you test them on an untrained pair, and you ask them to make an inference. So basically what we did is we told them, train them that A is better than B is better than C is better than D, and D is better than E. And then once we trained them on all that information, we said, okay, given what we trained you, here is B and here is D, and which one do you like more? And the idea is if they organize all of those trained premise pairs into a hierarchy and then use transitive inference to choose between them, they would prefer B over D. So how this actually works is we train them using colors. So for example, we might train them that blue is safe and yellow is shock. And then we train them that yellow is associated with safety and green is associated with shock. And then we test them that green is associated with safety and purple is associated with shock. And finally, we train them with purple associated with safety and light blue associated with shock. And then we say, okay, we've done all this training. We're just going to ask you a question. Which do you like better? Do you like yellow better or do you like purple better? And if they organize all those trained stimuli into a hierarchy and use transitive inference, they're going to prefer yellow over purple. So that's the basic idea. You have to do a lot of controls. Obviously, we switch up the colors to be sure that color preference doesn't influence things. We also change the order of training to be sure that it's there's no recency effects. So sometimes we train them starting with D and sometimes we train them starting with A. All right, so if support for transitive inference would require three things. First of all, we would need to be able to train wasps to learn all 4 of those premise pairs. Second, they need to be able to remember the premise pairs for the whole experiment, so it takes a few days to run through them all. And finally, during the untrained inferential test, we need to, the wasp would need to choose B over D. So this is an experiment done by a couple of fantastic undergraduates who spent a lot of time in a little room training wasps. And the basic setup is we put the wasp in a small maze. Most of the floor was electrified. The area next to the safe color was not electrified. So we put them in there for 30-second trials, 6 30-second trials. And then after the training, we tested them without shock for 10 trials. So we put them in that maze, and then we tested which side they went to, which color they went to. So we obviously switched the sides that the colors were on to be sure it was a color preference and not just a side preference. So we did 10 trials, and then we said, did they perform better than expected by random chance? All right, so first of all, did they learn those 4 premise pairs? And these are the results for the premise pairs. And they show you that wasps did learn to discriminate all of those premise pairs. This is the number of correct choices out of 10. The dotted line shows you the 50-50 random expectation. And you can see on each of those pairs, wasps performed better than expected by chance. So yay, we can put a check by the first one. They learned the 4 premise pairs. Now this result is actually kind of exciting because honeybees were not capable of learning all 4 premise pairs. What the honeybees did is, you trained them that A was better than B, and then when they got that really well, and then you went to train them that B is better than C, and they just couldn't do it. They were like, what? You just told me that B was bad. I can't learn that B is good. So they were unable to reverse that learning. So yay, wasps already win. All right, now on to our second choice. Do they remember the premise pairs for the whole experiment? And the answer is yes, they do. At the end of the experiment, we retested them on that original training without any additional training, and they performed just as well as they did the first time. So they remembered the training for the whole experiment. So finally, last of all, so check. They remembered that information for the whole experiment. So on to our last prediction. Do they choose B over D in the untrained inferential condition? And you're probably not going to be surprised since I'm talking about this experiment. They did, in fact, choose B over D. So this is pretty cool. These B over D trials, the wasps were never trained directly to compare B and D. We didn't train them this. This is just an inference they made from the other training that we'd done previously. When we tested B and D, there was no shock. It was done by trainers who were naive. They didn't know which color was B and which color was D, so they couldn't accidentally bias the results in any way. But nevertheless, wasps chose B over D. So it suggests that wasps are organizing the information in the premise pairs into an implicit hierarchy, so that the A is the best and E is the worst. And then they use transitive inference to choose between those untrained pairs. So it's tempting to say, yay, I proved that wasps are the best and they're way smarter than bees. But of course, that would be taking the result a little too far. I think really what's going on is that the social behavior of wasps is such that transitive inference is very beneficial. So Polistes live in cooperative groups, complex dominance interactions, social eavesdropping. So organizing the information they have linearly might be really beneficial because it allows them to make rapid inferences about novel social relationships. So without even fighting, they might say, wow, I beat Jenny last week and Jenny beat Susie, so chances are I'm going to beat Susie in the future. So I think that social taxa with these complicated social interactions might be predisposed to spontaneously organize information linearly and use transitive inference to make these deductions. Honeybees, their social life is totally different. There's a queen, there's a bunch of workers that are all equally ranked. Workers are more like cells in the body of the hive. They don't really have individual interests, they don't fight with each other for dominance, and their interactions are much simpler. So honeybees just don't need transitive inference because of their way their lives are organized. So I think the message here is that animals, even with tiny brains, like the size of a grain of rice, which is how big a wasp brain is, can do complicated things. They probably work out very simple mechanisms to do these complicated things when they need to do it because it's beneficial for their, because it's favored by selection, because it helps them have more efficient social lives. Okay. So given that Polistes paper wasps have these interesting, complicated social dynamics with social eavesdropping and transitive inference, it made me think about this hypothesis known as the social intelligence hypothesis. So the social intelligence hypothesis is one of the primary hypotheses that is thought to maybe explain why humans and some other creatures are so smart. The idea is creatures with complex, individually differentiated social relationships are smarter than other species. So for example, in humans, we learn, thousands of individuals. We keep track of relationships between lots of individuals. Who's nice, who's mean, who cheats, who doesn't cheat. Being able to keep track of those relationships is essential for our success. And it's so cognitively challenging that maybe it's selected for the giant brains that we have. So in support of the social intelligence hypothesis, many of the smartest creatures out there have these individually differentiated social relationships with things like social eavesdropping and transitive inference. So primates, some birds, especially the crow group, and also things like meerkats. So I wondered, do wasps fit into this category? Obviously they have some incredible capacity. Are these Polistes fuscatus wasps really like the smartest wasps out there because of their social behavior has selected and changed their brain relative to other wasp species? So I decided to test this hypothesis, and one of the nice things about paper wasps is there are lots and lots of different species. So the species I've been talking about the most is Polistes fuscatus. They have individual face recognition and all these interesting social dynamics. But there are other closely related species like Polistes metricus, pictured here, that lack individual recognition. So if you look at these pictures of Polistes metricus, you can see that all the wasps have identical faces, and they are not capable of individual recognition. We've confirmed that experimentally. Now, we think the reason that they don't have individual recognition is that they don't live in cooperative groups in the same way. So you don't need individual recognition if you have a simple social life. So we thought it would be interesting to compare these two species and see if there are any differences in their learning capacity. So we trained wasps to differentiate a couple different stimuli, to learn pairs. We had trained them to differentiate pairs of colors, pairs of caterpillar pictures. And we chose caterpillar pictures because wasps eat caterpillars. So it's a socially relevant, it's a relevant stimuli for them. And we also trained them to discriminate patterns. And what you can see is that there's no significant difference between the species and their ability to learn any of these different stimuli. So this suggests that there's, no overall general difference in learning. Now, I should acknowledge that these are only a few tasks, right? In the future, it'll be important to compare these two species on multiple different axes to see whether there, are any differences overall. But I think we can fairly safely say that there are no major differences in their general learning ability. So individual recognition hasn't made Polistes fuscatus super smart. So then the question is, how has it influenced their cognitive evolution? In particular, is there any evidence of specialized differences in cognition in Polistes fuscatus? And the reason the specialization hypothesis makes sense is in primates with individual face recognition, they are specialized for face learning. What that means is that they learn faces of conspecifics better than they learn other information. So, and often, like in humans, there's even a special part of the brain that's just for faces. Now, this kind of specialization is remarkable and has attracted a lot of attention because most of what animals do is, do behaviorally, is mediated by very generalized cognitive processes. You can't have a special part of the brain for each thing you do. I would say that that's like slightly impractical. So a lot of the time we have some general purpose machinery that can be used for many different situations. So there's a lot of interest in how and when specialization occurs and why specialization occurs. Okay, so like I said, humans, we are incredibly specialized for faces. We are so good at faces. It's probably something that you don't even realize. It's so second nature to us, but our capacity is really incredible. Just think about going to a conference. Maybe you might, over the course of an hour or two, you might learn 100 new faces. And you learn those faces without really focusing on it. It's just second nature. So I'm going to give you a little face learning test to see how you do. Can you find that face? Chances are you had no trouble finding that face, despite the fact that I only showed it to you for 1 second. Imagine doing a similar task with a complicated pattern. You just couldn't do it. So our specialization for faces is so strong that if you change the face in any way, like for example, turning it upside down, it really reduces our capacity. So look, see if you can find the upside down face. Chances are you found it again, but it was probably a little more difficult for you because our brain treats upside down faces differently than right side up faces. Here's an even more stunning example of that general phenomenon of the problem with upside down faces. So these two faces probably look kind of similar to you. Yes, maybe. If you turn them right side up, you can see they are not similar at all. So our brain is so specialized for faces that when you turn a face upside down, it tricks your brain and you no longer use those specialized face mechanisms. And the result is you're much worse at discriminating and identifying faces when they've been modified. All right, so we wondered, are these paper wasps, Polistes fuscatus, specialized for face learning? And if they are, we predicted that individuals will learn faces better than other visual stimuli. So this was a project done by an old grad student in the lab, Mike Sheehan. And this is a picture of Jackie Wright. She was one of the many undergraduates who helped us out with wasp training. So we trained wasps to discriminate between pairs of faces. And then we also trained them to discriminate non-face patterns like caterpillars and black and white patterns. And we also trained them to discriminate manipulated faces. So those are face pictures that have been changed in Photoshop so they're a little bit different. So some of the faces we jumbled. So we took the face parts and just rearranged them. Other faces, we digitally removed the antenna from the face image. So the idea with the manipulated faces is that they're composed of the same colors and patterns as a regular face. But because they've been digitally manipulated a little bit, it tricks the brain into not identifying them as a face. So it's really the best way to test for specialization. So here are the results. And first of all, you can see that wasps learned faces better than they learned non-face patterns. So that's a little bit of support for specialization. But even more striking is that wasps learned to discriminate regular faces much more quickly and accurately than manipulated faces. And I think the most stunning aspect of these results to me is the fact that just digitally removing the antenna dramatically reduced their learning. So that's pretty cool because the antenna don't have a lot of information. It's not like they have variable color patterns on the antenna. All of the variation is on the face. And yet just removing the antenna reduced learning. And again, the reason for that, I think, is that the wasp brain uses antenna to denote a face. So as soon as you remove the antenna, the brain no longer identifies the image as a face and uses more generalized mechanisms for recognition as opposed to the specialized face recognition mechanisms. So it's remarkable that this specialized face learning ability has evolved independently in humans, in many vertebrates, and in wasps. It suggests that specialized cognition might be a widespread adaptation for these kinds of complex behavioral tasks. So one of the neat things about paper wasps is that, again, we have lots of species, so we can compare species without face recognition and see if they also lack face specialization. So we took that same species, Polistes metricus, which we know lacks individual recognition, and we said, if the requirements of individual recognition have selected for specialized face learning, then what we expect is species that lack individual recognition will also not be specialized for face learning. So we trained Polistes metricus using similar methods, and what we found is that they are terrible at learning faces. You can see they performed just a chance. They got it right 50% of the time. They were unable to learn faces. So this result suggests that Polistes metricus are just bad at faces. But there is a slight little flaw with this experiment, which maybe you picked up. And that flaw is that Polistes metricus naturally lack face variation. So the stimuli are very different. We actually had to add variation in Photoshop to the faces so that they could, so that they had any hope of learning them. So to provide a really stronger test for the differences between species, what you need to do is train both Fuscatus and metricus on both stimuli. So that's what we did. So this over here, this shows Polistes fuscatus, the species with face recognition. You can see that they're awesome at learning their own faces, but amazingly, they're also significantly better than Polistes metricus at learning Polistes metricus faces. So this tells you it's not just about the stimuli. Polistes fuscatus, the species with individual face recognition, really are face experts, and they perform much better than other species. Another interesting kind of side note in these data is that you'll notice that both species learned Polistes fuscatus faces better than Polistes metricus faces, which is interesting when you think about selection for being individually recognizable. It tells you that those wasps not only have are favored to have specialization that makes them good at recognizing faces, their faces have also been selected to be really easy to recognize, which is, I think, a similar phenomenon that goes on in humans. Our faces, like, look at the faces of 100 different crows and the faces of 100 different humans. Our faces have been selected to be very easily distinguishable, not just to us, to any creatures. Even bees can recognize human faces. So there's selection on everyone in this scenario to make individual face recognition as efficient as possible. Okay, but the main message here is that Polistes fuscatus are face experts. They are truly specialized, and they really process faces differently than close relatives that lack individual face recognition. Okay, so we can conclude that the challenge of individual recognition has shaped cognitive evolution in paper wasps largely via specialization. And I think performing these kinds of complex behavioral tasks that are incredibly important to a species' social success can often favor the evolution of this kind of specialization. So we studied a lot of different wasps and a lot of kinds of recognition, which of course I'm not going to talk about today. But one thing I wanted to mention quickly that I'm really excited about is that we've recently found geographic variation in individual recognition. So I talked a lot about wasps, the population of wasps here in Michigan, where Polistes fuscatus have highly variable facial patterns and learn in remembering each other as individuals. But if you go to other populations of Polistes fuscatus, like for example, Polistes fuscatus from Pennsylvania, you'll find that they do not have variable faces, and they are in fact not capable of individual recognition. So there's this geographic variation in recognition, where recognition seems to be surprisingly evolutionarily flexible. And one of the reasons I'm excited about it is it means that they're a great model for understanding what kinds of neural and other mechanistic changes are happening that facilitate accurate recognition, because you have the wonderful opportunity for within species comparisons. Okay, so thanks so much to everyone who's helped with the research. A lot of graduate students have participated, many undergraduates who've done a lot of the grunt work of WASP training and WASP observation. And thanks also to U of M and NSF for funding. And thank you so much for your attention.
Thank you, Elizabeth, for the exciting talk. We have time for questions. There's several questions already coming here. Now, the first question is from Deb Gumuzio, the faculty member in the CDB department. So she actually have two questions, I believe. In the transitive inference test, did you also test the ability to select the A over C and A over E?
No, we didn't test them on each potential pair. We just tested them. We just trained them on those initial pairs, and then we tested them on A versus E and B versus D, but we didn't test them on all of them. There's like a limited amount that you can shock a wasp before it starts not responding correctly.
Right, makes sense. Is there any amplification or degradation of the effect? The further the inference has to go.
So I think that I'm not 100% sure about that question, but I think in some transitive inference tests, they use many more stimuli. So instead of just A to E, it could be like A to G. And then sometimes animals perform differently on different kinds of inferences. But we didn't do that. We just did the very basics.
All right. Yeah, so Dr. Barry Trimmer have a question. First, a fascinating work and a great talk. Do these abilities have a cost? With such a small brain, do they give up other cognitive capabilities compared to bees or ants, etc.?
So that's a great question. And the answer is there has to be a cost, right? But of course, we don't know what it is, right? Any sort of, these I mean, you guys probably know that even just forming a long-term memory is costly enough to show an effect. And if you select on lines of flies to make them really good at learning, they become less competitive. So there's lots of evidence that good cognitive abilities come at a cost. But in wasps, we don't know what the cost is, and it would be fascinating to know. I mean, you're right, they could be worse at other kinds of cognitive of tasks. They could require more energy. Like their development time is longer than other Polistes that I know, so maybe that's it, but it's definitely an interesting question.
Yeah, next upvoted question from Burton Sacker. How long does the inference training last?
So we did the experiment over the course of a week. So they know they can make these inferences for a week, but we haven't tested them for longer. And I don't really know. In their social environment, they remember each other for a surprisingly long time. They can remember each other for multiple weeks. But we don't know how long inferential learning lasts.
Yeah, next question is from Dr. Roger Cole. So are there just two types of the wasps in this species, reproducers and workers? If so, what is the purpose of a linear dominance hierarchy?
So the thing about them, I think that's really cool. And the reason they have the linear dominance hierarchy is that they're always kind of fighting over who gets what. It's not like a honeybee where there's a reproductive and then a bunch of non-reproductives. It's like everyone could potentially be a reproductive if they were successful. And then within reproductives, there's variation with some winners that get a ton and some losers that just get a little bit. So, it's more like a complicated society like humans or chimpanzees or something like that, where there's much more variation in how big a winner or a loser you are. So it's more important to keep track of everyone and how strong they are and how they're interacting.
Yeah, Elizabeth, that was a really thought-provoking talk. I have actually I have quite a few questions myself, but I ask one. So I've been wondering why face recognition? So because the animals could recognize body parts, for example, to recognize individuals. So do they, not necessarily wasps, but do animals actually recognize the body parts? This might actually also associated with the related to the logical tests for logical abilities, right? So if you just test the face recognition, maybe you'll miss some, the cognitive abilities.
Yeah, so animals communicate with each other all sorts of ways, right? Body color, odor, smell, sound. And so the fact that it's faces is cool, but I agree that, it's just like any form of complex communication. I think the reason that it's faces is because they do all sorts of, face-to-face social interactions and social signals that are used during these complicated interactions. You'd expect them to be on the face. It would be less efficient if they were on the back or something like that.
Especially for fighting, probably they are head on. So maybe.
Exactly. They just like walk up to each other and then they stare at each other in the face. You can see them like making decisions about what to do. So that's why.
Yeah. So I was just notified that we have time for a couple more questions. So I'll take advantage of this.
Okay.
Now, so is there any genetic screen that can be done to identify mutants that fail to recognize the face, or even fancier that fail to perform neurological tests?
Well, the thing I'm most excited about is the fact that there's this geographic variation, with some populations seeming to be good at face recognition, and some populations not good at recognition. So I think because of that natural variation, that presents the opportunity for us to really get a better window at what's going on genetically or developmentally. Why are some wasps smarter and some wasps dumber, basically, in a simple way. And I think there's so much that one could do with the mechanisms. I've just kind of like dipped my feet in, and I'm not so knowledgeable yet in all the details, but I'm excited about the potential. I should also say that one of the annoying things about wasps is their generation time is actually a year. So yeah, so you think it's an insect, it's going to be super easy. It's not as easy as you think.
Right. Much longer than Drosophila.
Yeah, unfortunately.
Okay, excellent. So I don't see more questions here now. So thank you so much, Elizabeth.
Sure, of course. Thanks for inviting me.
Right. I will now turn it over to Dr. Roger Cohn for closing remarks for today.
So thank you to the speakers for a wonderful group of talks today. And just want to let everybody know that we'll start up again tomorrow at 9 a.m. The symposium will go from 9 to 12:15 tomorrow. And once again, we're going to have a remarkable group of speakers covering areas from disease vectors, biological controls of disease vectors to lessons from extreme insects to the evolution of fungi and evolution of different plants. So should be an exciting day tomorrow as well, and hope to join you all then tomorrow at 9 a.m. It'll be the same web link that you'll use to join in tomorrow. So have a nice evening, everybody, and thanks again to all the speakers for some wonderful talks.
Good morning, and thank you for joining us for day 2 of the 2020 Saltiel Life Sciences Symposium. When we consider the need to broaden the biosciences, we cannot overlook the importance of biodiversity across biological and biomedical research. Biological diversity includes plants, animals, microorganisms, but also genetic differences within each species. Preserving and sustainably managing biodiversity is not only necessary for mitigating climate disruption and guaranteeing water and food security, but it also impacts human health. The COVID-19 pandemic has shown how human health is intimately connected to the natural world. Thus, biodiversity loss is a loss of humanity. The keynote lecture by Dr. Tishkoff yesterday afternoon illustrated the importance of understanding genetic differences in humans. This morning, we'll focus on exploring genetic diversity within and across less common model organisms as a way to control disease transmission, as well as to develop novel strategies that can be used in applications for human health. The mosquito, for example, is the deadliest animal on Earth and has killed more humans than all the other creatures combined in the wild. We have been actively combating this insect for more than 80 years, yet the main weapon remains unchanged: spray pesticides. A good local example is the use of aerial spray of pesticide to combat the eastern equine encephalitis here in Michigan. This is clearly inefficient for controlling mosquitoes and is harmful to humans and environment. There must be a better way. Our speakers this morning understand that we need better methods for mosquito control and are taking unique and creative approaches to tackling this problem. Hopefully, you will see that there is hope in the horizon, that in the near future, we could bring mosquito-borne disease under control, free of pesticides. Now, I'm going to introduce our first speakers in this session. The first speaker in this session is Professor Flaminia Catteruccia from Harvard University. Flaminia received her doctoral and postdoctoral training at Imperial College, London, where he worked on vector-borne transmission of disease. She continued her work in the same institution as an assistant professor in 2006. She assumed the rank of Associate Professor of Immunology and Infectious Disease at Harvard Chang School of Public Health in 2011 and was promoted to full professor in 2018. Her research group investigated molecular and behavioral parameters that are key to the ability of mosquitoes to transmit malaria, with special emphasis on reproductive biology and vector-plasmodium interactions. She hopes to provide crucial knowledge to aid the development of new, effective tools for mosquito and malaria control. For those who just joining us, please use the Q&A feature to add questions during the talk for Professor Catteruccia, and use the thumb up icon to upvote questions to prioritize them. Without further ado, let's welcome Professor Catteruccia to present a live talk.
Thank you, Shangyu, and thanks so much for inviting me to this symposium. It's a real pleasure to be here today and have the opportunity to tell you about some of the work that we're doing at the Harvard School of Public Health on the interactions between mosquitoes and the malaria parasites that they transmit. And so I know that mosquitoes are probably not very popular in general. They're probably the least popular animal in the world. And many people would like to see them all dead. But I hope that by the end of my talk, a few of you will actually realize and will believe that malaria and other infectious diseases might be interrupted without the need to eliminate all mosquitoes that transmit them. So why malaria? Well, because as Shangyu said, malaria is a major public health burden, and it's been really afflicting humanity since the start. Many explorers and generals and even US presidents like Lincoln and Roosevelt fell sick because of malaria. And here in this map, you can see how malaria looked like until the beginning of the 20th century. So there were vast areas of the world, pretty much every area of the world, including North America and Europe, that were afflicted by this disease. And these days, malaria mostly affects tropical and subtropical countries, as shown here by dark purple. But it's still a major public health issue. It infects more than 200 million people every year, and it kills, out of these 200 million people, it kills about 500,000 of them, mostly in Africa, and young children under the age of 5. So malaria still represents a huge burden for those regions that are afflicted. And Plasmodium parasites, that are the causative agents of malaria, are transmitted exclusively by one mosquito genus, the Anopheles genus. And there are about maybe 200, 300 species of Anopheles that have been detected so far, but about 30 to 40 are considered to be important vectors, and their distribution is shown here. And as you can see, this distribution closely overlaps the distribution of malaria that we've seen in the previous slides. And for this reason, the control of the mosquito vector has been really central to malaria control strategies. And 80 years ago, in the 40s and the 50s, there were very strong malaria control strategies started in North America and Europe and other regions of the world. They had, as Chengyu said, were mainly focused on two methods: controlling the mosquito by eliminating the larval breeding sites. So mosquitoes have anaquatic juvenile stages, so they need water. And so eliminating water, stagnant water, can also eliminate mosquitoes. But also mostly it was based on the use of insecticides. DDT had been discovered as a potent insecticide only a few years earlier. And so DDT was deployed in military style in large parts of the world. And here you can see campaigns in Italy, which is where I'm from, and in the US. And although probably some practices that were used then, like smoking a pipe while fumigating a child inside their homes, would not be accepted right now, 80 years down the line, we're still using the same tools. We are using insecticides for controlling these mosquitoes populations. And we're using still insecticides on wall sprays, although with better PPEs. And then we are also incorporating insecticides onto mosquito nets. And this is because these two strategies target important mosquito behaviors. Mosquitoes, at least the mosquitoes that transmit malaria in Africa, they feed at night. So they bite people at night while they are asleep. So protecting people under a bed net offers both a barrier to mosquitoes, so like a physical barrier, but also mosquitoes that become in contact with those nets are killed by the action of the insecticides. While after biting, and so after engorging on blood, mosquitoes will have to digest the blood for a period of a few hours. And to do that, they normally go and rest on the house walls. And so if those house walls are treated with insecticides, then that's how mosquitoes are killed. And these two methods of control have been really instrumental in decreasing the number of malaria cases since the beginning of this millennium. So since 2000, we've seen a sharp decline in cases, as shown here by this red line. And models by the Oxford group, led by Bat et al., have shown or have predicted that the majority of these cases have been prevented with the use of insecticide-treated bed nets, as shown here in green. But also endorrhecidal sprays in purple, and drugs like artemisinin combination therapy, which is the main drug that we used to treat patients, have contributed to this decline in malaria cases. However, what we've seen in recent years, we've seen that these strategies are not sufficient to achieve elimination and eradication of the disease. In fact, we've seen that malaria cases have stalled, so they're not going down anymore. And the reasons for this are probably multiple. However, there is probably a major factor responsible for this lack of declining cases, which is resistance. Mosquitoes are becoming more and more resistant to the insecticides that are used on mosquito nets. And so we see resistant populations all over Africa. But also parasites are becoming resistant to the action of drugs that we use to treat humans. So this is highly alarming because we are risking our major malaria control strategies. And so we need innovation, and we need additional strategies that can be incorporated into malaria control programs. And we, in my lab, like as many others, we believe that understanding the basic biology of transmission is essential to generate novel ideas and also novel tools for controlling this deadly disease. And to do that, we study the interactions between mosquitoes and the malaria parasites that grow inside those mosquitoes, both in lab settings here in Boston, but also in malaria endemic settings with our collaborators. In particular, we've been collaborating with groups in Burkina Faso in West Africa for many, many years. And one of the aspects that we've been focusing on is the interaction between mosquito reproduction and parasite development. And this is because these two processes are intimately linked. In fact, female mosquitoes, they feed on us, they feed on blood, not because they want to transmit disease, but because they need the nutrients from the blood for their reproduction. So they, after blood feeding, they will incorporate those nutrients into the eggs that develop in the ovaries. So those nutrients incorporation into the ovaries induces ovarian development in the space of about 2 days. And then when the eggs are mature, the female will be able to lay those eggs and then start another reproductive cycle. Then again, she will feed on blood, she will develop eggs and lay those eggs for multiple times until she dies. And the standard lifetime of a mosquito in the field is about 2 to 3 weeks. So not that long, but long enough to have multiple reproductive cycles. But if that female happens to feed on a human being that is also infected with Plasmodium parasites, that's when the development of the parasite will start in the mosquito midgut. And this is a very interesting and very tricky development because the parasites will have to undergo transformation into 5 different developmental stages. So first of all, the male and female gametocytes that are circulating in our blood will be picked up, and then they will transform into gametes, the sexual forms, which will fertilize and form a motile form, which is called ookinete. And the ookinete will have a very difficult job of leaving the midgut blood bowls and escaping the midgut and crossing the midgut epithelium to form a cyst, which is called oocyst, just underneath the basal lamina that surrounds the midgut epithelium. And then this oocyst will expand massively in the space of the next few days, up to a week, really enlarging in size and producing thousands of parasites called sporozoites. And these sporozoites are the infectious forms that will then travel to the salivary glands of the mosquito in the neck of the female, from where then they can be injected together with the saliva when the female bites another person. So it's a very long and complicated cycle, which overlaps pretty much with the developmental cycle of egg development in the mosquito females. So in my lab, we've been interested in understanding whether these two processes are somehow linked. So whether the presence of parasites affects egg development and whether egg development in turn affects the survival and development of the parasites. And this is done with the work of 4 really brilliant scientists in my lab, Christine Werlin and Perrine Marsanak, 2 PhD students that have recently graduated, and a postdoc, Maurice Itoe, and research scientist Rob Shaw. And one of the first questions that we addressed is whether the presence of parasites in the midgut negatively affects reproductive fitness of the female. So like as humans, we do get sick when we get Plasmodium parasites. What about mosquitoes? Do they get sick? Like, do they have a reduced reproductive output if they have parasites in the gut? And to answer these questions, we are working with combinations of mosquito and parasites that are naturally occurring in the field. So we work with African mosquitoes, the most important malaria vector called Anopheles gambiae, and Plasmodium falciparum parasites, which are the deadliest parasites that are responsible for the death of so many people. And so when we actually look at whether the presence of parasites in the midgut affects reproduction, we see that that's not the case. In fact, here on the right, you see the number of eggs developed by mosquitoes that are infected compared to the number of eggs developed by mosquitoes that are not infected. And you can see that those numbers are pretty similar. So each dot here represents one single female and the number of eggs that she develops. So pretty much the same number of eggs developed by those females, which suggests that per se, the presence of parasites does not impair the reproductive fitness of those mosquitoes. And we can go a step further and look at mosquitoes that are infected and in the same female count the number of eggs that she develops in the ovaries and the number of parasites that she has in the midgut, and see whether these 2 parameters are correlated positively, negatively, or not at all. And what we see over and over is that these 2 parameters are actually positively regulated, which was quite a surprise at the beginning for us. So we see that a mosquito that develops more eggs is also a mosquito that harbors more parasites, which again seems to suggest that there are no costs to infection. But this work was initially done in our laboratory with using Plasmodium falciparum parasites that have been in culture for many generations, and also mosquitoes, Anopheles gambiae, that have been in our lab for many, many years. So we've repeated these experiments also in using field parasites and field mosquitoes from Burkina Faso in collaboration with Thierry Lefebvre and Abdullah Diabaté from IRSS and IRD, and we see exactly the same. Mosquitoes that have more eggs also harbor more parasites, which suggests no fitness cost, but also suggests that maybe parasite development is somehow linked to processes that are important for egg development. And a way to address this question is to prevent egg development altogether. And now we can do that by using CRISPR-Cas and generating mutants that are severely impaired in egg development. As you can see here, these are ZPG mutants where we have knocked out the function of zero population growth, a gene that is important for egg development, and compared to wild type and mosquitoes which have fully developed eggs after blood feeding. And so we can take these two groups of females that develop eggs, females that do not develop eggs, infect them with Plasmodium parasites and see what happens. And what we see here is that these two groups show largely different numbers of parasites, and the mutants for egg development show reduced number of parasites. So they have fewer parasites. Parasites are somehow not developing in those females. So probably processes that are important for the development of the eggs also affect the development of the parasites. So how is egg development happening in those mosquitoes? Egg development after blood feeding happens mostly through the function of a steroid hormone called 20-hydroxyecdysone, or 20E. I will be mentioning 20 multiple times, so just remember that 20E is a steroid hormone which is synthesized by female mosquitoes after blood feeding. And its synthesis peaks at about 24 hours after blood feeding before dropping down. And it occurs in two tissues in the female. The synthesis of this hormone starts in the ovaries. So the ovaries are essential for the synthesis of this hormone. And then it's completed in the fat body, which is like the equivalent of the liver. Once 20 is synthesized, it induces the expression of lipid transporters like lipophorin and beta-glucanin, which then induce egg development by incorporating lipids and proteins into the developing eggs. So if we interfere with the function of 20E, we interfere with egg development. And we can interfere with the function of the steroid hormone by different means, but what I'll show you here is just interfering with the expression of the steroid hormone receptor heterodimer, ECR ultraspiracle, to which 20E binds, inducing a downstream signaling cascades. So if we can silence the receptor by RNAi, what we see is that there is a reduced number of eggs after blood feeding, as expected, because 20E is important for egg development, but there is also a reduced number of parasites. So while the same number of females, or the prevalence of female is the same, so the same number of females become infected, they harbor fewer parasites. So parasites are somehow killed in those females. And we know that this is not due to the function of a major immune factor, TEP1, which has been shown in studies in rodent malaria parasites to be an important component targeting O kinases in the midgut. Because when we silence the ECR receptor together with TEP1, we see that parasite numbers are not rescued. So it looks like 20E signaling affects the number of parasites in a TEP1 independent manner. And what we know is that the stages that are affected are the transition between O kinases into oocysts. So while there are the same number of O kinases, there are fewer oocysts, suggesting that this transformation is actually what is affected by 20E signaling. And we are currently interested in understanding what are the mechanisms that prevent this transformation, because this is like probably one of the most understudied aspects of the parasite biology in the mosquito and in general. So we're very much interested in trying to understand the basis of these effects that we see, this reduction in oocyst numbers. But I don't have results to show you right now, although we're working on it. But one thing that we can conclude from this data is that maybe targeting mosquito reproduction is also an additional benefit, because it induces a reduction in the number of parasites. And so this would suggest that this could be a good strategy for malaria control. We reduce the number of mosquitoes that are out there, and we also reduce the number of parasites that those mosquitoes harbor. However, when it comes to mosquito-parasite interactions, things are never this simple. And in fact, one thing that we realized is that those oocysts developing in the midgut they look different from control oocysts. And in particular, they looked bigger. As you can see here from this micrograph, those oocysts are larger in size than oocysts growing in control of females. And we can precisely measure their size throughout development. And you can see here with this red dotted line that their size in females that have a reduced number of eggs, because we impair 20 signaling, is consistently larger than the size of oocysts developing in controlled females. And we know that this means that those oocysts are developing faster. They have faster developmental rates, because when we look at the next step in development, which is sporozoite invasion of the salivary glands, we see that at day 10 after infection, when in control females, only few of those females have sporozoites, double the number of females instead have sporozoites in the gland. So sporozoites reach the salivary glands sooner. And moreover, we also see that more sporozoites are present in those glands at day 10 and also later during development. So parasites are developing faster, but they do this without losing their infectivity to humans. Because if we look at, if we take those sporozoites and we use them to infect a primary human hepatocytes in collaboration with Sangeeta Bhatia at MIT, and that's because the liver is the first tissue where parasites establish infection in humans, we see that those sporozoites have exactly the same infectivity than sporozoites that are growing slower in our controlled females. So they grow faster without really impairing their infectivity. And so how do they grow faster? We now know that they grow faster because they utilize mosquito nutrients, in particular mosquito lipids, that become redundant when females invest less in reproduction. So in females that have fewer eggs, lipids become more abundant in the midgut, and also lipid transporters increase in both transcript and protein levels, in this case lipofaurin as shown here, which is the major lipid transporter that is also important for egg development. So these lipids and lipid transporters become more available when eggs are reduced in numbers, and parasites can use those lipids, because if we co-silence the ECR receptor with the lipofaurin lipid transporter, we see that we lose the increase in oocyst size that is observed when we only silence the ECR receptor. So what this data shows is that mosquito lipids become more available if the mosquito invests less in egg development, and the parasite is able to exploit opportunistically those lipids for its own growth. And so if we look at the mosquito egg development cycle, as I told you earlier on, mosquitoes don't just feed once, but feed multiple times to develop multiple egg batches. And so another question that we are asking at the moment is, what happens if we blood feed a female again while parasites are already developing? So what happens to oocyst development if we provide a second blood meal to those females while the oocyst is already there developing in the midgut? And probably what you would expect, but it was very surprising for us to see the extent of this effect. We see that those oocysts are actually much larger if we give females a second blood meal compared to oocysts that grow in midguts that have been fed only once. So they double in size, and also what we see at an early time point after infection, 8 days, where there are no sporozoites in control females, we see that sporozoites are already formed and they are already exiting the oocyst, and so those mosquitoes become infectious much earlier than mosquitoes that have been fed once. And this is a major advantage for those parasites, because as I told you, the lifespan of the mosquito is not that long, it's about 2 to 3 weeks. And so if a parasite is capable of developing faster in that mosquito, that parasite will also have a much higher chance of being transmitted. So what we can tell out of all this data that I showed you is that what we know now is that parasites development is very intimately linked to egg development, and so that the parasite is very well adapted to the reproductive biology of its Anopheles vector. And this is probably due to a coevolution occurring over a very long period of time, because parasites and mosquitoes have been interacting for millions of years, and much longer than we as humans have been on Earth. Also, we see that parasites do not inflict fitness costs to their mosquito vectors, which is very important because it suggests that mosquitoes don't have a real interest in eliminating infection. So there are no evolutionary pressures on those mosquitoes to become refractory to infection, so to fight infections. We also see that parasites can opportunistically utilize mosquito nutrients for their own development. And so they can accelerate their development when females invest less in reproduction. And so this is a very important, a very interesting biological findings that we generated over the course of the last 3 years. But they also have important repercussions for mosquito control strategies. Because we have mosquito control strategies that are aimed at reducing the reproductive output of mosquito females. Because the idea is like, if we reduce reproductive output, we reduce the number of females that can transmit malaria, and so we reduce malaria transmission. However, what our data show is that impairing mosquito reproduction can also have unintended consequences of increasing the speed of parasite development, and therefore increasing the chances that those parasites will be transmitted. And this is particularly important for strategies that you might have heard of, like gene drive, population suppression strategies that have been in the news a lot in recent years. And for those of you who don't know what a gene drive is, so a gene drive is like a selfish element based on CRISPR-Cas, which can spread throughout wild populations in a super Mendelian fashion. So can be used to spread desired genes or desired traits into populations in a much faster way than normal alleles would spread. And so there are gene drives that have been built at the moment, which are based on targeting mosquito genes that are important for reproduction. And our data suggests that at least for a period of time, while the drive is spreading, those mosquitoes with reduced reproductive output could be better at transmitting malaria. And so this could cause negative unintended consequences for malaria control. And so it's important to evaluate carefully the effects that mosquito control strategies have on the parasite rather than just on the mosquito. And so what can we conclude? Well, we can conclude that mosquito control is challenging, because when we target directly the mosquito, mosquitoes become resistant to insecticides and other control strategies that we have used against them. But on the other hand, we also see the parasites can adapt to those mosquito strategies in ways that might be detrimental. So they might actually favor parasite transmissions and increase malaria prevalence in malaria endemic regions. And so we've been thinking a lot about these issues and how we could ensure a safe and effective malaria control without really inducing this effect. So we've been thinking a lot about how we can block transmission and the transmission cycle without really affecting too much the mosquito vector. Because although it's true that the mosquito, pathogens transmitted by mosquitoes cause a lot of death and suffering, it's also true that the pathogens are the responsible for this diseases rather than the mosquito itself. So let's go back to the two major control strategies that we are currently using for malaria. We target the mosquitoes with insecticides. So we want to kill the mosquito with the use of insecticides. And our major tool is insecticide-treated nets. So those nets are impregnated with insecticides so that when a mosquito comes and sits on the net while attracted by mosquitoes, attracted by humans that are sleeping under the net. And so with the legs, it comes in contact with those insecticides that are impregnated on the nets. And because those insecticides are lipophilic compounds that can cross the waxy layer that surrounds the cuticle of the mosquito, then the insecticides are taken up, they are absorbed by the mosquito, and so in a few minutes, the mosquito is dead. Instead, for humans, we use drugs. Obviously, we use drugs that can cure people and can kill the parasites that are in circulations. And so we started thinking about whether it would be possible. What if we merged these two strategies into one? And rather than killing mosquitoes with insecticides, we cure them by putting antimalarials on mosquito nets, so that a mosquito that in touch with the net would absorb the antimalarial, and the mosquito itself would not be affected, but the malaria parasites growing in that mosquito would be killed. So at the beginning, we thought this was a bit of a crazy idea, but we tried it nevertheless. And this project has been led by another brilliant postdoc, Doug Payton, who started using this potent antimalarial called atowacone. And Doug chose atowacone because it's very potent, but also highly lipophilic compound. And so we thought that atowacone could have a chance of penetrating the mosquito cuticle the way insecticides normally penetrate it. So what Doug did, he coated a surface with different concentrations of atowacone and allowed mosquitoes to sit on these surfaces for a few minutes, 1 minute or more. And then after this exposure time, he infected these mosquitoes with plasmodium parasites. And then a few days later, went and looked at what happens to those parasites in the midgut of these exposed mosquitoes. And to our surprise and delight, we found that exposure of mosquitoes to atowacone completely suppressed parasite development. While control females had normal infection levels, so pretty much every female was infected, so the prevalence was very high, and also the number of parasites in those mosquitoes was quite high. We saw 0 parasites developing in mosquitoes that were pre-treated with atowacone. And at the beginning, we couldn't believe it. So believe me, we repeated this multiple times. I was actually in Africa, in Burkina Faso, when Doug did this first experiment. And so it took him a while to convince me that this was actually working. Then he did dilution series and realized that these effects were actually very potent with an IC50 or 1.77 micromolar per square meter. So then Doug also started playing with conditions. What if we expose mosquitoes a day before they feed on blood, like mimicking a mosquito that, for instance, gets in touch with a net, but then is put off and then goes and bites the next night? Or what if we first infect mosquitoes and then we treat them with atowacone, in a way representing mosquitoes that bite someone and then go and rest on the house walls. And so those are the mosquitoes targeted by indoor sprays. And again, what they found that in both conditions, there was 100% killing of the parasites. We could see no parasites developing in the atowacone treated mosquitoes compared to very good infections in the controls. But what about the mosquito itself? Is the mosquito affected? The answer is no. We see no strong or evident effects on mortality. So the longevity curves are exactly overlapping in atowacone treated and control females. And also the number of eggs developed by those females is exactly the same. So there are no obvious fitness costs in those mosquitoes, which would suggest that mosquitoes wouldn't have any reason to develop resistance to these strategies. So there will be no selection pressure on those mosquitoes to develop resistance, which is what we want to avoid and what happens when we use instead insecticides. So with this promising data, we went to our friends in the epidemiology department, Caroline Bacchi and Lauren Childs. And Lauren Childs is now at Virginia Tech. And we asked them to model what difference it would make in malaria prevalence if we added an antimalarial on mosquito nets. So what they did, they took a number of regions in West and East Africa, for which we have data on malaria prevalence, bed net usage, so how many people use bed nets, and also the level of insecticide resistance in mosquito populations. And then they model what would happen if we added onto those bed nets an antimalarial that is as potent as atowacone. And here you can see the results in both West Africa and East Africa. Adding an antimalarial onto those nets would strongly reduce the prevalence in the human population, the prevalence of malaria parasites in the human populations, with the strongest effect observed in regions where there is very high insecticide resistance in mosquitoes, and so in regions where bed nets are actually not working anymore. And so this is, okay, it's only a mathematical model, so it has to be proven in the field. However, it's a promising result that suggests that we could use a strategy like this. We could incorporate antimalarials on nets, especially in regions where mosquitoes are not killed anymore by insecticides, which is pretty much vast regions across Africa and also other regions in tropical countries. But so for this to be true, for this to be real, this strategy has to work on Plasmodium falciparum parasites that are currently circulating in Africa, and also mosquitoes that are from natural breeding sites, and those mosquitoes are strongly resistant to insecticides. And we thought that maybe the mechanisms that are inducing insecticide resistance in those mosquitoes could also possibly interfere with the function of drugs, and so could abolish the effects that we see in insecticide susceptible mosquitoes. And so we've done these studies again in Burkina Faso in collaboration with Thierry and Abdullah. And there we can use their amazing setup system where we go out in the morning before school starts, and we screen children, because children have the highest prevalence of malaria, unfortunately. So we screen children for the presence of parasites. We take a drop of their blood, we go back to the lab, we screen for parasites. And then 2 of the children that have high parasite loads are taken back to the lab the next day. Their blood is drawn, and we use this blood to infect our mosquito from the laboratory, but also mosquitoes from directly collected from the field. So in Bobo Dioulasso, which is where we do this work, and is the second largest city in Burkina Faso, there are villages surrounding the city that are full of rice fields, which are perfect breeding sites for these mosquitoes. And so we can take those mosquitoes and infect them with the blood from children. So really natural combination of parasites and mosquitoes. And what's also interesting about this is that this is also a small little contribution of our research to those communities, because every child that is found infected with malaria is then treated with drugs, and so is cured of malaria, which is like a small contribution, but still is a little impact that we have on those communities. And so when we do this experiment using field parasites and also field mosquitoes, we see again the same results. No parasites are developing when they're infected, used to infect mosquitoes treated with atowacon. So showing that really this strategy could be working in Africa. However, what's important to note is that for this strategy to work, we should use drugs that are not used as human therapeutics. So atowacon, as I told you, is a potent antimalarial and is used as a prophylactic drug for travelers, and also used as a stopgap therapy when ACT, when artemisinin combination therapies don't work. And so if all of a sudden we started coding atowacon all mosquito nets in Africa, then we would risk inducing resistance to atowacon in parasites. And so we would risk losing this drug as a human therapeutics. So we need to identify compounds that are different from the drugs that are used in humans. So that's a very important point to make. But we also saw that using different drugs in humans than in mosquitoes will have an additional advantage. And because then this would probably limit the chances of parasites that are resistant to human therapeutics to spread. Because if you can imagine, for instance, an artemisinin-resistant parasite that develops in humans is taken up by a mosquito, but is treated with another drug, then the chances of these parasites to develop also resistance to the second drug would be more limited. And so it's like we could then attack parasites with multiple drugs in their different stages in what we like to call human-mosquito combination therapy. And we actually showed proof of principle that this can happen in collaborations with Diane Wirth and Selina Bopp at Harvard, where we take advantage of their collection of artemisinin-resistant parasites from Cambodia. So these are parasites that are strongly resistant to the frontline drug, artemisinin. And if we feed those parasites to mosquitoes that have been pre-treated with atolacone, we again see that those parasites do not survive, so they are killed 100%, which brings support to the possibility of using drugs on nets, also to delay the spread of drug resistance against human parasite resistance against human therapeutics. And so what we are doing at present, we are trying to find other compounds that can be used for these mosquito targeted strategies. And for that, we have a vast library of compounds that have anti-malaria properties from GSK, from Medicine for Malaria Ventures and other partners that we can screen for their ability to kill parasites in the mosquitoes. And hopefully we will soon have additional compounds that could be incorporated into bed nets or into indoor sprays and to prevent malaria transmission. So what I showed you today is that targeting mosquitoes is challenging, but it's also been successful in the past and even at present. However, we need innovation in this area if we want to achieve malaria elimination and malaria eradication worldwide. We provide a proof of principle that we can actually treat mosquitoes with antimalarials. We can cure mosquitoes with antimalarials and to prevent transmission to humans. And the models predict that this strategy would be very successful at reducing malaria prevalence in the field. But for that, we need to screen for additional compounds and compounds that are not used in humans to reduce the risk of drug resistance developing and spreading. And maybe in the future, we can even envisage a world where we will do without insecticides and we will have more targeted strategies to block transmission of these deadly pathogens. And then finally, I want to go back to the people that are doing the work. Big thank to Christine, Rob, Maurice, Doug, and Perrine for their amazing work. And also the rest of the lab that has contributed to this project: Naresh Singh, Ali Prost, Erica Ma, Ezra Du, Kelsey Adams, Emily Salan, Duo Peng, and Megan Marquette, which have worked on these different aspects of projects. And the rest of the lab, which I didn't have time to discuss their projects, unfortunately. And also, again, I thank our collaborators, in particular, our collaborators in Burkina Faso, Thierry and Abdullah and their teams, and also the people in the villages from Sumuso and the villages near Bobo Dioulasso, where the work is done, and also our sponsors, NIH and HSRMI and Bill and Melinda Gates Foundation for their support to our work. And I'm happy to take questions. Thank you very much for your attention.
Thank you very much, Professor Catteruccia, for a very interesting idea and very innovative idea about curing a mosquito of malaria instead of wiping them out. I never would have thought about it. So it's really cool to see that. Again, we really appreciate these innovative ideas. So I would like to let the attendee know that Professor Catteruccia has a few minutes to answer questions live. So please use the Q&A feature that you can see at the bottom of your screen to submit your questions. And so if you share a similar idea and questions with other participants, please use the thumb up icon to upvote on the question. And then we'll address the question based upon the most vote first. So I guess I will open up the question myself and while others are typing in their questions. So I'm wondering, you mentioned that atolacone can cure malaria and can kill these disease parasites. Is it pretty effective for how many species of these plasmodiums have you tested?
Yeah, so that's a very good question, because we have been working only with Plasmodium falciparum, which is the deadliest parasite that causes malaria. And we've seen these effects against falciparum, which are very strong in both humans and mosquitoes. However, there are other parasites that can cause malaria, and in particular, there is one parasite called Plasmodium vivax, which is very widespread, and it's a very important parasite also because it cannot be completely eliminated at present, because it also has some dormant forms which go silent and then are woken up due to some stressors, which is very difficult then to eliminate and to completely cure. So we haven't been able to yet to target those parasites, and so to do our studies on those parasites, because that is Plasmodium vivax cannot be cultured effectively, while we can culture Plasmodium falciparum and so use it for our infections in the lab. We cannot culture Plasmodium vivax. I mean, people have tried for decades, and now there are some results that are promising. However, we still don't have a stable and robust culture system, which means we cannot do infections in the lab. We have to go to places where malaria transmitted by, malaria caused by Plasmodium vivax is endemic. And it's something that we would like to do with our collaborators in South America and Southeast Asia, or even Africa, because now Plasmodium vivax is also quite widespread in Ethiopia. But we haven't been able to do that, definitely not this year in the COVID era, and we hope that we'll be able to resume our field work sooner rather than later, because this has been one of the also major repercussions of this pandemic, is that we cannot go and perform our studies in field settings, which is very, very, very upsetting.
Thank you. Thank you so much for the very thoughtful answers. So while you are answering the question, there was a new question coming in from Zena Labs. She complimented your great talk and very cool research. She loves the idea of treating mosquitoes. She wondered if you can imagine the approach will eventually lead to another plateau in the decrease in number of infections, like we have seen in recent years with current treatment strategy, as you mentioned at the beginning of your talk. If so, do you have other ideas about how to potentially eliminate malaria entirely in the nature? Thank you.
Yes, that's also a very good question, because it's true that everything that we've done to contrast malaria has met resistance in mosquitoes and in parasites. And so we should expect that even this strategy would at some point meet similar problems. Parasites could develop resistance to the drugs that are used on mosquito nets. And so we would see the effectiveness decreasing. A way to go around this is to use multiple drugs that can therefore, so with different mechanisms, different modes of actions that then would limit the chances of those parasites to develop resistance. So that's one of the things that we are trying to do now. So using multiple drugs on nets, and also different drugs that are used in humans that would reduce the chances of parasites developing resistance. It's also true that malaria is a very complex disease, and so we don't only have mosquitoes that bite indoors. So because bed nets and sprays, they only stop transmission by mosquitoes that bite, and so they feed and rest inside our house walls. However, there are other mosquitoes that have different behaviors. So mosquitoes that prefer to bite outdoors. And so those mosquitoes are not targeted at all by any malaria control intervention apart from elimination of larval breeding sites. So it will be very important also to generate tools that are targeted against those mosquito populations that cannot feed, that do not feed and rest indoors. So they have different behaviors that is not controlled by the strategies that we are using now. So we have some ideas about that. We can use sugar traps. This is something that has been developed by others. They use sugar traps that are incorporate insecticides, because even if every mosquito needs to feed on sugar, they don't just feed on blood, they feed on plants, they feed on nectar. So the incorporated antimalarials also on sugar traps that's like a place outside the homes or in different locations in villages. So mosquitoes could be attracted to those sugar baits, but then the parasites would be killed. This still uses antimalarial concepts, but this is really the project we are investing at the moment. We are still thinking about other ideas. We really like the idea of controlling malaria without affecting the ecosystem, without affecting mosquito populations, and also without introducing toxic compounds. So we are trying to develop new ideas also in that direction. But at the moment, unfortunately, this is the idea that we're focusing on. We don't have anything so developed yet.
Thank you. Thank you. So there's another question that can just come in. This is from Tom Blackwell. He is wondering, what is the assay procedure for a large-scale antimalarial screen?
Yeah, so this is like Tom has identified a really crucial problem. So how can we do large-scale screens? And at the moment, the answer is we cannot. There is no easy way to do high-throughput screens against mosquito stages, because for human stages of parasites, we have culture systems, so we have cell cultures. We can culture parasites in red blood cells, so we can plate them in 384 well plates and try different drugs. But for parasite stages in mosquitoes, that's not possible at the moment. We don't have a robust in vitro system. And so what we can do is just to, every time we have a new compound, we need to test it in mosquitoes. So we need to actually test it in vivo, which obviously reduces the throughput. And now we have developed a system by which we can test multiple compounds at the same time. So we can expose mosquitoes to like 10 different compounds and then test which ones are effective. However, we also invest in quite a lot in an in vitro culture system. And we have some success based on in vitro culture systems that were already developed by other people in the past, but were not really optimized. So we are trying to focus on optimizing a culture system that then would allow us to do screens at the same or at least similar throughput that is done for human stages of parasite development. And that's really, at the moment, is a rate limiting step. It's what's limiting our ability to develop these strategies and expand it.
Thank you. So I'm very intrigued by your presentation. So I'm just going to volunteer another question. So following Tom's question, I wonder, what if we use other type of insects, for example, like fruit flies? Can we infect them? Can we turn them to become a carrier of malaria? And because they also use 20-hydroxyecdysone signaling pathway, the same genes that work in fruit fly most likely also work in mosquitoes. So if we can turn these fruit flies as walking incubators that carry the malaria parasite in their gut, and these fruit fly will feed on sugar water, and that might be a platform to screen for compounds. I'm just wondering if you could comment on that.
Yeah, so yes, definitely it would be great to use fruit flies as a model system for malaria. And there's been some work done by David Schneider and others that has shown that the initial stages of parasite development can be recapitulated in fruit flies. So all kinds can be formed with different in different conditions and different feeding regimes. So that's doable, although then the entire life cycle I don't think has ever been done. So that would need some specific investment in this area, which might be worth doing. So to have an alternative system for doing the screening, and also for studying the biology of those interactions. Although I should say that even studying different mosquito parasite combinations can lead to different results, because it looks like those parasites are very well adapted to the specific mosquitoes that transmit them. So what we could see with a different system, like an artificial system, like the fruit fly, might not provide biological results that are entirely applicable to the interactions between mosquitoes and parasites. But it would be definitely a good way to increase our understanding of parasite biology and possibly identify new strategies.
So I guess I'm going to just ask a far-reaching question. For example, do you imagine similar strategy being used to trigger things like the tiger mosquitoes, highly vicious Viron in Asia that carry dengue fever pathogens?
Yeah, so as you correctly said, there are other mosquito species like the Aedes mosquitoes, Aedes aegypti in primis, but also the tiger mosquitoes, Aedes albopictus, that are good vectors of viral pathogens like dengue, Zika, chikungunya. And so far there is no cure for those pathogens, and also there's been no effective way of preventing transmission by the mosquito, because they don't bite indoors, so they are not really targeted by mosquito nets, for instance. So for those mosquitoes, there are though very, very interesting and novel strategies that are being designed and they are being used at present. So there are big trials that have started in the last few years, which are based on infecting those mosquitoes with a bacterium called Wolbachia, which many of you might have heard of. So Wolbachia is this very interesting bacterium that has the ability to spread through populations. And also some forms of Wolbachia have antiviral effects. So if a mosquito is infected with Wolbachia, viral development is aborted in those mosquitoes. So there are massive campaigns based on releasing Aedes mosquitoes, so tiger mosquitoes infected with Wolbachia to take over wild mosquito populations, and at the same time prevent transmission of those viruses, because those mosquitoes are not competent for viral pathogens. And so that's probably the best strategy that we have at present, and I'll be looking forward to reading the results of those big trials. And there's been also considerable efforts to move these strategies onto Anopheles mosquitoes to block malaria transmission. However, Wolbachia is not so happy to sit and to develop in Anopheles as it is to develop in Aedes, for some reasons that we also in my lab are trying, and also in many other labs, we're trying to understand why Wolbachia does not develop happily in Anopheles mosquitoes. And so far, this lack of infection, of reliable infection, has limited our ability to study this system also to block malaria. But definitely also in terms of our strategy, if we could find drugs and compounds that inhibit viral development, that could also be utilized to cure mosquitoes. However, those drugs are not really there yet. So this is still early days for the strategies in other mosquitoes.
Well, I really like to tell you that it's very, very interesting, very cool. Thank you again for sharing the very exciting work from your laboratory and ideas that others might find them useful for their own work. Again, we really appreciate your participation in the symposium. Thank you very much.
Thank you very much for this invitation. It's been a pleasure.
Thank you. I'm going to welcome our next speaker, Professor Nicholas Teets from University of Kentucky. Nicholas received his doctoral training at the Ohio State University and carried out his postdoctoral research training at University of Florida. He became an assistant professor in the Department of Entomology at the University of Kentucky in 2016, where he has been since. Nicholas first gained appreciation of how insects can tolerate extreme environmental stress when he was an undergrad student working on Antarctic midge. As an independent investigator, his research group continues to investigate molecular and physiological adaptation of insects to extreme environments. He hopes to use genetic technology to improve control of disease-bearing insect pests, as well as sustainability of agriculturally beneficial insects. Again, for those of you who just joined us, please use the Q&A feature to add questions during the talk of Professor Teets, and use the thumb up icon to upvote questions to prioritize them. Without further ado, we'll turn it over for Professor Teets' pre-recorded presentation. Thank you.
All right, I want to thank the organizers of the symposium for giving me a chance today to share some of our research on some really cool insects. Before I do that, I want to convince you that there's a real need for cryopreservation research. One reason is that the waiting list for organ transplants has been rapidly increasing over the last 30 years, while the number of transplants has not been able to keep up. So if we had the ability to store human tissues and organs in the cold for long periods of time, it would significantly improve transplant outcomes. In the realm of research, cryopreservation would allow us to be able to store precious germplasm in the cold for long periods of time until it is needed for experiments. So my lab works on Drosophila in addition to the insects I'm talking about today. And as many of you know, it's impossible to store Drosophila in the frozen state for long periods of time. And so as a result, facilities like this Bloomington Stock Center spend lots of time and resources every year continually maintaining fly stocks. So one of the driving forces behind our research is this idea of whether we can use lessons from naturally freeze-tolerant animals in order to make other organisms freeze-tolerant. And so when we look at cold-blooded animals that have to survive in low temperatures, they're divided into two major categories. Most species, including well-studied models like fruit flies and tribolium, are freeze-intolerant, meaning that if ice forms inside the blood, it is lethal. However, a select few species have evolved the ability to be freeze-tolerant. So depicted on top are two insects that I have worked with, Urasta solidaginus and Chimomyza costata, and there are also a couple of vertebrate species, including wood frogs and painted turtles, that are freeze-tolerant as well. So we think that these freeze-tolerant species provide us a potential wealth of information that we can use to improve freezing outcomes in other organisms. The species I'm going to discuss today is the Antarctic midge, Belgica antarctica. It's the only insect species endemic to Antarctica and the world's southernmost living insect. A fun trivia nugget you can take home to your colleagues and families: it's the largest terrestrial animal in Antarctica, so you can see here, it's pretty huge. And we're interested in it because the larvae depicted here are freeze tolerant year-round, meaning that at any point of year they can survive freezing, and so we think they're a good system for studying the physiology and molecular biology of natural freeze tolerance. So when we study the mechanisms of freezing tolerance, it's helpful to think of distinct phases that are occurring during a freezing event. So there are things that are happening before freezing as the insect is being cooled down that allow it to prepare for the freezing and improve its chances of survival. There are some things that are happening during freezing, although we think that this is largely a period of inactivity where most biological processes cease. And then there are important processes that are happening after freezing that allow the insect to repair the damage incurred during freezing and resume normal function. So today I'm going to show you some examples of some things that are happening both before and after a freezing event that we think are important for surviving. Now, one way that this insect can prepare for a freezing event is through a process called rapid cold hardening. And so rapid cold hardening is a type of phenotypic plasticity where brief chilling prior to freezing significantly enhances survival. So what this looks like is if we take larvae and directly freeze them at minus 18 degrees Celsius, so plunge them to minus 18, almost all of them die. But if we first allow them to hang out at minus 5 degrees Celsius for a short period of time, we see that survival significantly improves. So in as little as 15 minutes, we start to see an improvement in survival. And by the time you do a 2-hour pretreatment, almost all of the larvae survive. Now this is great. The issue is though is that it's very difficult to study molecular mechanisms in a dead insect. So the first study I'm going to present is testing the hypothesis that rapid cold hardening protects against sublethal freezing injury. So if we can demonstrate that rapid cold hardening protects against injury prior to the onset of mortality, this would be important for allowing us to study the mechanism because as I just mentioned, it's much easier to study mechanisms in a living insect. So for this study, we took larvae and we directly plunged them to minus 9 degrees Celsius in this directly frozen group. This is a temperature they can survive just fine. And in the rapid cold hardening group, we first paused them at minus 5 for 2 hours prior to taking them down to minus 9 for 24 hours. And then we assessed physiological and molecular parameters during recovery from these events. The first thing we observed is that rapid cold hardening improves neuromuscular function, and we assess this by looking at how long does it take the larvae to recover after freezing and start moving normally. So we can see here that the RCH larvae in blue recover much more quickly than the DF larvae in orange, which is consistent with improved neuromuscular function during recovery. And we think part of this can be explained by the preservation of normal metabolic rate in the by rapid cold hardening. So we see that in orange, the directly frozen larvae at both 2 and 24 hours of recovery have suppressed metabolism relative to the control larvae, whereas this is not observed in the RCH larvae. And what we think what might be happening is that rapid cold hardening is protecting against mitochondrial function. This is a hypothesis that we're actively exploring at the moment. In addition, we see that rapid cold hardening preserves tissue function or tissue viability at the suborganismal level. So we have a microscopy assay where living cells are stained green and dead cells are stained red, and we can use this to assess the health of tissues after a freezing event. And so in the case of control tissues, which were just dissected out and stained, we see mostly green cells, which is good. But if we do this directly freezing treatment, we see that there's a high rate of cell mortality that's starting to occur. But much of this mortality is prevented by rapid cold hardening. If we quantify it, we can see that there's a nice improvement in cell survival in the RCH group, although it doesn't completely return to control levels. And we repeated this in 2 different tissues, and pretty much the same result was obtained in both tissues. We also observed that rapid cold hardening reduces protein damage. And so here we use heat shock protein expression as a proxy for protein damage. So we've established in previous work that heat shock protein transcript abundance is a useful biomarker for indicating the level of suborganismal stress that these midges experience, and that they're produced in direct response to protein damage. And so what we see here is that after 24 hours of recovery, in the expression of 2 heat shock proteins, HSP60, a mitochondrial heat shock protein, and a general chaperone, HSP90, they're both elevated in the directly frozen larvae, but have returned to control conditions in the RCH larvae. So this is consistent with sustained protein damage that's occurring in these larvae that are directly frozen, and that damage is prevented by rapid cold hardening. So that's great. So this series of experiments has demonstrated that rapid cold hardening does protect against sublethal freezing injury. And then the next question that we're trying to work out is, how does this actually work? What are the protective mechanisms that are conferring this enhanced freeze tolerance? So we have some clues from other studies from my lab and other groups, partly in this insect, also in other systems as well. We are pretty confident that gene expression is not a major contributor to this phenotype. Simply the time that it occurs and the temperatures are not conducive to gene expression. So rather, this process seems to be triggered by the cell signaling events, including calcium signaling, P38 MAP kinase, and some other reversible phosphorylation events. We also observe that cultured insect cells are capable of rapid cold hardening, suggesting that it's not mediated by the nervous system or by hormones. However, as I mentioned, we don't really know what are the actual mechanisms that are conferring protection. And so this is an open area of research that we're exploring. All right, so that was an example of something that can happen before a freezing event. I also want to tell you what can happen after. And we think this part is particularly important for a lot of reasons. As an example with humans, for those of you familiar, the Hall of Fame baseball player Ted Williams, his body is cryogenically frozen at a facility in Arizona. And it's doing fine. It's hanging out there frozen. It's been there since 2002. But the issue is that we don't know how to wake Ted Williams up. And so these processes that are occurring during thawing after a freezing event are critically important for determining the ability to successfully go through a freezing event. So to assess this, we're using a transcriptomics approach here to identify transcriptional mechanisms that are specific to freezing but not cold per se. And the way we can do this is we can take advantage of this process called inoculative freezing. So if we take larvae down to -3 degrees Celsius in a wet environment, ice crystals that are present in the environment will cross the cuticle barrier and trigger freezing inside the larvae. However, if we leave these larvae in a dry environment at -3 degrees Celsius, they can stay supercooled, meaning their body fluids can stay at a liquid state at -3 degrees Celsius. So by comparing these 2 groups, we can look at transcriptional mechanisms that are specific to freezing and that are not activated by cold per se. And so again, getting back to our schematic here, this process will allow us to identify those challenges that are specific to freezing. So our first prediction was that freezing and supercooling would elicit distinct gene expression profiles, even though they're happening at the same temperature. And that was definitely the case. So if we use a principal component analysis here to break down our variables into a single variable that explains 35% of the variance, we see that there's, especially early in recovery, after 2 and 12 hours of recovery, there's no overlap between the frozen and the supercooled larvae. There's some nice separation here. So this suggests that there are distinct gene expression profiles, although they do converge at 24 hours. Our second prediction is that we'd have higher expression of stress-related genes in frozen larvae to deal with the stress of ice formation. And again, we see that as the case. So if we pick out 2 examples here, HSP70 and its co-chaperone HSP40 tend to be more abundant in frozen larvae. Again, it's consistent with higher levels of stress. Now, if we do this in a more global fashion, we did some KEGG pathway analysis. I don't have time to go through the details, but we see that during early time points of recovery, we see an abundance of genes that are related to protein processing in the ER and also endocytosis, which are abundant, which are differentially expressed between frozen and supercooled larvae. And then later in recovery, we see a shift where we see an apparent importance of lipid metabolism in determining recovery from freezing events. And so we have a lot more work to do to make sense of those results, but that's sort of an active area. So to summarize here, to build a freeze-tolerant animal, we think that we can use information from these naturally freeze-tolerant insects to help guide these processes. And so freeze tolerance involves processes that occur in preparation for freezing, during freezing, and during recovery. I also showed that rapid cold hardening can quickly enhance freezing tolerance, but its mechanism is unclear, and that there are specific molecular processes that are activated by freezing, although we need functional validation. Some of the work we're doing now is moving towards using this species that I mentioned before as a potential genetic model for freeze tolerance because it's much more conducive to laboratory studies than the Antarctic insect I presented today. All right, so with that, I'd like to thank a lot of people, the Life Sciences Institute for inviting me for this talk, my lab members, as well as our funding sources, which for this project is primarily the National Science Foundation. Thanks.
Thank you so much, Professor Teets, for the very cool model organism that you mentioned and the new sort of the more laboratory accessible model that you are trying to put together. So again, I want to remind the attendees to type in your question if you are interested in whatever you have in your mind. So while you're working on this, I have a couple questions to ask, if you don't mind. So what do we know about these vertebrates that are cold tolerant?
So yeah, there's been some work done on those. They're behind on the molecular side. There's been some really good physiology and biochemistry done in these freeze-tolerant species, particularly the freeze-tolerant wood frogs that I mentioned. And there are some similarities with insects. One of the things that the wood frogs seem to do is while they are freezing, so while they're in that process of cooling down, their bladder produces, or they produce a lot of glucose that's reabsorbed into the bloodstream through the bladder. And so this seems to be a way to produce an antifreeze molecule that can protect them during the freezing. But really what's happening at the molecular level in these species has not been as well described as some of the insect work, although there have been a few studies starting to come out now. Yeah, that would be a great system to work with. Again, not very easy to work with in the lab. Hard for the wood frogs. There's really no good way to get them to breed in the laboratory. So you have to go out every year when they're mating and collect them once a year. So it's a little bit more challenging to do experiments on them. But there has been some good work out of other labs.
Okay, thank you so much for answering my questions. While you are answering the questions, a couple of very interesting questions come in. So the first question is from Josie Cloning. She's wondering, how do animal size / dimension affect the cold or freezing tolerance?
That's a really good question, and I don't know if I have a good answer to that. One of the things that's interesting about freeze tolerance, at least in the insect side, is that it seems to have evolved multiple times. So there's no clear phylogenetic signal to when freeze tolerance evolved in insects. And so with that kind of leads to the possibility that different insects might have different ways of becoming freeze tolerant. And so that's something we would like to look at in the future, is doing close comparisons of different freeze tolerant species to see the extent to which they're relying on the same mechanisms or species specific mechanisms. Now, in general, we do think that it's more challenging for a large animal to freeze than a small animal. And partly is that one of the important processes that occurs during freezing is being able to control the rate of ice formation. So a lot of insects do this through molecules called ice nucleators, or they even have bacteria present that serve as a surface to allow controlled ice crystal growth. And so if you're a larger animal with many more body compartments or larger body compartments, we think this can be a little more challenging to do. And so in general, we don't see a lot of very large animals that are freeze tolerant. So but that's a good question though.
Oh, wonderful, thank you so much. Another question is from Dr. Roger Combe. He's wondering, I'm trying to find the questions. Are there any anti-freeze proteins in the midge? You kind of answered the question already.
So we don't think they have anti-freeze proteins necessarily in the true sense of the word. So there are some insects and fish that do have antifreeze proteins that either prevent ice formation, or if they are freeze tolerant, they prevent the ice crystals from growing too fast. We don't see any evidence of that in the midges per se. One of the things about the midges is that even though they can survive very long periods of freezing, the environments we find them in are really not that cold in the sense that they're buried under snow and ice in the winter. So they stay frozen for 8 or 9 months at a time, but they're really only getting down to -5 or -10 degrees Celsius, whereas we have species here in North America that can survive much lower than that. So with the Antarctic species, it's more the duration that's impressive in terms of their ability to stay frozen like this for so long.
Wonderful, thank you so much. Another question is coming from one of the symposium presenters, Professor Barry Trimmer. He complimented your great research. He's wondering if there is a difference between insects that can survive brief freezing, such as Manduka caterpillar, and those who adapt for long-term resistance.
Yeah, so that's another good question. And actually, I enjoyed your talk yesterday, Dr. Trimmer. We raise Manduka for our little side note. We raise Manduka for the entomology class that I'm teaching this semester. So it was nice to learn a little bit more about them, in addition to just how to grow them in a cup. But yeah, no, I think as far as the difference between insects that can survive short-term versus long-term cold. So one of the key distinctions is Manduka, you can put them in the cold for a little bit and they'll be fine. But if ice forms inside their body, then it is lethal for them. Manduka is considered a freeze intolerant species. So they can survive below zero through supercooling that I mentioned before, but they can't survive freezing. And so that's one of the challenges of this research is, again, none of the freeze tolerant insects are particularly good genetic models, at least at this point in time. So we have a decent understanding of the mechanisms involved with general cold tolerance, but not so much with specifically those insects that can survive freezing. So some of the things we're trying to do are use comparative genomics and other comparative approaches to try to see if we can identify key adaptations that might have allowed for the evolution of freeze tolerance within certain groups of insects.
We have one question coming from my co-organizer, Bing. Please go ahead, Bing.
Yeah, fascinating talk, Nicholas. I cannot type in the question because of panelists. So in Michigan, in the winter of Michigan, the temperature can go down to -20 Celsius. Mm-hmm. But then in the spring, you still see blue flies. So do Drosophila in the wild survive the cold temperature, or are they just from the houses? Do you have any insight on that?
Yeah, Drosophila, they do have some ability to survive in the cold, but for places like Michigan, they're most likely finding shelter to spend the winter. So that may be inside human dwellings, but even places, we trap for them all year round in some of our field sites that we work at. And we'll occasionally find them in like storage sheds in the winter, places like that near farms or near fruit places where there's fruit. So they do seem to require human. Drosophila originated in sub-Saharan Africa, and so it does seem to be dependent on some ability to find relatively warm places to survive the winter. But it can go have some limited degree to survive in the cold as well. but probably not the temperatures that, not definitely not the air temperatures you're seeing in Michigan for sure. So they have to find some sort of shelter.
Thank you.
Yep.
Well, thank you so much, Professor Teets, for the very interesting, fascinating presentation that you shared with us. And we really appreciate participation in this symposium. So we are going to take a short break and resume the second half of this session at 10:35. Thank you again, everyone, for attending, and thanks to our presenters. Hello. I think it's time for us to resume the second half of this session. I would like to welcome our next speaker, Professor Raymond St from the University of Maryland. Raymond received his doctoral training at the University of Bath, United Kingdom. He then moved to the Boyce Thompson Institute, Cornell University, Ithaca for his postdoctoral training, and rose up in rank to become a central scientist. He was recruited to the University of Maryland as a tenured associate professor in 1996, and then was promoted to full professor in 2001. Raymond has dedicated his entire career in understanding the biochemistry and molecular biology of enteropathogenic fungi with the aim of using them as a source of innovation for the agricultural industries. He has led the effort to identify and characterize fungal toxic products that are specific to particular biochemical organ system in insects. This effort has provided a vast resource of genes that enhance the pathogenicity of insecticidal fungi and viruses and to produce insect-resistant plants. For those who just joining us, please use the Q&A feature to add questions during the talk of Professor St. and use the thumb up icon to upvote questions to prioritize them. Without further ado, we'll turn it over to Professor St. pre-recorded presentation. Thank you.
So this is my talk for the day, Transgenic Fungi for Mosquito Control. Here's my co-authors. Well, there's me there. These are my co-authors. This is Abdullah Diabiti, who's head of entomology at Centre Maraz in Burkina Faso. This is his graduate student, Etienne Bilger. That's my former graduate student, Brian Lovett. And we're here earlier on this year at the AAAS meetings, getting an award for a paper we published in Science last year concerning the application of these transgenic fungi in Burkina Faso. And I'll be talking about that work a little later on. So let me tell you something about transgenic fungi. They almost always infect insects via the spore of some kind, which germinates to produce an episodian infection structure, which produces a PEG, which uses mechanical pressure and enzymes to penetrate through the cuticle. And then when the fungus gets to the blood of the insect, it buds off this yeast-like phase called blastospores, and they colonize the insect. This poor little ant here has been colonized by a fungus, one of the zombie fungi, showed some worrying symptoms. The fungus basically took over the brain and the behavior of the ant. And once the ant was dead, what you see is the fungus emerging again from the cadaver, and it's producing this great big fruiting structure from the head of the ant. And any ant within about 6 foot of that fruiting structure is in deadly danger because it's going to spray out spores all around itself. Now, these insect pathogenic fungi are very common, they're very widespread, and they often decimate insect populations in spectacular episodics. Most insects don't get to die of old age, or they don't get snapped up by a passing bird. They die of disease, and fungi are the major disease-causing agents in insects. Which brings us to the question of, can we use those fungi for pest control? Could we grow them up and put them out in fields in place of chemical insecticides? Well, the answer is, for the vast majority of these fungi, no, we can't. They're obligate pathogens. They can only basically grow in their insect hosts. You can't culture them. There is, though, a group of insect pathogens, which includes the genus Metavisium, which have lost the sexual stage, and instead they spread from insect to insect via these asexually produced green spores called conidia. And these fungi can be cultured, whether they're a generalist broad host range Metavisium, like the strain which killed this cockroach, or whether they're highly specialized, like the locus-specific Metavisium acridium. You can grow them up. And because you can grow them up, you can mass produce them. In China and South America in particular, they produce thousands of tons of Metavisium. Here's Cheng Su Wang, a former postdoc of mine, and that's Yuxin Yu. And this is their factory, and they can produce Metavisium as cheaply as you can produce a chemical insecticide like pyrethroid. The Chinese mostly apply Metavisium acridium, so they're controlling locusts. You can apply the fungus any way you would apply chemical insecticides. You can use it as bait, which has been done here, or you can spray it out of a plane. The problem with this fungus, the problem basically with biocontrol agents in general, is they take a long time to kill. This fungus takes 8, 9, 10 days to kill. That means you have to apply the fungus when the locusts are still very young, they're hoppers. If you were to wait and apply the fungus when the insects are mature, the insects would cause much too much damage before you get control of them. So that's been a major limitation to the application of all biocontrol. So let's turn our attention to mosquitoes, the vectors of malaria. It's been calculated that malaria has been responsible for the death of half of all the people who've ever lived on our planet. So mosquitoes then have killed more people even than people have. Currently, there are up to 500 million new cases every year, and about 500,000 to 1 million die from it each year, mostly children in sub-Saharan Africa. Consequently, there's been a huge effort by the World Health Organization, by the EU, and particularly by America, which commenced with President Bush, to control mosquitoes and thereby malaria. And the major means we have for controlling mosquitoes is to use insecticide-treated bed nets. So from about 2010, 145 million nets delivered per year, and that's led to what's been called net culture. People obviously sleep under those nets, they hang these nets up in their houses, they even have meetings and go to school under those nets. So what's going to happen? You expose millions and billions of mosquitoes week after week, month after month, year after year to these insecticides. How are the mosquitoes going to react? Well, they're going to evolve resistance. In the good old days, 20 to 30 years ago, you slept under a bed net. When you woke up in the morning, the net would be covered in dead mosquitoes. Now, you have to sleep in the middle of the bed. You can't have any surface contact in the net because the mosquitoes will just probe right through. In Burkina Faso, which is where we do our work, let's look at the statistics. 25 years ago, 50% of mosquitoes were killed within 2 minutes exposure to pyrethroid. By 2014, only 26% of mosquitoes were killed by 10 hours exposure to pyrethroid. The other 74% were doing pretty well. So the World Health Organization put out an urgent call for new means of controlling mosquitoes. This is my favorite, a laser beam used to zap the mosquitoes out of the sky. But we can't really have billions and billions of laser beams scattered across Africa. We needed something a bit more practical. So we had a fungus which specifically targets locusts. Could we find a fungus which specifically targets mosquitoes? So employees of the Centre Mauvaise in Burkina Faso went out and they searched where mosquitoes hide out. After they have a blood meal, female mosquitoes like to find somewhere dark and secluded to hide, to digest the blood and to make eggs. So they looked in those places. These are all the mosquitoes and midges collected from this hut where this poor little goat lived. Now, we want a product which can control all the strains of mosquito which carry malaria. It'd be very, very nice if we had a product which also controlled Aedes aegypti, which is the mosquito that vectors dengue. But we don't want a mosquito which has such a broad host range that it could target honeybees of any other arthropod mankind deemed to be useful. So we collected a lot of fungi from Burkina Faso from various sources, including mosquitoes, and we bioassayed them against a panel of insects. So S26, that's a pretty good killer of mosquitoes, but it also killed grasshoppers and bees. So we wouldn't want that, would we? We settled on a group of Metarhizium pincensia, which was specific to mosquitoes and probably represent a new subspecies of mosquito specialists. However, we had the same trouble with this mosquito pathogen as we'd had with the locust pathogen. It takes too long to kill, 10 to 14 days. So before dying, the mosquitoes could easily still transmit malaria, and of course they'd lay eggs and keep the population of mosquitoes going. So what could we do about that? Well, we developed genetic engineering. This is Cheng-Su Wang. You saw him earlier in the factory in China. He was a postdoc of mine. And he developed this technology when he was with me. In this case, we're using a gene from a scorpion. And it encodes a very well-studied peptide, a sodium channel blocker called AAIT. And our first step is to synthesize the gene for the toxin using Metarhizium's preferred codon usage. And then what we do is we put it under control of a particular promoter. It's called the MCL1 promoter. And this promoter in the fungus operates a collagen-like protein which masks the fungus from recognition by the insect immune system. So it only works in the insect blood. It doesn't work anywhere else. So here's the fungus penetrating through the cuticle, and only when it gets through the cuticle does it release the toxin. So it's basically functioning like the stinger of a scorpion. The stinger penetrates the cuticle and it injects the venom. So the fungus is like the stinger or like a hypodermic, whichever way you want to think of it, penetrates through the cuticle, and only then does it release the toxin. This provides us with biological and environmental containment. The fungus in the environment or whatever is just going to be like any wild type fungus. It's only in the blood of the insect is it going to produce the transgene product. So here's our general strategy then. Mosquitoes will feed off people, including people infected with malaria. The malaria, the Plasmodium, is taken into the gut of the mosquito where it undergoes a series of complex developmental processes. It exits the gut and then it swims in the blood as this kind of little tadpole-like stage called the sporozoite. And they swim to the salivary glands so they can infect someone else. Here's our fungus. Our fungus is going to penetrate through the cuticle and go into the blood of the mosquito. So there's going to be a time then when the fungus and the mosquito are co-inhabiting. That's the fungus there. That's its blastospores and hyphal lengths. Those are green. And that's the sporozoites of the malaria. They're red. So our strategy is to get the fungus to make either an agent which kills the mosquito or an agent which kills the malaria. So look at that first. This was Waigu Fan, who was a postdoc of mine, and he worked with Joel Vigo Rodriguez, who was a postdoc of Marcella Jacobs-Lavina. And Marcella has this wonderful facility at the Johns Hopkins Institute, whereby they can keep, it's a very high containment facility, they can keep human malaria mosquitoes and human malaria. And what's happening here is that the Anopheles gambit, the human malaria mosquitoes, are feeding on blood infested with malaria. Not many facilities in America allow you to do that. So we engineered our Metarhizium in green to express anti-plasmodials, including a single chain antibody, PFN-PNA-1, which recognizes specifically and attaches to the sporozoites and causes them all to agglutinate, to stick together. We also got the fungus to express a peptide called scorpion, which is produced by a scorpion and is very effective against Plasmodium. And we made it even more effective by producing a fusion product, which was the scorpion fused onto SM1 peptide. This peptide specifically binds the salivary glands, which means that the salivary glands become covered in a layer of the scorpion. And that protects the salivary glands from invasion by the Plasmodium. So we could actually, in a way, cure the mosquitoes of malaria. We could reduce the incidence of the Plasmodium mosquitoes by more than 98%. And our strategy here was kind of to try to be evolution proof. We could use an attenuated strain of the fungus, which didn't kill the mosquitoes, but did produce those proteins. So the mosquitoes would not be selected for resistance to the fungus, but we could cure them of malaria. However, our stakeholders and colleagues in Africa, they thought this was a great idea, but they really wanted to kill the mosquitoes, not just cure them of malaria. Besides, mosquitoes can carry a variety of different diseases. So our alternative tactic was to stack insecticidal genes into the fungus. We could get those genes from other fungi, which kill insects. We made a lot of our own genes, gene shuffling, bits and pieces from different genes fused together. Our best results, though, came from relying on a couple 100 million years of evolution, using the very selective, very potent toxins which have evolved in arthropods. We started with the imperial scorpion, which, as you can see, plays well with children. And that's the source of our sodium channel blocker. And this is the best characterized of all arthropod toxins. So we were in good shape there when it came to regulation. But most of the genes we've worked with came from this. Our best friend, the Blue Mountains funnel web spider, produces a huge array of different toxins, and you can fractionate them when you get the venom. It's easy to get the venom. You just poke the spider and it rears up and it produces a drop of the venom, then you fractionate it. One of those toxins in particular has been well characterized. So well characterized, it's actually been approved by the EPA, and that's hybrid. So this shows a neural junction in an insect, and what we're looking at here is the target for hybrid, which is calcium and potassium channels. The AAIT, the sodium channel blocker, has the same target as the pyrethroid insecticides. In all cases, they're highly selective for insects. So this is the time course of an infection. As you can see, the wild-type fungus takes more than 8 days to kill the mosquitoes. And then we can put into the fungi individual toxin genes and see what happens. The sodium channel blocker reduces time of death to about 5.5 days. Hybrid is a bit more effective. You're killing the mosquitoes in about 4 days. So how does that reduce the dosage of spores required to kill by, let's say, 4 days? Well, if you use the fungus expressing the sodium channel blocker, then you reduce the number of spores required to kill in 4 days by about 9-fold. If you get the fungus to express the hybrid toxin, which blocks potassium and calcium channels, you reduce the number of spores required to kill it in 4 days by 40-fold. And if you get the fungus to co-express the toxins, to make both toxins, then you reduce the number of spores required to kill in 4 days by about 150-fold. And that's basically, you're getting down to 1 spore will kill 1 mosquito in 4 days. But it's better than that, because you're not just, you don't really just want the fungus to kill the mosquitoes. What really matters is that the fungus stops the mosquitoes feeding, because if they're not feeding, they're not infecting. So in the papers, we report on the use of guinea pigs. We have these choice chambers. We put the mosquitoes at the one end, and we count how many of them fly towards the guinea pigs. So the fungus takes a few days to kill the mosquitoes, but they stop feeding before then. 85% of mosquitoes exposed to the fungus expressing hybrid cease to respond to guinea pigs within 70 hours post-infection. Well, that's why we report the matter in the papers. In the lab, actually, though, we rely a lot more on the Wagyu test. So Wagyu was a postdoc in my lab, and it turns out to be extraordinarily attractive to mosquitoes. Sounds like hyperbole, but it isn't. Mosquitoes will literally fly right round me to get to Wagyu. So Wagyu would infect the mosquitoes with different fungi. then simply put his hand near the cage. When did the mosquitoes stop responding to Wagyu? So the World Health Organization has a series of processes they want you to go through before they will authorize to authenticate a new product as being useful for mosquito pest control. Well, the first phase is the development, which is in the laboratory where you produce your product and you do your initial tests. You then go on to physically confined trials, sometimes called semi-field trials, in a malaria endemic region of the world. If that works out, then you can go on to deployment, which is where you do open field releases of increasing complexity, and you do post-implementation surveillance. And the transition from one phase to the next is subject to go, no-go decision criteria. Efficacy, the new product has to be so good, so much better than what went before, that it justifies any potential risk. You, of course, have to obtain regulatory approvals before you go on to each new phase, and social acceptance. You have to be very close to constantly interact with the stakeholders to get approval from them, the local people, to get approval from them to carry on the research. So our next stage then was the semi-field trial. Thanks to money from NIH, we built a mosquito spear just outside the village of Somoza in Burkina Faso, an area where you can expect to get a couple of hundred bites from mosquitoes every day, and a couple of % of them are going to carry malaria. So you can see here, this is when the mosquito spear was being built. You've got these 4 huts in these 4 chambers. You've also got a mosquito rearing facility and housing for the bullocks, which will be used to feed the mosquitoes. So each of these chambers is going to be surrounded by mosquito netting. Citizen science. The citizenry of Somoza went out to collect eggs and larval anopheles to be reared and used in the spear. We didn't want to use mosquitoes which had been sort of semi-domesticated, just reared generation after generation in the lab. We wanted to look at the diversity of wild mosquitoes out there. And this shows why it's very difficult to control mosquitoes by targeting eggs and larvae. These mosquitoes will lay eggs in little puddles, even footprints. So we collect our mosquitoes. This is, we tried to find a way of applying the fungus which would be really cheap and easy for the local citizenry. We ended up using black sheets, just black cotton sheets, which are locally available. We put the fungus in sesame oil. Sesame oil is quite expensive here, but it's cheap out there. So we put the fungus in sesame oil and then just painted it on those black sheets. We're using in these trials metrorrhizum expressing the single gene hybrid. So not our best strain, not our strain expressing 2 toxins, but it took some years to get the regulatory approvals for this. And when we started, that was our best strain. It's not the strain we necessarily would use now. We'd use a strain expressing 2 toxins, but this is the gene we got regulatory approval for. And the fungus also carried GFP, green fluorescent protein, so we could trace it, follow it, identify it. So there's the black sheep. We can hang those anywhere in the house. Here's our little bullock. Here's Etienne, and he's releasing the mosquitoes. And then at various times, you can harvest dead mosquitoes. Here he's got the sheet on the ceiling rather than the wall. And we can monitor the mosquito's life cycle. We've got little breeding sites in each chamber. And we can count the eggs, and we can count the larvae. And we can put little nets over those breeding sites to count the emergent adults. So here's the results. % survival, days after capture from the semi-field. These huts were supplied with black sheets with no fungus, so that's the control. Black sheets covered in the wild-type fungus, wild-type virulence, but expressing the red fluorescent protein gene, and the transgenic fungus expressing hybrid and GFP. As you can see, fungus expressing hybrid kills more quickly. We've got this 25% of mosquitoes that don't get killed. We found out that those were the ones that didn't contact the black sheets. They had not got infected with the fungus. In terms of establishing populations in the spear, in the control without fungus, you can see there's a couple of generations and the mosquitoes are getting more abundant at each generation. The control fungus suppressed the population of mosquitoes, but is still having a second generation. And fungus expressing hybrid, as you can see, the population collapsed and there was no second generation. Because the transgenic fungus kills at lower spore dosages, it also has greater effective persistence. So there's the controls, there's the wild type fungus, and there's the transgenic fungus, and it's killing for longer. So it's 6 weeks and 7 weeks and so on. As I say, that's because the fungus is dying over time, but even after several weeks, there's still sufficient numbers of the hybrid spores to kill the mosquitoes. These were our worst results we obtained over 3 years. That particular rainy season, the temperatures going up and down, up and down, up and down, which killed a lot of the fungus quite quickly. But you have to present your worst results in these cases. And we think that if we use the fungus which is expressing 2 toxins, we could improve on this because you have 1 spore killing 1 mosquito, and we can still detect a sufficient dosage of the fungus even after 12, 13, or 14 weeks when we use the fungus expressing 2 toxins. These experiments got quite a lot of coverage in the media. We were ranked for several days the second most read story on the BBC website. It had several million readers. We never got to #1 because we were always kept off that site by a rotating set of stories about President Trump. So what are we doing now? Well, we're not necessarily proposing to use our Metavisium as a standalone technology. We want to integrate it with other technologies. We found that the transgenic Metavisium actually makes insecticide-resistant mosquitoes susceptible to insecticides again, which we hadn't expected. We're investigating how that happens. But in the meantime, we've got approval from the Burkina Faso regulatory authorities to do trials in these open huts, mixing and matching different combinations of the fungus and chemical insecticides. We want to see if the fungus in tandem or used synergistically with the insecticides could be used to manage resistance and perhaps make insecticides useful again, give them a second chance, give them a second life in the field. Other experiments, we want to know what is the potential for host range changes if the pathogen persists. Our mosquito pathogens have relations which attack other insects. That means there must be mechanisms for host shifts. What are these, and can we predict when they occur? One of the set of experiments we're doing is to look for potential for pathogen adaptation. So we take a Metavisium which likes Anopheles and we passage it on Aedes. Will it adapt to Aedes? We take a strain which likes Aedes mosquitoes and passage it on Anopheles. Can we get the mosquito, so can we take the fungus to like Anopheles mosquitoes? We're also looking at what we call realized evolution in nature, which is the end result of natural selection. So we're getting a whole range of these different fungal isolates with different host ranges, and we're doing the genomes and we're looking for the differences in signal transduction and so on, which may account for differences in host ranges. Now, we've already done similar studies like that on the locust pathogen Metavisium acridium. So it's specific to locusts. It won't attack cockroaches. We're comparing it with the strain here, which will attack both locusts and cockroaches. So looking at the way that the insect is seen by the fungus, how it's recognized by the fungus, how the fungus recognizes this on an appropriate insect host. And in the case of Metavisium acridium, we found it was looking for specific chemicals on the locust, which signal to the fungus, aha, I'm on a locust. We found other Metavisiums, like this caterpillar specialist, which look for topographical signals, physical shapes on the cuticle surface. So this particular fungus meanders over the surface of the insect. It only produces the infection structures when it comes across the hair socket. The hair socket represents a zone of weakness in the cuticle of the caterpillar. So we got quite good at this sort of thing, and we're actually now able to mix and match genes. So we could take a particular gene from a generalist, for example, and put that into the locust specialist and get that specialist to attack a couple of other insects besides locusts. Didn't do it well, but we were surprised to see it done at all. So we should be able to identify what allows mosquito pathogens to be specific and perhaps to alter that specificity. Increase their host range or reduce their host range. We can also enhance or diminish environmental survival of the fungus or the transgene. So we've got mechanisms now whereby we can actually get the transgene excised from the genome of the fungus after it's killed the insect. We can also change the properties of the fungus. We can take out the photolyase from the fungus, and that makes it much more susceptible to UV. So now it's trapped in the house. It will die if it goes into daylight. But we can take genes from archaea, which are highly resistant to UV. We can put those genes into Metivisium. We make a Metivisium which can survive all day in the African sun. That's part of a long-term set of experiments. We could produce a fungus which recycled through mosquito populations, permanently suppressing them. That would require a lot of regulatory approval before we could ever do that, but it's now technically possible to do. Could insects develop resistance to insect pathogenic fungi? Does a population of insects have the variability for natural selection to operate on? Hard to do that with mosquitoes, much easier to do that with fruit flies. And here's John Wang, and in his experiments, he got 188 Drosophila lines, which have had their genomes done, and he tested them against a different type of Metivisium called Metivisium anisopliae. And we found that some were quite susceptible down here, and others were comparatively resistant up here. Now, what do you think is the major predictor that a fruit fly is going to be resistant, comparatively resistant, to the fungus? Turned out to be nothing that we expected. It turned out that the resistant flies tended to have a lot of naps. They tended to take a lot of little sleeps. And somehow that correlated with resistance. Because these flies have had their genomes done, we were able to do a GWAS study. So these are the genes with variable alleles which account for differences in the susceptibility of a fruit fly. What you don't find here is the canonical immune genes. There are some genes for phagocytosis and engulfment, but you're not getting the different pathways which control the immune system in the fruit fly. What you're getting is a lot of genes like the FOXO gene here, which are involved in how energy is shunted in the fly, how decisions are made as to where energy is going to be allocated in the fly. And what you're also getting is a lot of genes which are involved in sleep processing in the fly. So if you're going to look to see why mosquitoes could increase their resistance to these insect pathogenic fungi, it won't necessarily be by mechanisms or genes which we could find easy to predict right now. There's a lot of politics involved in our project. We're working with social anthropologist Leah Pareto to engage with the community. We want to know what they know about the disease, vector control and our technology, where they get their information from, and how would they like the technology to be introduced and proven safe. We're basically trying to find out what it would take to get permission from the stakeholders, from the community, to carry this work further. And what form would that permission take? Are we talking about one person, one vote? We're inputting at various different levels in society, the king of the Mossa, the various other traditional authorities, religious authorities, various parliamentary groups, local authorities, women's associations, where we have a lot of communications with women's associations, of course, the media. We have very good relationships with the Minister of Research and his assistants and deputies have been very supportive of our work. One thing we'd like to do, because very little is known about African public opinion on these issues, is to conduct a survey. The first steps before you conduct such a survey is to actually design the survey to try to find out what kind of questions are you going to ask. You need to develop sample questions, and then you do what's called a qualitative survey, which is sort of a question and answer session with different people. You're trying to find out what questions work. This is called questionnaire piloting. And this is the stage we're at now. So questions on familiarity with genetically modified organisms. How would you test their familiarity? So you might ask, which of the following genetically modified organisms have you heard of? And then you just give them a long list. How would you define genetic engineering? And this is in English, but these questions are going to have to be translated into French and several local languages. So here's our definition of genetic engineering. Perhaps you can think of alternatives or improvements. Genetic modification is another way to develop animals, vegetables, or fruits with characteristics desired by humans that is faster than selective breeding and could have unknown consequences for human health or the environment. So we're telling them that as far as we can work out, this technology is very safe. But there could be things that we haven't thought of, that we haven't seen. There could be consequences. We're not ruling those out, which is saying to the best of our ability, we don't know what they are. So we've got a lot of local stakeholder support. We've talked to lots of people. We've got lots of community engagement. This is the chief of Somoza, who's been a very strong supporter of our work. The Centre Moraz, which is close to Somoza, is where they first tested bed nets, insecticide-treated bed nets. And the chief of members is a young man participating as a sleeper. So he used to spend the night under the new nets to determine their effectiveness. And he's seen how they've lost their effectiveness over the years. And he's proud of the support and the work his villagers, his people have done in developing new technologies. So these are our backers, these are our financial supporters, and this is where we did the work. And let me be the first to wish you a Merry Christmas. Thank you very much.
Thank you very much, Professor St. for the very creative way to approach insect controls. Fascinating. I guess I'm going to start liking fungus a lot more than I have always been, thanks to your lecture. So during your presentation, there's a question that comes in from Xiaoyan Wang. He or she is indicating that there are millions of genetically modified mosquitoes that have been approved to be released to control mosquitoes against mosquito-borne disease such as Zika, dengue, and yellow fever, and so forth. So do you foresee these genetically modified fungi doing similar job? And what are the pros and cons of using these genetically modified fungi? Thank you.
You produce the mosquitoes being released in Florida is the kind of sterile male technique. You're releasing male mosquitoes. We've developed approaches with Abdullah Diabiti in Burkina Faso, whereby we can actually get those male mosquitoes to walk on a surface with the fungus. They pick up the fungus on their feet, and then they can actually transmit it to females. So you could actually use those different technologies together. We've kind of designed this fungus to be able to dovetail with other transgenic technologies, as well as with chemical insecticides. You're going to need to integrate all these technologies together. It's not going to be one size fits all. Different technologies will be better in some situations than in other situations. You can mix and match these different technologies. Just using the fungi, you can get fungi which will survive in different environmental conditions, which will have different effector genes, which will either target the malaria or target the mosquito. And then you can mix and match them with other technologies as well. So even in a small area of Burkina Faso, what works in one set of villages may not be optimal for what works in another set of villages. And I think we and the people who are developing the GMO mosquitoes, we're just trying to develop a series of technologies which will be then made available to stakeholders to mix and match for themselves.
Thank you. Thank you very much. There's another question that came in while you're answering the question. This person commented on your talk, indicating that you have given a wonderful talk. This person is wondering if you could talk a little bit more on why the strategy inserting a new gene could not be used for insect controlling fungi, and why are they not tenable?
Why couldn't they be used? Well, we are using them. I wasn't quite sure I followed the question. Why are the techniques not tenable?
Right, so why this strategy, inserting a gene, could not be used for insect controlling fungi, and why are they not tenable?
You're not following that. They are tenable. We are using those approaches. Are you asking insect controlling fungi why we couldn't use them against other types of insects besides mosquitoes? So we developed a transgenic fungus which specifically targets locusts, for example. We developed transgenic fungi which specifically target whiteflies and aphids. So it's a very amenable, it's a very flexible technology. Fungi are probably the major disease-causing agents of insects. There's 12,000 species of ants. There's reckoned to be about 12,000 species of fungi attacking just ants. So there's a huge amount of flexibility out there in terms of the resources which are available. Scorpions are believed to produce at least 500,000 different toxins. So again, that's a huge natural resource which is available. One of the advantages of working with our insect pathogenic fungi is they're very easy to work with. It's much easier to produce a transgenic fungus than a transgenic mosquito. We can actually easily put genes into the fungus. And it acts as like a little hypodermic. It's going to inject that protein into the insect. So we can test the protein. We can test it with the fungus as a little injector. And if that works, we can actually then produce transgenic insects which express those proteins. The fungus is a much more facile way of doing these experiments. And that's one of the ways we started with Jacobs Marcelo Lavina, who's fellow Jacobs Lavina, who was the first person to develop transgenic mosquito. We worked with him because our fungus is a much more effective, much more rapid way of testing these proteins, which are anti-insect or anti-malaria, and just trying to produce a transgenic insect.
So I have a personal sort of question that I'm wondering if you could try to answer. What's the stability, what's the half-life of these toxins? So what if, say, can these toxins persist long enough and high enough a dosage that could cause deleterious effect on human or they can happen to local stakeholders, especially kids who touch things and touch their face, put their fingers in their mouth. Is that something that concerns?
So to date, we've worked with two toxins in particular. One's called hybrid, and one's that sodium channel blocker AAIT. They've gone through very extensive tests, which even involves them being fed to mammals and birds and so on. They're harmless. So they're peptides. They're a string of amino acids. They'll break down in the gut. You might imagine with large levels of these, you might get an allergic response, but that hasn't been found yet. So these peptides are intrinsically safe. Then we put those peptides under the control of a promoter in the fungus, which only makes the peptide in the insect blood. Doesn't make it anywhere else. It's not going to be in the environment. As peptides, they can be degraded in the environment. When the mosquito dies, the peptides will be degraded as the mosquito is degraded. So we've got a level of steady-state peptide only produced by the fungus. When it's in insect blood, the fungus only attacks mosquitoes.
So I guess the other question, which you, during your presentation, you mentioned about the specificity, but I wonder how about this sort of the horizontal gene transfer of these transgenic construct, and would you mind commenting on that?
Yeah, we've been studying horizontal gene transfer since 1992. We were the first people to actually demonstrate horizontal gene transfer in the fungus. We produced a paper last year in PNES where we did all these different genomes of the fungus. I was working with Wagyu Fan, Zhejiang University, all these different genomes of these different fungi, and we looked at the rate of horizontal gene transfer. So in evolutionary scale, it's been significant. There's been about 20 HGT events in Metavysium in the last 50 million years. In human historical terms, then it's a very, very, very rare event. If it occurs, it can be significant. What we're doing with here is, we've got, so say it did occur, we've got our gene under control of a very specific and complicated promoter. It doesn't work, that promoter, in other fungi or yeast or whatever. It has to have the right transcriptional factors and things to actually go to play to turn it on. Those transcriptional factors which get activated when the fungus makes its way through the cuticle and detects very specific set of circumstances in the insect hemolymph of oxygen levels, particular chemicals, and so on. So one of the promoters we use right now, we've only ever been able to get it to work in living insect blood. Never been able to get it to work in any other situation, circumstance we've tested.
Wonderful. So do you, so you do not foresee, so did you ever encounter resistance from the local stakeholders when you asked for their participation, and how did you deal with?
Not from local stakeholders. Currently, in Symposium and elsewhere, there's real difficulties and issues with insecticide-resistant mosquitoes. They're looking for alternatives. In the cities and so on, there's a small group there who regard any sort of interaction with Western technology as being risky and antagonistic to Burkina Faso and its way of life. But this technology and its introduction is being led by Burkina Faso scientists, so Centre Muraz, particularly Abdullah Diabetes, we mentioned a couple of times here today. He is the leader of this endeavor. Everything that happens is done by Centre Muraz scientists out there. They're usually the public face of this project. And we came in to introduce this technology as a possibility, but a large amount of the work we've done there has been capacity building, new buildings and training in data analysis and statistics and so on, so that the Centre Muraz scientists, the scientists of Burkina Faso and Western Africa in general, can produce their own technologies. They don't need any input from us at all.
So you can imagine that those local stakeholders, they obviously had a very difficult time for a long time. They have to live with these diseases. But there are other places, for example, in the state of Maryland, if you envision to try to deploy this type of technology in the state of Maryland, how would you go about convincing the locals that this is safe?
It's an interesting because it's risk-benefit analysis. You look at the potential benefits of the technologies and you look at the potential risks. Currently, the risks associated with mosquitoes and the diseases they carry are very low in Maryland. They're much more severe in Burkina Faso. Current predictions suggest that with global warming, Anopheles gambia may be able to overwinter in Maryland in about 20-25 years' time. Then that will be a different circumstance and a different situation. And then the risk-benefit analysis will change again.
So it's definitely good to stay ahead of a curve.
Yes, exactly what we're doing. And again, There's other people speaking today and working with different approaches, just providing this big range of technologies which people can choose from as their needs change.
The question that Nicholas just submitted is there an environmental component to the performance of the transgenic fungus?
Yes, there is. And that is something you need to carefully select on. One of the reasons we isolated fungi from Burkina Faso is these fungi have adapted to Burkina Faso conditions. You know, I mentioned the locust pathogen, that's Metalisium acridum, that's adapted to deserts. There's another fungus we work with called Metalisium frigidium, which evolved in the Ice Age, and that's adapted to cold conditions. So those are two extremes. In between, you find that Metalisium is very common, almost ubiquitous, and is adapted to different environments, different degrees of humidity and temperature. You need to carefully consider those components. So the Metalisium you use in Kenya or Tanzania might not be the same Metalisium you use in Burkina Faso. They're adapted differently. Different temperatures, different humidities, and so on. So, you know, that particular year, the third year, which I showed you, where the fungus lost control after 8 weeks, that's because the conditions out there were atypical. The temperatures going very high, then they were plummeting, going high, very plummeting. And you might expect those extreme weather conditions as global warming increases. And that the fungus, therefore, was not adapted because those weren't the normal conditions for Burkina Faso, and our fungus wasn't adapted to deal with them. Normally, the fungus lasted a lot longer than that, but you always have to show your worst data. You know, how badly it can work, how well it can work. So, for credibility, you need to lead off the worst case scenario. That was a very unusual year, I'm told.
Thank you. Thank you very much. So if we don't have any additional questions for Professor St. Ledgers, I would like to thank you again to share with us these very exciting results. It's a lot of fun, especially when you show the movie of shooting a bug with a laser.
That's wonderful, isn't it? I normally have sound effects for that.
Anyhow, we really appreciate your participation in this symposium. Thank you again.
Thank you for asking me.
I'm going to turn over the hosting responsibility of the symposium to my co-organizer, Professor Ken Cadigan. Ken?
Thanks, Shang-Yu. So now we're going to have a short talk from my colleague, Tim James. And just a brief introduction. Tim got his undergrad at University of Georgia, majoring in plant biology, and then started to work on fungal systematics and biology at Duke University in the lab of Ritus Vagalis. He then did a postdoc at Uppsala University working with Hanna Johannessen and Jan Stenlid. And after a short postdoc in Canada, joined the faculty at University of Michigan in 2009, where he's currently the Louis E. Waymeyer and Elaine Prince Waymeyer Professor of Mycology and Professor of Ecology and Evolutionary Biology. So you've already heard a lot about fungi, and there are more than 5 million species in this group. And Tim hasn't worked on all of them, but he's published over 100 papers working on a variety of free-living, parasitic, and pathological species. And I'd say to summarize his lab, they combine high throughput sequencing with in-depth analysis of particular organisms to really try to understand fungal evolution and ecology. And I'm sure he's going to tell us about some of that today. So before giving Tim the floor, I just want to remind everyone to use the Q&A feature, adding questions for Tim during the talk, and to use the thumbs up icon so we can prioritize questions after the talk. Tim, are you ready?
Ready, yeah. Thank you to the organizers for this opportunity to give this talk. Thank you to Ken for the introduction. It's a real treat that I never expected to have the opportunity. And I'm gonna go ahead and start sharing my screen. The talk title is a bit unusual because it says recombination versus mutation, but in reality, all evolution is driven by the combination of these two forces. So they do work hand in hand. But the point that I wanted to consider in this talk is how mutation and heterozygosity influences recombination, and also in evolution in regards to how rapid evolution might occur. So evolution on a very short time scales. I'm couching this talk in terms of super fungi, and super fungi are those fungi that are extremely successful. And of course, success is not really something that fungi are shooting for, but from our perspective, super fungi are organisms that are doing things that cause us to notice them. So causing epidemics such as, amphibian disease, white nose syndrome, but they also include organisms that are not agents of disease necessarily, but they are really successful. They're ubiquitous and they're common. Things like rhizopus molds and brewer's yeast, which are really common in the environment. What makes those organisms so successful? And my lab tries to approach this question using a number of different sort of mindsets. And the one that I'm going to focus on today is population genetics, where we're looking at the role of mutation, recombination, and mating system in terms of understanding what is making species as well as lineages successful. The question that sort of prompted this talk, and as some of the research in the lab, is how much does natural occurring variation is used during rapid evolution? So we definitely live in an age of rapid evolution. The world is changing right before our eyes, and we're seeing species change. They're evolving, they're increasing in frequency, they're undergoing this arms race essentially with us as we're trying to combat them with drug therapies and antifungals on our crops. In natural populations, there's a lot of variation. And I think it's still an open question of how much of that natural variation is used when species undergo a rapid change. And so in this cartoon, I'm just showing that there is this polymorphism for meaningful mutations in a population. And that polymorphism often is maintained by frequency-dependent selection. So an allele that maybe actually is beneficial in a species on a particular environment may have a cost or be less beneficial relative to the other allele in a different environment. And so you can imagine that because the environment is heterogeneous, and say like we go into a mixed hardwood forest, these alleles are maintained as polymorphisms, and they may provide fodder for rapid evolution. On the other hand, there are mutations that that might only occur under strong selection. And so a clear example of this could be drug resistance alleles. And those alleles are very costly potentially for the phenotypes that they generate. And so in the absence of that environment, you may not see them in the population. And it is only after the development or the application of the drug that those mutations, which have a huge benefit in that environment, can arise and drive evolution. And so the open question in my mind is how much of both natural occurring variation and de novo mutation is required for rapid evolution, as this is going to allow us to predict the evolution of these rapidly evolving populations. The first species I'll talk about is Schizophilum communi. This is one of my favorite organisms. It's a model species, a mushroom. You can get it to fruit in the lab on a petri dish. And it's really thriving in the Anthropocene. So it's a disturbance-loving species. If you want to find it, you can go to like a clearing in the middle of a forest. And there's a footbridge with it growing on it. And here's urban environment in Brazil where you see the species thriving in the middle of all this garbage. And it's cosmopolitan, and it's found on a diverse amount of wood. It's cosmopolitan, but there's strong population structure. And we developed this population genomics study where we sequenced the genomes of some strains from North America and some strains from Russia, and we compared their genomes. And it was pretty obvious when we looked at the genetic structure that the two populations were not really undergoing genetic exchange. So here's our North American populations in this kind of pink, and then these green are the Russian populations. They're completely separate, and there's very little structure within a population. But there's an incredible amount of divergence or diversity. And so that was one of the things we found that was really striking. And we tried to publish a paper that suggested that this species, the split-gill mushroom, is the most genetically diverse eukaryotic species described to date. And so here's a figure basically cryptid from Lynch and Connery, where they kind of scaled all these different organisms in terms of their genetic diversity. And this data comes from basically looking at silent sites in exons. So, you know, these are sites that shouldn't have much effect on the phenotype of the organism. And you can use this as sort of like a benchmark for all the diversity, we found that the North American population of the split-gill mushroom was as diverse as any other eukaryote. And it's very diverse, and it's also highly recombining. So if we looked at linkage disequilibrium, so essentially the association of sites in space in the genome, are they correlated with each other, which is basically the linkage disequilibrium statistic. That linkage disequilibrium, or that statistical association of sites, disappears very rapidly as you go from along the chromosome. And so of all the organisms we could get population genomic data for, we found that the split-gill mushroom had the most rapid decay of this linkage disequilibrium, such that when you went to about 1,000 base pairs apart in space, there was really no association at all. So essentially it looked like the sites were randomly mixed. So this is a highly recombining, highly diverse species. Now we wanted to then study how the diversity impacted recombination at the local level. And to understand how that heterozygosity impacted crossing over, we took these two different populations and crossed two parents. And they're incredibly divergent from each other. So this diploid we created had 14% heterozygosity. So basically 1 out of every 7 sites was different between the two. Yet they can undergo plenty of crossing over. So what's shown here are, it's about 17 segregants, haploid segregants that we took from that highly diverse heterozygous cross. And we sequenced the genomes of those 17 segregants. And then what's plotted here is essentially the scaffolds or chromosomes, and each one of these colors is for one of the parents, so that either the Russian parent or the North American. And you can see that there's a lot of recombination going on here. So recombination is not inhibited in this species, despite this incredible diversity between the two parents. And what was, we then looked to see at where those crossing over events occurred. Was there anything special about them? And with this high level of diversity, you would imagine that because of this anti-recombination mechanism, which prevents homologous recombination when sequences are divergent, that we might see lower heterozygosity in the areas near the crossing over. And that's exactly what we saw. So here in these more bright lines are the two different, there's the Russian and then there's the North American population. And this is the diversity near the crossover event. And so there is this depression of diversity about 500 base pairs away from the crossover event. And so there's lower variation there. And what we found is that lower variation is highly significant relative to what we'd expect randomly, these other lines. And that that variation depression was primarily located in exons, so that crossing over mostly happened in exons, and it was also had the signature of purifying selection. So selection is essentially reducing variation in these species, and it's happening independently in the same genes in Russian and North America, and it's creating essentially this ability to the species to recombine and stay cohesive. I'm going to skip now and kind of embracing this tree of life concept to talk about a completely different fungus, and I'm talking about the amphibian killing chytrid. This fungus looks like this in pure culture. It reproduces with a swimming spore, and it attacks the keratinized parts of amphibians. And here's this region of a jaw sheath that has been depigmented because of colonization. It's primarily on adult skin, though, where it causes an issue, and it's found globally, but causes severe declines only in specific regions and in specific species. I'm going to just highlight an observation that we made when we were using population genomics to understand this, the origins of the species. What we found is that where declines were occurring, there was a single genotype or related set of closely related genotypes that we were later named the global panzootic lineage, so the BDGPL. And there are other lineages or strains out there. But one of the things that immediately caught our attention was that if we look at the heterozygosity in the genome, and this organism is diploid, that you have, and so this y-axis is heterozygosity, you find regions of the genome that are completely homozygous, so this chromosome 2, and they're homozygous because they share an actual event of loss of heterozygosity. And we infer that this is due to, this is occurring during mitotic recombination or during mitosis. So this led me to start to pursue the question of how often is loss of heterozygosity during mitosis adaptive? And what is the relationship between heterozygosity and the ability to recombine and the process by which recombination occurs? And you can't study that in this amphibian chytrid, so we switched over to looking at model yeast species. And this is the basic design of the experiment. It's an experimental evolution approach where we had constructed the strains from known ancestral haplotypes and evolved diploids. And we did that for 100 passages. And then we looked at these replicate populations and sequenced individuals from those populations and tried to look for evidence of parallel loss of heterozygosity, which might suggest adaptation. The heterozygosity in these two different species is quite different, and that leads to totally different processes by which they undergo loss of heterozygosity. So in Cryptococcus, which has about 0.1% heterozygosity, you see entire chromosomes that undergo. In this graph, we have here's the haploid ancestors, and then these are the diploid ancestors before evolution and after evolution. These regions of blue and red are regions where loss of heterozygosity has occurred. And so, like, for example, in chromosome 2, the entire chromosome has undergone LOH, and it's always in the same direction. And Saccharomyces, well, where the locations of breakpoints in the genome in Cryptococcus were strongly determined by the levels of heterozygosity. So these green boxes here indicate where crossover events occurred, and the blue indicates heterozygosity. And so this is high heterozygosity, this is low heterozygosity. And you can see that the crossover events are mostly where heterozygosity is low. So there's a strong relationship between heterozygosity and crossing over, as we might expect. In Saccharomyces, we have something quite different. We don't have chromosome level loss of heterozygosity, and we have environment-specific LOH, and we were able to show that this environment-specific LOH is due to a very specific mechanism and a very specific genes that are driving the favoring of one parental allele. And I'm sorry that I ran out of time to discuss this in more detail. But we were able to show that this particular event is driven by selection and it happens relatively rapidly through this course of the 100 days of evolution. So the take-home messages are that even extremely heterozygous genomes can undergo recombination, but the locations at which they undergo recombination are determined by the level of heterozygosity, and in part that level of heterozygosity is determined by selection, in this particular case, negative selection. And we were able to show in some cases LOH is adaptive and specific to the genotype, and that these anti-recombination mechanisms are important for considering the way in which these genomes undergo loss of heterozygosity during mitosis. And that I'd like to wrap up by acknowledging the people who helped, particularly Lucas Michelati, who did most of the serial passaging and the schizophyllum work, which was done in collaboration with Alex Kondrashov and Sheng Sun, who set up some of the important crosses for the LOH project. Okay, thank you very much. Happy to take any questions.
Tim, thanks a lot. And while people are thinking about questions, I'd like to start off with one from the first story. When you mentioned that most of the recombination occurs during exons, I was really struck by that because that's not the way I normally think about it in some eukaryotes. But what's the genome structure like in these mushrooms? Are they as compact as baker's yeast?
Well, for the split gill mushroom, not so much. But it's still pretty small relative to even Drosophila. We've got about like a 34 megabase genome. That species has a lot of introns, and what I think the reason that this pattern occurs is that the intergenic regions in an organism that's this diverse are not constrained by selection. And so those regions get highly heterozygous, highly diverse. And it's only because the exonic regions are constrained by this purifying selection that they actually become sort of these landing pads for where recombination can occur. Introns were even more likely to have recombination than the intergenic regions, but indeed these are compact genomes.
Thanks again for the great talk, and yeah, and always enjoy hearing about your work.
Yeah, thanks for the opportunity.
Okay, and so the last talk for the symposium is from my colleague in molecular cellular developmental biology, MCDB, Cora MacAlister. And Cora, like Tim, started in as a plant biology major, but at Michigan State University. She then went on to do her graduate work at Stanford in Dominique Bergman's lab, looking at cell fate decisions in stomata using Arabidopsis as a model system. She then did postdoctoral training in Zachary Littman's lab at Cold Spring Harbor, where she looked at the timing of flowering in tomato. And then joined the faculty at University of Michigan in 2015, where she's currently an assistant professor in MCDB. And while she was at Cold Spring Harbor, she got interested in a family of plant-specific glycosyltransferases that add ribinose to secreted proteins. And Cora and her co-workers have used the Arabidopsis and the moss Fiscomitrella, hope I'm pronouncing that right, to show that these enzymes are critical for cells to make extensions. And that's a particular challenge for plant cells that have a relatively rigid cell wall. And I'm sure she's going to tell us about this in more detail and some of her really interesting genetic observations that they've made. So before we listen to Cora's pre-recorded talk, I just want to again remind everyone to use the Q&A feature to add questions, and then for the questions that are posted to use the thumbs up icon to prioritize the questions after the talk. So Cora, are you ready to?
I am ready. So thank you for the very kind introduction, and thank you for the very kind invitation. And it's actually pretty rare for two plant biology majors to have talks right after each other. So I learned something new about Tim today. But with that, I will switch it over to past Cora to give you her talk. Hello, I'm Cora MacAlister. I'm an assistant professor in MCDB here at the University of Michigan. And my lab is really interested in how organisms coordinate their insides and their outsides. And by their outsides, I mean the extracellular matrix that they produce to surround their cells. The ECM serves several important functions, and really all organisms produce an ECM of some sort. It provides structural support for individual cells and tissues. It also helps glue neighboring cells together and coordinate within tissues. And it serves as a site for signaling molecules to move from one cell to another. But not only does it have this very important function, but the ECM is also dynamic over time. So the ECM will change and remodel during development and aging. And the composition of the ECM will determine various mechanical properties of cells and tissues. In plants, which is what my lab works on, the ECM is the plant cell wall. And the plant cell wall is a complex composite of multiple interacting polymer systems, including various types of proteins, glycoproteins, and carbohydrates that interact in this space. And in order for a plant cell to grow, it needs to be able to push against its cell wall and expand its cytoplasm. And when this happens, that results in a thinning of the cell wall. And in order for the cell to maintain its structural integrity, it needs to be able to reinforce that cell wall as cell expands. And I've drawn it as two separate steps here, but this is really a continual process. As the cell expands, it's reinforcing the wall that is thinning at the same time. And if there's a failure of this process, the cell will become too weak and the cell will rupture if it's too thin. And if it's too thick, that prevents the cell from expanding further. So it's not able to mount sufficient pressure to push against the force of that wall counteracting its expansion, and that will arrest cell growth. And in the context of a multicellular organism in a tissue, neighboring cells need to be able to coordinate their expansion. So here's an example of an epidermis in a Arabidopsis, and you can see these large puzzle piece shaped pavement cells that have been produced by the expansion of what were formerly much smaller, more or less isodiametric cells into these large lobe cells. And so the neighbors need to be able to coordinate their expansion to make a nice flat continuous tissue. And this is a fairly complicated process when it's happening in a multicellular organism where you have a lot of things expanding at the same time. So my lab studies this process in a much simpler system, specifically tip growing cells. And in our case, particularly the pollen tube. And so in a tip growing cell, you have a spatial distribution of growth activity. So you have a tip region where you have an extensible cell wall. This is where all of the actual growth happens. And then you have a more rigid, reinforced shaft cell wall. And in order to expand, you have to deliver new cell wall material to this extensible cell wall region, and then the old cell wall will get displaced and eventually get converted into this more reinforced form. And that new cell wall material is delivered primarily by secretory vesicles, which carry pectin, glycoproteins, and cell wall remodeling enzymes. And the nice thing about pollen tubes in particular is that not only do they have this tip growing cell organization, but they also grow very, very quickly. So this is not quite real time, but these cells are the fastest growing plant cells. And you can see you have this region near the tip, which is very rich in secretory vesicles, and then you have the other organelles that are sort of farther back within the cytoplasm. So all of this continued growth requires the maintenance of this polarity in cell wall structure between your extensible and your reinforced cell wall. And we know a good bit about how this organization occurs in the pollen tube. So we know some of the polymers that are required for reinforcing the cell wall shaft region. So here's a polymer which becomes more abundant as you move away from the tip. Here's another polymer which is excluded from the tip and then starts appearing in the shaft region. And we also know other polymers that are associated with this more extensible type of organization at the tip. So here's another polymer which is enriched at the tip, which has a more extensible sort of behavior. And So we know something about what happens when you convert from this extensible form to this more reinforced form. So you have a rigidification that happens on particular polymers as they get displaced. You have further reinforcement, which is the deposition of new polymers along the shaft. But we really don't understand much about what's happening at the tip itself. So how do you replace the cell wall as it's being converted to this more reinforced form? And we know this has to balance the rate of cell expansion over time, so how fast the pollen tube is actually growing, how fast it's able to secrete these new wall materials, and we know we have to also maintain cell wall integrity, because if this tip region becomes too thin, the cell will actually rupture. And so my lab is trying to understand how this process takes place, and we primarily use genetic tools to do that. So here's an example of a mutant in which cell wall polarity has been disrupted. So along the top here, this is the wild type polymers I already showed you. And here are the same polymers as they are disrupted in this mutant. So you can see that you no longer exclude these reinforcing polymers from the tip, and you no longer enrich this more extensible polymer at the tip here. And so you go from a nice polar distribution of polymers to a much more uniform distribution. And this has some pretty serious consequences for the growth of the pollen tubes. They're not able to elongate as well, so they don't achieve the same lengths as wild type pollen tubes when we grow them in vitro. And they're also very prone to rupture. So they're not able to maintain structural integrity, and they will actually pop and spew their cytoplasm into the growth media. And this particular mutant occurs in an enzyme called a hydroxyproline O arabinosyltransferase, or HPAT. And these are required for the production of a specific type of post-translational modification that occurs in plants, but not in other types of organisms. And the structure is something like this. So you have a proline within a protein or peptide backbone that gets converted to a hydroxyproline, and then you add a series of arabinose sugars in this linear form. And there are a number of enzymes that are responsible for this modification, including the HPAT. So these add the first arabinose. So in the absence of HPAT activity, you're not able to initiate the arabinose chain, and so you don't form this modification. And this modification takes place most commonly in a particular protein sequence context, which is a serine followed by a series of prolines. And this sequence context occurs most often in proteins that are secreted cell wall associated structural proteins of the extensin family. And here's a sequence of a typical extensin. There's a signal peptide, and then I've highlighted again this glycosylation motif in teal. And you can see it's a very repetitive protein. So there's a lot of proline here to be arabinosylated. And I've also highlighted this tyrosine cross-linking motif. So these tyrosines are able to form covalent bonds, forming intra and intermolecular cross-links between other extensins. And so in this way, they can form a dendritic network of covalently cross-linked protein, at least in vitro, and presumably in the wall space as well. So what do we think is actually going on here? Why is this so important in pollen tube growth? Well, if you look at the distribution of this modification using a specific antibody, you can see it's localized in the cell wall generally, and particularly around the tip region of the pollen tube. So what we think is going on is that we have modified these extensins by the addition of these arabinose side chains, and that helps the protein assume the appropriate conformation and encourage its cross-linking. So these are secreted as monomers. They get cross-linked within the tip region cell wall space. And these extensin proteins have an overall positive charge. So they are able to electrostatically interact with pectin. And pectin is the most abundant polymer in the cell wall. And it is the primary constituent of the tip region as well as the shaft region of the wall. So what we think is happening is that we're forming this extensin scaffold, which allows the pectin to assemble to form temporary structural support for pectin formation. Over time, the pectin will become directly cross-linked between other pectin monomers, forming by calcium salt bridges. So we have this temporary scaffold, which allows enough space and reinforcement to form this more permanent scaffold as the wall moves towards the shaft region. And so with that, I would like to thank the members of my lab who've done really fantastic work on this and other projects, particularly graduate students, Roop, Stephen, and Cecilia, and other various temporary members of the lab. Also thanks to our collaborators and funding support from NSF and MCDB. And with that, I will send it back to future Cora to answer any questions you may have. Thank you.
Okay, before opening the floor for questions, and we have 7 minutes or so, so hopefully we can get a discussion. But I have 2 questions, Cora, or future Cora, if you don't mind. The first is I love these pictures of the different pectins and the extensible MGHD pectin that's just at the tip. But if the system is so dynamic and it's the localization at the tip, then there must be a lot of turnover. So what's known about how you get rid of this particular pectin?
Yeah, so that form of pectin actually gets converted to the other form of pectin. So MEHG is methyl esterified homogalacturonin. It's the long, complicated name of that particular type of pectin. DMEHG is D-methyl esterified homogalacturonin. So you essentially take your methyl ester groups and you remove them enzymatically through these pectin methyl esterases that are also secreted into the cell wall. So you have these methyl side groups that prevent the formation of calcium salt bridges between adjacent monomers. So just by removing those, you can rigidify your pectin essentially.
Okay, great.
So if you make jams or jellies at home, you have to be careful about acidity and certain things to make sure you get a nice gelling going on. And that's essentially what's going on in the cell wall. You have this enzymatic conversion. So initially they're secreted in this very extensible state, and then they get converted into these cross-linked forms.
Okay, fantastic. And I have a question from the Q&A from Bingye, who says, very interesting work, Cora. And the question is, are Golgi apparatus located near the tip?
So there are, they're spread sort of throughout the pollen tube. So there's this clear zone, what's called near the tip. And that is mostly entirely secretory vesicles. There's also a very fine actin network in that region. So that's where a lot of membrane dynamics is happening. So you have these secretory vesicles getting secreted, and then you have endocytosis to recycle this excess membrane. The Golgi tend to be excluded from the clear zone and more further back, so where all of the mitochondria and ER and kind of everything else is. And the distribution of that, it's actually not quite as nice and organized as it is in a lot of animal cells, where you have this localization around the nucleus and then ER and then Golgi right there. So the Golgi tend to be more dispersed in plant cells and generally, and that's also true in the pollen tubes.
So just to pick up on Bing's question, and maybe Bing, I don't know, you're thinking about neurons, and I'm also thinking about that, where the secretion is really localized to the tip. And Cora, do you know in the pollen tube if the rate of secretion is higher at the tip than along the shaft?
Yeah, so these secretory vesicles are being directed towards the tip, and I'm always very careful to say the tip, because there's actually a debate in the field about whether it's to the very, very tip of the tip or whether it's sort of around the periphery of the tip. And I think it's likely that certain secretory vesicles are being directed to different places, and this is likely going to explain the sort of contradictory observations about exactly where secretory vesicles go. But definitely the bulk flow of secretion is happening towards the tip region. There's also some evidence of potential direction more along the, what we would call the subapical region, so more along the shaft. I try not to make too many obscene hand gestures describing the pollen tube, but it's a little tricky. So you have this sort of ring that is where the shaft meets the tip, and there's some evidence that there may be some secretion happening there as well. But it's mostly directed towards what we would call the tip, sort of generally.
And I have a question from our MCDB colleague, Wang. who writes, in animals, hydroxyproline is highly enriched in collagens. And is this a coincidence, or is there something more in common?
I think it's likely a coincidence. It's so the evolution of the extracellular matrix is actually a really interesting question. So it seems that the ECM has evolved more or less independently in all of the major lineages. So the plant ECM and the animal ECM and the fungal ECM all use sort of different basic polymers. So in plants, it's a lot of cellulose and pectin and glycans and things. In animals, it tends to be a lot more glycoproteins and proteoglycans. And that might be a consequence of just, if you're a plant, sugar is very cheap. You just make it out of sunlight and water. So you can build a cell wall out of sugar, and it's much less expensive for you than trying to build a cell wall out of a lot of protein. And in animals, that's not quite the same situation. So this evidence that hydroxyproline is in both of them, I think it could be telling us something about either just the utility of hydroxyproline and the way it influences conformation on proteins. I can tell you that in plants, those hydroxyprolines get glycosylated to a much stronger degree than that happens in animals. So I talked about the arabinoxylation, but there's also another large type of glycosylation that can be added to hydroxyprolines in animals. So I think it's just probably two different ways of getting proteins to be sort of more or less elongated in shape using different systems.
And I guess I have one more question, and it's about the extension connection. That I'm not a plant biologist, but my understanding extensions are famous because the mutants don't bend towards light. Hopefully.
So the extensins are actually, we've known about them since the 1970s. So they're really abundant in the cell wall, and there was these early biochemical characterizations of them. There's the extensins, there's the expansins, and then there's other things with similar names. So every time I give this talk, someone asks me a question about the expansins, but those are different.
I fell into
that. You may have fallen into that. So the extensins for a long time were just thought to be these sort of cross-linking things, and they're responsible for pathogen response and other sorts of, we got to lock this down right now, sort of cell wall responses. Our evidence shows that they're much more a part of sort of the normal growth of plant cells, and they have this sort of more fundamental form. So I think they're likely to be pretty central to plant cell growth, and also they're generally in pretty large protein families. So in Arabidopsis, there's 20 of the canonical extensins, like the sequences I showed you, and there's probably about 60 proteins with an extensin region with some other functional domains. So they're pretty big protein families, which kind of complicates the study of them directly. So using the glycosyltransferases is kind of a workaround to try to study the function of these proteins if we can't just make, whatever the 20-fold mutant would be for these.
Penny has the last question, trying to rescue my faux pas. And Penny's question is, how does the tip know it's the tip? Or the tip of the tip? Can that identity be altered by physical properties of the adjacent ECM?
That is a very, very good question. So we know that the tip can actually reorient its growth. So as the pollen tube is growing in the female tissue, it actually has to respond to these guidance cues. So it will be growing along, and then it will sense an ovule over here that it would like to fertilize through chemical signals and possibly physical signals, and it will actually reorient its growth to turn. And those turns can be quite abrupt, actually. So we know that there's at least some flexibility in this tip region that allows you to sort of reorient the direction of secretion and sort of remodel the cell wall there. So most of the time, the tip region knows it's the tip region because it's been the tip region for at least a while. So if you try to, there's a bunch of ways you can mess up the tip region and stop its growth. And normally that's sort of the end of the thing. But what I didn't have time to talk about is that in the HPAT mutants, you actually have sometimes the addition of a subapical tip. So these guys will be growing along and they'll arrest for a while, and then they'll start a new tip from down in the shaft region. And that's a really, really rare phenotype. That almost never happens in mutants. So there's something about the cell wall in these mutants that is, they're actually able to start growing again from a new point. So the question of how the tip knows it's the tip and how it maintains its identity as a tip is a really good question, but it's a little bit.
Oh, okay. For the sake of time, even though it's a great discussion, I need to end it there, but I hope everyone can thank Cora along with me, and thanks for all the great questions. But at this point, I want to yield the floor to Roger Cohn for concluding remarks of the symposium.
Thanks, Ken. I want to thank all of our speakers, both from yesterday and today, for a remarkable series of talks. You know, I was looking on the NIH website, and right now, about 50% of R01s, new R01s, are for research in the mouse. And of course, the mouse has been an incredibly valuable model system. But I think what we learned from the last 2 days is that there's an amazing amount of biological diversity out in the world. I loved hearing from Raymond St today that there's not only 12,000 species of ants, but 12,000 different species of fungi to infect those 12,000 different species of ants. So, you know, whether it's the amazing amount of genetic diversity in Africa in the cradle of civilization, or the cradle of humankind, to diversity in ants and diversity in fungi. There's an incredible amount of diversity in the world, and we can learn from that diversity. We can learn about the evolution of learning from facial recognition in wasps, as we learned from Elizabeth Tibbetts' talk. We can learn about how to make soft robots from studying mobility in Manduca sexta. The discovery by Sarah Tishkoff, essentially, of the amazing amount of genetic diversity in Africa will not only help address ultimately health disparities, but will also, across different groups of people, but also allow scientists to better understand genotype-phenotype relationships in humans. And lastly, all of this diversity, of course, is important for advancing biomedical research. And I thought I was tickled by Dr. St. Leger's talk, in which, you know, he was essentially using information from 5 different species to deal with malaria, taking toxins from scorpions, putting them in the right species of fungi to infect a mosquito that carries a parasite that infects humans. I think he gets the award in this series for the most number of species used to attack a biological problem. But in any event, I want to thank all of our organizers, Dr. Chengyu Li, Dr. Beng Yi, Ken Cadigan, and colleagues at MCDB for putting together such a thought-provoking and diverse group of talks for this year's Life Sciences Saltiel Symposium. And I look forward to seeing all of you at this same time next year for next year's symposium. Thank you all very much, and have a good afternoon.